He did his postdoctoral research from Oxford University, UK from 1996 to 98. And he was also as a bioinformatics specialist at UAMS USA from 2003 to 2006 and visiting professor at Postech South Korea. He worked around 30 years at different scientific positions in field of bioinformatics at CSIR IMTech Chandigarh. Raghav's group has published more than 250 research papers in internationally reputed journals. In 2014, he has been listed in the world's most influential scientific minds in the field of computer science. His group developed more than 300 in silico products, that is web servers, databases, and software packages, which is highest contribution by a single group in the world. He's a strong supporter of open source software, web servers, and he had more than 150,000 hits per day to his website. So you can understand that how many people are getting benefit from his research. He got all major awards like Santi Swarup Bhatnagar Award in Biological Sciences in 2008, National Bioscience Award. He is a JC Bose National Fellow. He got major recognitions like FNASC, FASC, FNA. So all kind of recognition what a scientist can get, he has everything. Welcome, sir, and over to you.
Thank you, Evan. Thank you very much for inviting me for the talk. So today talk will be more on the protein-based therapeutics. Normally we have the therapeutics based on the small molecules actually. So today I'm talking about the protein-based therapeutics actually. How we can use the protein, mainly biological molecules for the research actually. That's the whole purpose. So I will start my presentation. My presentation is visible. Is it shared?
Sir, not yet. Yeah, now it is. Now it is coming, sir.
Okay. Is it visible now?
Sir, if you keep it in the slide mode.
No, it's in the slide mode now.
Okay, now it is visible, sir.
Okay. All right. So as you can see the title, the computer-aided protein or peptide-based therapeutics. So protein, peptide or polypeptide, they are same thing. They are made of 20 natural amino acids. Only different is size actually. If it is a limited number of amino acids, then we call peptide. If it is more, then we call polypeptide. If it is even bigger than 100 amino acids, we call protein actually. So I will use these words exchangeably. So sometimes I will call protein, sometimes I will call peptide basically. But you know the meaning is same actually. So we all know the discovery of the drugs basically, how the drugs have been discovered. So because this whole workshop is on the drug discovery, so I will not go into the detail that initially the drugs come from the plants, then it comes from the simple observations, and then it's basically based on the genomics and proteomics, based like receptor-based drug design or ligand-based drug design. There are different way of designing drug. But in history, most of the drugs, they are mainly based on small chemicals. And there is a reason, because small chemicals have the stability as well as cost cheaper to make, number of other advantages actually. That's why most of the drugs in the market, they are based on the chemicals. Okay. But there are a number of biological molecules which are heavily used nowadays for designing the drugs basically. So today I am taking only one molecule that's called protein basically. I'm not taking the other molecules like RNA, DNA, or maybe carbohydrates or lipids. I'm taking protein. Why I'm talking protein? Because you are all biologists, so you understand protein is called the servant of living organism, and it is everywhere in your body. Means in any living organism, it plays wide number of roles actually, whether it's related to the innate immune system, adaptive immune system. So protein is an important factor. So most of the drug, even if you are designing a small chemical based drug, the drug target is normally protein. Normally it is protein. Okay, so you basically design a small chemical which can inhibit the protein target, right? But sometimes it is difficult to design the chemicals which can overcome some limitation of the protein. Let's say some defective proteins is there. Then how you will design the drug to replace the defective proteins basically. Then you need another type of drugs which can remove the defective proteins or which can improve the function of the protein basically. The small chemical is not possible to design it. And good example I can say, which is heavily used till today, is antibody. Antibodies are heavily used nowadays for the number of diseases actually, particularly cancer and other diseases. So before I go further, I will focus on this database. This database is developed to collect and compile all the informations related to the therapeutic peptides and proteins discovered so far actually. So far means this database has been developed in 2017 actually. Up to 17 is there. We have to update this database further. But I'm talking about up to 2017. What we did, we collect all the informations from the FDA. You know very well that any drug, if it has to go to the market, international market, then it has to be approved by the FDA. So we collected all the information from the FDA, and we only take the information which are related to peptide or proteins basically. And that's why we call the therapeutic proteins basically. And these are the FDA approved drugs actually. So there is a different type of FDA approved. We only focus on the protein and peptide. And this is a website. If you have the time, you may use it because this is a workshop. So you can use this site. This site is available to the public, right? I'm giving just the statistics about this database. This database has total 673 entries, and it has the 160 FDA approved proteins or peptides basically, which is basically patented in 165 patents. Literature we collected from 552 research papers. Drug variants. It's not just collection of information from FDA. In fact, it is a comprehensive information compiled from the FDA as well as from other resources. FDA is the first initial search where we get the information about FDA approved protein peptide. Later on, we extract, let's say, sequence of the protein, structure of the protein from the different sources, or we predict the structure of the protein using the structure prediction methods. Okay. So this is not, you will not get this information from any other resource except this THPDB actually. And it provides the complete information. You can see the field from right side, that name, sequence, functional classification, molecular weight, blah, blah, blah. Okay, so complete information you will get from here. The question is, you will ask from me that what type of protein and peptide based drugs are there actually? Because that's an important question. So first group, which is basically therapeutic with enzymatic or regulatory activity. So these are the basically designing of some enzymes or some proteins which have some important activity in the body. and these can be further classified in the group 1A, B, 1A, 2A, 3A. It means replacing, let's say first group, replacing a protein that is deficient or abnormal. Let's say, for example, you are unable to make the insulin actually. So what we are doing, we are giving the insulin from the outside so that your defective protein, defective polypeptide will be removed, and that new polypeptide will be used to digest the sugar basically. Okay, same way there is a augmenting an existing pathway. Let's say some pathway, it's not working fine actually, and some of the proteins are not working fine. Then you can give the protein which can help to activate this pathway, or to make it more efficient basically. Third is providing a novel function or activity. Maybe you want to include or insert a activity, providing novel function activity, then you can replace the protein. So here the idea is to design a protein, whatever maybe enzyme or protein, and replace the defective one in your body actually. This is a group 1 type of therapeutics, and number of proteins and peptides are there in the market actually. Another is basically therapeutics with special targeting delivery. You know, one of the challenge in drug discovery is delivering the drug at the right target. What happens? Sometimes you design the drug is perfectly all right, but it's not reaching on its target. Then it's again useless actually. So there is a different ways to deliver a drug on the right place. Okay. So here they are mainly designing the proteins which can deliver the drug at the right target. And one of the example I can give you is cell-penetrating peptide or cell-penetrating proteins. These peptides have the capability to enter into the cells. So if you want to deliver a drug inside a cell, then you can attach this drug with the peptide, and peptide will not only enter into the cell but will deliver the drug inside the cell. And nowadays this technology is used heavily to deliver the drugs, because these peptides or protein based drugs, one advantage they have the less toxicity because they are made of natural amino acids, so it is digested easily after some time actually. So the another part called ADMAT, this has the good advantage. The toxicity is less in designing the natural molecule based drugs. Third drugs we all know very well, and that is vaccine basically. Third group of vaccine is basically vaccines, or you may consider the antibodies here. Okay, so these are basically related to activate our immune system or to design the proteins which work like as an immune system basically. And you know very well, nowadays everybody is working on the coronavirus vaccines actually. So that type of vaccines come under this category, where you design a peptide based. Let's say in most of the protein vaccines which are designed for this coronavirus, against the coronavirus, they are based only on spike protein, because spike protein is surface protein of coronavirus, and which is given as a vaccine actually in most of the vaccines. So this group is heavily used from long time and responsible for removing the number of disease from the world actually. And we have the small we have the examples of smallpox or polio, which had been eradicated from the world actually. Fourth group of peptide or protein based drugs are basically used in the diagnostics actually, because what happens in certain cases of disease, some proteins or peptides are released in your body fluids, maybe in the urine, maybe in the blood, or maybe saliva, number of other body fluids actually. and we can use these peptide protein released in the body fluid as a diagnostics actually, or called biomarkers basically. So this is a fourth category. So these are the major categories where the FDA have approved the drugs for the use actually in the market. Some facts I would like to discuss, because in the market it has been shown in the statistics that 84% of biologics, because we don't have the specific data for the protein, but 84% of biologics clear phase 1 to phase 2, where in case of the small molecule it is only 63. Okay. Similarly, in case of phase 2 to phase 3, only 53% for biologics clear, where in case of a small molecule it is 38%. Same is true from phase 3 to FDA approval. So it is clear that peptide or protein or biologics based drugs have the better chance to clear the clinical trials rather than the small chemical based drugs actually. And there is a huge market. So if you are interested in protein or peptide, then you can use as an alternate to the small molecules. Because I know this workshop is mainly focused on the drug discovery or drug design, and I'm talking about the another alternative actually. That's basically protein. I would like to point out here actually. Our group from beginning always searched some alternate systems actually. Though initially it looked difficult, but later on it helped you to create a new field basically. So you are already doing a lot of work in the field of small chemicals. If you try this new field called protein peptide, the chances of success will further improve actually. Or you may mix the two actually, because one of the reasons that these protein or peptide based drugs fails because they have a limited half-life, or their delivering is the problem. Then you can attach some small molecules which can increase the half-life or stability of the protein and peptides. Okay. So now I will talk about vaccines actually. Vaccines is an important factor actually, and they are heavily used over the time, and the vaccine have the advantage that it gives you the protection for the long time actually. Let's say we all know about pandemic actually. That's why we are on the online actually. If this pandemic was not there, probably I will interact with you physically actually. I was giving in the big auditorium the lecture and you are listening to my lecture actually. But due to this pandemic, we are doing all online lectures actually. Okay. And when this coronavirus comes, then what we are expecting from long time? To get the vaccine. Because drugs are not sufficient actually. We need the vaccine which can provide the protection actually. Why we are not discovering the drugs? Because we know it's difficult actually. So, idea here is just to explain to you that proteins or peptides are very important for any medicine actually. So you should consider it as one of the possible alternatives. Let's say now I will talk about the vaccine actually. So let's say just think about it. These are the different pathogens. So the pathogens have the problem that they cannot enter. There is a layer, human host have the layer that is called skin actually. Once they cross the skin, then there is a biochemical barrier which are released some certain chemicals which provide the protections. And then there is a innate immune response, or we call innate immune system. This innate immune system have the receptors called like toll-like receptors, or in simple word we call PRRP, pattern recognizing receptors. They normally recognize these cell membrane of bacteria, or fungus or virus. So they recognize it and then they release certain type of chemicals. It means they are releasing chemicals without bothering what type of enemy is there. So it's not a specific to any type of pathogen actually or disease causing agent. They are basically in general by nature. So that's why it is called the non-specific defense, or this is also called the first line of defense actually. So still some of them cross the first line of defense. The one thing, the innate immune system provides a good thing, that once it is sensed that enemy is there, it gives a signal to the adaptive immune system and it activates it, because you cannot keep all the immune system activated form actually. So it gives a signal to adaptive immune system be activated. There is an enemy have been entered inside the system basically. Then the adaptive immune system come into the picture. Adaptive immune system has the property, they recognize the particular type of pathogen and accordingly they generate the memory based B cells and memory T cells basically. So these cells, there are two types of cells they are generate, plasma cells and memory cells actually. Plasma cells help to clear the pathogen, and memory cell remember it for the future fight actually. Maybe you are fine now, your system has overcome the some pathogen. Okay, maybe after let's say 5 years later on, you got infected again. So you have the memory cells, they know it, what type of enemy is there and what is the best defense for it actually. That's why this is called immunization actually, where it's nothing, it's basically memory cells, increasing the number of memory cells which remember how to handle this particular type of pathogen basically. And one of the region, though we are talking that US or Europe are suffering actually more from the coronavirus than the other countries like India, Bangladesh, Pakistan, or some African countries. It has the reason behind it actually. Reason is simple, because these countries, we are already exposed to large number of pathogens, bacteria, virus, and other things. So we have large number of memory cells. Means our immune system have already challenged, have got the already exposure to different type of disease actually. So we have generated the large number of B cells, T cells, as well as antibodies, which can fight the wide range of pathogens basically. So it's not that we are very careful and we have a good treatment on it. It's there because our immune system has been exposed very well. Okay. So this is a layer we have to understand. So this is an important slide for anyone who wish to work in the field of vaccines. And please remember, I'm not going to talk about traditional vaccine. And traditional vaccine, you are basically taking a pathogen, killing it or annotating it and giving it to the body basically. So immune system thought that enemy is there. In reality, there was no enemy, but defense will be there. Okay. So what I'm talking about today is how to minimize the size of the vaccine candidate so toxicity can be removed, and we mainly we are focusing on the peptide-based vaccines basically, which we call epitopes, B cell epitope or T cell epitope. So in system we have like non-specific defense and specific defense. Specific defense normally provide B cell epitope and T cell epitope. I'm not going into the detail of B cell epitope and T cell. I will cover later on actually. Just think about it, that your system somehow have been hijacked by the pathogen. Pathogen may be anything actually, fungus, virus, parasite. Okay, so they may be any one actually. So they come inside the cell. Now because the this pathogen is inside the cell, so this is protected. Because how our immune system will know that pathogen is there? But there is a mechanism. So mechanism is that these pathogens release proteins actually, secreted proteins, and our immune system recognize the foreign proteins, and there is a called proteasome actually. But how the proteasome will chop secreted proteins, that's a challenge actually. Then the number of groups, including our group, have developed the software for predicting the cleavage site actually in any secreted proteins. So we have the software like p-cleavage. NetChop is not our software, developed by some other group. Will recognize it and will tell you that what type of fragment or peptides will be generated from these secreted proteins. Once these secreted peptides are here actually, then some of them can cross the ER if they can bound to the TAP proteins actually. So which one will bound with this TAP can be predicted with these softwares, TAP-RAD, TAP-RAD, TAP-Hunter. These are the different softwares, and some of them will cross the ER. After crossing the ER, they have to bind to the MSC class 1. Not necessarily all will bind. Certain peptide will bind to the MSC class 1 or HLA1. Once they bound, they will go to the cell surface. On cell surface, this peptide, which will come from the secreted protein, will be presented by MSC class 1 molecule in form of the bound form actually. Okay, this bound form is recognized by the CD8 cells actually, or killer cytotoxic T cells. They are already doing monitoring of the system or surveillance of the system. They will recognize it, and after recognizing it, they will release certain type of cytokines, and these cytokines will kill this cell. It means they will destroy the infected cell. That's the concept actually. If any cell got infected and the signal come on the cell surface that the cell is infected, then our cells, killer cells, will destroy that cell actually. So pathogen will also be destroyed in the process actually. That's a so what we are doing, what means we means immunoinformatics or informatics person are doing here actually. So we are developing a software which peptide will be recognized in the bound form or not, which will bind to the HLA or not. This whole process we have developed the software actually to understand the whole immune system actually. Similarly, there is another type of pathogens which never enter into the cell. They are outside the cell in the environment. So they are recognized because some antigen presenting cell, they engulf the antigens actually. Some proteins of the antigen means here proteins of pathogens actually. They are not the self protein, they are proteins generated by pathogens basically. We call the antigen actually. So the antigen will be engulfed by the cell and then it will be exposed to lysosome actually. Lysosome is full of enzymes. It is full of enzymes and it will chop the antigen in the small pieces. Then they will bound to the MSC class 2. Please remember, when I was talking in my previous slide, I was talking MSC class 1. This is a MSC class 2. That's a difference actually in 2 actually. And in previous one, it was MSC class 1. Here is a MSC class 2. And this will be presented on cell surface. So what the message will go here that MSC class 2 bound peptide will be recognized by CD8 4 cells actually. Earlier, I was talking about the CD8 cells actually. Here is a CD4 cells. So CD4, if CD4 cell recognized, it means the message is clear that in environment where these cells are there is a enemy. Enemy means disease causing agent. And once this signal go there, then they release the different type of cytokines actually, or interleukins, which will activate our whole immune system, means B cells as well as these CD8 cells. So this is very important when you are designing the basically vaccines actually. So again, the question is what we are doing. We means in computer science person are doing here. So we are developing the different prediction methods for predicting the different type of cytokines released by peptide actually. So we recognize the pattern, which peptide or which protein region can activate or be recognized by the T cells actually. So that's the question what we call the T cell epitopes basically. And our group is working from last 20 years in the field of immunoinformatics on each and every aspects of the immune system. This is the example that one of our server, which was developed 20 years back, is still heavily used actually. Let's say this is a one antigen. So this is a one antigen sequence, and this is basically one MSC class 2 allele. This is another MSC class 2 allele. Because we have to understand all human beings are not same. They have the different type of MSC class 1 and class 2 alleles. So we have to predict or we have to identify the peptide which can bind to large number of MSC class 1 or 2 alleles, and that is called promiscuous MSC binder. That is what is required for designing the better vaccine candidate actually. So locating the promiscuous binding is a challenge actually. So we develop it and we present in the form of the graphics. So you can see here this region, it is bound, the peptide is binding with large number of MSC alleles. These are the left side, these are number of MSC alleles. So same region will bind to the large number of alleles. That can be a promiscuous binder which can be used by the public for the designing the better vaccine actually. Because sometimes if you design the vaccine, it works for the certain population, it doesn't work for the other population actually. So designing the vaccine candidate which work for the wide population is a challenge, right? There is another challenge that when CD8 recognize what type of cytokine they will release actually. So we develop the methods which help to identify what type of cytokines, whether it's from gamma or interleukin 4 actually. So we develop the methods for predicting the specific type of cytokines will be released by the particular peptide, which we call epitope basically. Same like there is a B cell epitope. Earlier I was talking about, so far I was talking about T cell epitope. There is another class called B cell epitope. Epitope definition is very simple. B cell epitope is that peptide which can activate our B cells, which can induce our B cells actually. That's called B cell epitope. And the peptide which can induce the T cells is called T cell epitope. That's a simple definition. So in case of the B cell epitope, when we start to work on this area, we realize that most of the existing methods, they have not been evaluated on any standard database because there was no standard data sets actually. They are collecting few examples and then they are developing the methods, but there was not a big data set. So what we did, we simply create a database called BCI-PEP actually. That's what we have done the different from the most of the bioinformaticians in the worldwide. Most of the bioinformaticians, they are only interested in developing the prediction methods actually, or using the prediction tools actually. Our group is not only interested in developing a prediction tool, but we also create the resources which can be used by the world. Most of the research earlier, they have not been developed in India. Mainly they are developed in Europe or US and other countries. So whoever is developing the database, they utilize its maximum and then they release to the public. What we start to do, we start to create the primary databases, though it's difficult. It involved a lot of literature search and other things, but it provides you the base, it gives you the depth. So data set is with you, you can develop the prediction method and then you can release to the public actually. So that developing database is an important area actually. Okay. Earlier, the people are using the physiochemical properties for designing B-cell epitope, like hydrophobicity, accessibility, flexibility, surface. Somehow, most of the researchers prefer to use some beautiful terms like hydrophobicity, accessibility, flexibility, surface, area, polarity. These are the physiochemical properties basically. It is not necessary they are highly accurate. We demonstrate whatever the method has been developed, maximum performance, they get the from flexibility that was 57.53. And this is not a big accuracy. Why? It is not big accuracy. Because even if you do the two class classification randomly, you will get the 50%. So 57% is not a good accuracy at all actually. So we demonstrate that whatever the method has been developed up to the 2003, their performance is not better than random actually. And then we develop a method which can combine most of the physiochemical properties. Here was the challenge actually. The challenge is very simple because machine learning techniques somehow drive the rules which cannot be driven by using the physiochemical property of the chemical. Okay. So the question was how to implement the machine learning techniques in the field of B-cell epitope prediction. That is the challenge to the world actually. Nobody was able to understand how to implement because machine learning techniques, they have certain limitations actually. One of the major limitations, they need some data or input in form of a matrix or vector where the dimensions should be fixed actually. Where in case of the B-cell or T-cell epitope, I'm talking about particularly B-cell epitope, the length vary from 6 amino acid to 60 amino acid. So how you handle the situation actually? That's a problem actually. So in order to handle the situation, we go for the mathematical detail, and in mathematics, based on our experience, it says there are a number of problems which cannot be solved directly. So you have to make certain type of assumptions. Though it's not necessary, your assumptions are correct, but that's the only way to solve it. It's like that actually. You have a building in your front, and somebody say, tell me the height of this building. And you don't know. You have no way of measuring it actually. So what you will do? Best way is do the approximations. Divide in certain, let's say, number of floors actually by eyes and see how many floors are there. Then you take the guessing that what will be the height of the one floor and then multiply by the number of floors. You will get some idea. Though it's not necessary, this is the correct one actually. But that's the only solution. So we did the same thing with this method. First time we were able to implement new machine learning technique, particularly artificial neural network. And in artificial neural network, we mainly use recurrent neural network, okay, which help us to predict the B-cell epitope. So nobody was able to predict the B cell epitope, and then we improve the accuracy, because ultimately whatever you do, it's fine, but you have to improve the accuracy of prediction. Earlier it was 57 something. Now it is reached to 65.93. Though it's not very high accuracy, it's still poor accuracy, but increasing the accuracy 6 to 7% is a big thing actually, when nobody is able to do it. This method is so popular actually. It got nearly 900 citations, and this year itself actually more than 150 citations actually, because due to the coronavirus, a lot of people are using to predict the B cell epitope to design the vaccines actually. Similarly, we have the other models for designing the B cell epitope, which generate a specific type or a specific class of bodies, maybe IgG, IgE, or IgA actually. This is required to generate the specific type of defense against a pathogen actually. That's why our group, despite we are working from last 20 years, still we feel we can do a lot, we have done limited, because we are always searching for the new problem. We are not just fixed for one problem actually. We are always looking at what is challenge for a biologist actually, and how to solve it actually. If you think in that term, then you can do the better actually. Most of the researchers, they did the big mistake. They think about themselves actually. They think what is good for them actually. But please remember, you are a researcher. You have to understand the need of the user actually, need of the market actually. Based on that, you have to frame your research actually. Then you will be able to solve more and more problems actually. Okay. Then there is another part. So far, I will talk about the T cell epitope, B cell epitope. They are all part of the adaptive immune system. There is another part is called innate immune system, which recognize the pathogens cell membrane actually, mainly cell membrane. So these are called pattern recognition receptors. We create a database which is available to the public. You can use it, but using the different type of receptors, we design peptide-based vaccine adjuvants. Because one thing you remember, if you are in the field of drug discovery, there is a one word called adjuvants, or simple language we call salt actually. This is very important. It's not doing anything. It's only activating our innate immune system. And that once you activate our innate immune system, it gives the signals to the body, look, there is an enemy. As well as it helps you to identify immunotoxicity. Because sometimes that problem here, basically, sometimes it may be immunotoxic actually. So how to predict the immunotoxic? That this software will help you to identify the immunotoxic proteins or peptides. This software we designed for predicting the allergen, means allergenic proteins, means the proteins which cause the allergy. Let's say nowadays it's not difficult to design the new protein which can be used as probiotics or maybe used as GM food and other business actually. But challenge is how you will know that this protein will not be allergic actually. Because if it is proved allergic later on, it will be a lot of loss actually. So this type of servers help you to identify the allergic protein. So you can modify the allergenic regions in order to remove the allergenicity from the protein. So this is the overall compilation of the softwares developed from our group. These are not all softwares. I have given the major softwares from our group, which are designed for designing the vaccine candidate. And please right, these are the epitope-based vaccine candidates. So far, I talk about vaccine part, because I told you in initial phase that peptide or protein are used for the multiple purpose. Okay, And so far, I have taken only one part, that is called vaccine actually. Right. Now, I am going to talk about you the another angle actually. That angle is regarding the delivery of the drugs. I told you that you can use the protein or peptide as a drug delivery vehicle. You can use it drug delivery vehicle. So this is a database we create for drug delivery. These are basically cell-penetrating peptides. This is a updated version. First version is called CPP site only. This is CPP site 2.0. This is updated few years back, and it collect and compile all cell-penetrating peptide reported in the literature. Please remember, all these databases are primary databases. They are not derived from any other database. And we collected all this information from the literature. So in this one, we have the most of the peptide which have the L conformation. Only few have the D conformation, and some of them have the L and D conformations basically. Most of the peptides are linear peptides. There are only few examples of cyclic peptides. Structures have been annotated for each peptide, and here we collected the information from the PDB. If it is bigger, then we use the itager. If it is a small size, we use the pep structure mode, okay, to identify the structure. Then this software, cell PPD, help you to design the cell penetrating. Like you have the softwares for designing the small chemicals. Similarly, we have the softwares for designing cell-penetrating peptides. So what is the advantage? It helps you to generate the all possible analogs. Because how you design a chemical, drug chemical? Okay, that's simple. You take a chemical, generate the analogs by putting some small changes actually. Okay, and once you got the all analogs, then calculate the therapeutic property of each analog. Same way we did for the cell-penetrating peptide. We have one peptide. We want to design the better cell-penetrating peptide. What we'll do? We'll change each amino acid at a time for all 12 positions in the amino acids, and we get the large number of analogs. And for each analog, we will compute the cell-penetrating potential of each peptide. So this software will help you to design a cell-penetrating peptide or to identify a cell-penetrating peptide which have the highest potential to enter into the cell. Okay, so it will help you. Then there is another database that is called topical PDB actually. In topical PDB is basically drug delivery through the external routes, maybe through the nose or through the eyes or through the skin. Drug can be delivered actually. So we collected all the information from the literature, which is experimentally validated data. So in all databases, the information is experimentally validated by the experimental scientist. We just collect and compile information from literature actually. So it's look easy, but reading all the papers and compiling that information is a heavy job actually. I'm fortunate that we have the excellent students with us which do this job, and we are able to produce a wide range of databases. So these are the basically topical PDB can be used to design the drug delivery vehicles, as well as there is another type of peptides, very interesting peptide, is called tumor homing peptides. Means tumor homing peptides. The word itself is giving the hint that they have the property, they will specifically go to the tumor and will bind to the tumor. But they will not go into the body fluid, but it will not bind anywhere. It will specifically bind to tumor homing, means to the tumor actually. So if you attach a drug with this tumor homing peptide, they can deliver the drug on the tumor site actually. So these peptides can be used for delivering the drug at the right place actually. Right place here is tumor basically. So far we talk about the vaccine, drug delivery vehicles. Now I am talking about the inhibitors. Like in chemicals, what you are doing actually, you are mainly designing the inhibitors. What are the inhibitors? They simply bind to the protein or peptide and then they inhibit the activity. So these are called inhibitors. So, but I am talking about you about the peptide based inhibitors. So it's a different than the normal inhibitors. So I'm talking about peptide based inhibitors. And here we have taken the anti-cancer. So these peptides have the property that they can lyse the cancer cells actually. Specifically cancer cells. They will not lyse the normal human cells actually. We developed this software as well as database earlier, but this year again we developed the updated version because that's a complaint from the number of experimental researchers that most of the servers or most of the databases are not updated with the time. Okay, so our group is not only developing new tools, but we also try to maintain all these tools, right? So this is another type of server which predict or which help you to design anti-hypertensive peptides actually. So this is a basically anti-hypertensive peptides. Similarly, I'm not going into detail. Then we have the server for predicting the antifungal peptides actually. So these inhibitors or these peptides have the capability to kill only the fungus actually. So they are called antifungal peptides. Similarly, we have the antibacterial peptide. So for each type of organ, we have the different type of servers. This is specifically designed for the anti-tuberculosis actually. So though we have the antibacterial peptides, that can cover the this tuberculosis also, but tuberculosis sometimes organ is very difficult to kill. So we design specifically for anti-tuberculosis. Means these peptides will kill tuberculosis basically. Now, so far I talked to you about the therapeutic potentials of peptides. But it's not easy. Why it's not easy? The peptides are facing large number of challenges. One of the problem we face when we design the peptide or protein based drug is their stability. Stability means their half-life. If half-life of a drug molecule is less actually, then it will not be delivered on the right place actually. Because you need the time, you need it to be in the body, so it reach to the right target actually. So half-life is one of the major challenge. So what we did here, we create a database of all the peptides half-life in body fluid actually. In half-life in the body fluid actually. Mainly in the blood basically. And we also have the server, I have not mentioned here, we also have a server which help you to design the peptides by making the small change, analogs by making a small change, you can increase the half-life of peptides. There is another challenge. That challenge is not only for the peptides, but it is also true for the chemicals actually. That is called toxicity. Because all peptides are not non-toxic. There are the peptides which have the heavy toxicity actually. So how to predict toxicity of a peptide or protein? That's a challenge actually. Because number of drug molecules or even protein molecules, peptide molecules, they rejected or they have not cleared the clinical trials because they have the toxicity. So how to predict toxicity is a challenge. So we develop a method called toxin predict actually, which help you to predict the toxicity of a peptide as well as to design the non-toxic peptides actually. Means it will generate the analog and it will tell which analog is the least toxic actually. So that will help you to design the toxic peptide. And I will tell you the good part of it that this year this toxin pred got more than 170 citations in year 2000 itself actually. That shows that last year it was heavily used by the community because a lot of people are working on designing the peptide based therapeutics basically. So this toxin pred is heavily used by the scientific community. There is another type of toxicity. It is you have to remember it. That is called this RBC toxicity or hemolytic potency. Means they specifically lyse. They specifically lyse the RBC actually. Okay. So how you will predict a peptide whether it will be this hemolytic or not hemolytic? Means it will lyse the RBC or not RBC. That's the important part. So we have the database as well as prediction server, which help the users to identify whether a peptide is toxic, hemotoxic or not actually. So if it is there, then you can remove the toxicity by just mutating a few residues actually. So that so far we are talking about the different type of properties. Now I'm coming in the next step because any protein or peptides function can be easily understand if you know the structure of this molecule. Okay. Though there are a number of methods have been developed over the years for predicting the structure of a protein. We have the competition called CASP, and every year, after every 2 years, they evaluate the performance of the protein structure prediction method tools actually. Okay, but question is, can we predict the structure of peptide by using the software which is developed for predicting the structure of proteins basically? That's a challenge. So here we propose that we should have the separate method for predicting the peptide structure, not to use the protein structure methods to predict the structure of peptides. Initially, it was a lot of objection from the reviewers. It was not published. Fortunately, somebody else shows that our method performed better than any protein structure prediction method on the peptide structures, on peptides. So we got the hint, we got the basically validation from some other groups who publish a paper, and based on that we publish a paper. One good thing we had actually, sometime it pay back to us from the long time actually. Once we work on any problem and we develop the server for the solving the problem, then we never retract the server from the website whether the paper is published or not published, because we believe our server is providing service to the community. Whether we are able to publish or not publish, that's not a challenge. Main question is whether we are able to provide the service to the community or not. So for us, providing the service, free service to the community is a major challenge. lot of people put me the question, why you are not charging the money from the customer actually. I told, look, most of the people in third world countries or maybe developing countries, they have limited resources. They cannot afford the commercial softwares. So what they will do, if they cannot afford the commercial software, then they will not use them. So they will be in the always in disadvantage actually. In order to make them at par with the other scientists, we are providing free service, and these softwares are as good as any commercial softwares because they are published in the reputed international journals actually. So which has been already reviewed actually. Then next part is, so far I talk about peptide structures actually and other parts. Now we have started new thing actually because we observed that in FDA approved drug, most of the drugs, most of the peptide or protein based drugs, they are chemically modified. Because why a peptide is unstable or it degraded very fast? Because there is a different type of enzymes. If you remove the enzyme cutting side, then it will block. Let's say, for example, most of the peptide or protein, they are basically chopped from the internal. There is a aminoprotease actually, which will chop it from the internal side. If you put some modification on internal modification, some chemical, you will moiety on the internal, then aminoprotease will not be able to recognize it. So it will not cut. Peptide will increase. Same way, in some cases, we can put some moiety at the C terminal, or maybe in the we can replace L amino acid with the D amino acid, or maybe we can glycosylate some of the residues in a peptide or protein so that our immune system will not recognize it. Right? So chemical modification of protein or peptide is important to make it suitable for vaccine or drug. Pure natural natural protein, it's very difficult. But the question is basically, can you predict function of a chemically modified peptide? That's a challenge. Because most of the methods developed so far in the literature, they are basically for natural amino acids actually. So we take the initiative to start to work on the chemically modified peptides actually. So this is the first server where we develop the method for predicting the self-penetrating potential of chemically modified peptides actually. If you, let's say, make modification, then we can tell that whether self-penetrating potential will increase or decrease. That will help you that you are making the modification in order to increase the half-life, but same time you are not compromising with the self-penetrating potential of the peptide. Okay, so that's the advantage the server has actually. You can put the sequence and it will predict whether it will self-penetrating or not, even if you can put the chemically modified actually. So that's the advantage. So same line we have developed for antimicrobial peptides, which are chemically modified actually. Whether a given peptide will be antimicrobial or not, for chemically modified peptide, it can predict. Now there is another question. Most of the methods, even for protein structure prediction or peptide structure prediction in the market, they only predict structure of the peptide which have the natural amino acids only. There is, best of our knowledge, there is no method for predicting the protein structures which have the chemical modifications like glycosylations, all the post-translation modifications. A wide range of modifications are already proposed in the literature and they are playing the excellent role, but there is no method which can help you to predict the structure of chemically modified peptides. In order to overcome this problem, we developed a server called Pep Structure Model. And this server helps you to predict chemically modified peptide structure actually. That's why we are there from last number of years. Let's say I started my group in 2000, actually 20 years our group is working and we have large number of softwares, still we feel we have not done anything actually. There are a number of problems which is not solved actually. Because sometimes most of the researchers have the problem that what to work actually. Their whole life, they take a small problem and they spend the whole life in that problem. That is not good sometimes actually. Because then it limit you to limitation of that particular field actually. So sometimes you have to explore the opportunities beyond the small area of research actually. So anyway, if you are interested, you can use this server. All servers are freely available to you. There is another thing is peptide diagnostics. That's another area. So what we did, we collect all the cancer patient, their body fluids, and what type of peptides have been released in their body fluid basically. So that will help you to design a biomarker to diagnose the particular type of cancer. So this database is available. So far, we are not able to develop the prediction method because I don't have any PhD student to work on this problem. But maybe we'll get the student and we'll work on it. Okay, that's another example how to design the vaccine. So here is the example based on the genomics, because in today's time, sequencing whole genome is not a problem. Even you can sequence the cancer cells separately. But how to find out the regions or mutations in a cancer cell, which is not available in healthy cells actually. Once you recognize it, then you have to check it. That same region can be used to induce the memory B cells or T cells, so that you can use it as a immunotherapy or as a vaccine candidate actually. So we proposed the fully personalized cancer vaccine, partially personalized vaccine candidates. So we have given the concept. So I'm not talking about the detail of the cancer vaccines here. This is the example we have created for the myeltuberculosis. I will tell you the problem. And you will ask the question, you have designed a lot of servers. Why you are not design any method for in real life actually? Whether you have any vaccine in the market? No, that is not practically possible because we don't have the experimental lab. And same time, we don't want to die with the knowledge. That's another problem. So what we are doing, according to theoretical predictions, whatever the best possible vaccine candidates are there, we predict it and we put on our website. So any experimental biologist who's interested in designing vaccines, rather than going here there to search the vaccine candidate, they will get the vaccine candidate, potential vaccine candidate here, and then they can use it. There is no need to give any credit to us actually. Okay. So that's our philosophy actually. Either we can do it, that's great. If we cannot do it, share the knowledge. Maybe somebody, other person will benefit it actually. Whether they give the credit to us or not doesn't matter actually. Okay. So I will tell you a very good example, because sometimes what I observed actually, there is a communication gap or there is a difference between the experimental scientists and theoreticians actually. And particularly this is true for the experimental, the Indian community. They don't trust on each other actually. If you talk to any experimental biologist, they will say theoreticians are nothing, it's just there doing something actually. Doesn't mean anything actually. It doesn't work in the real life. Same time, if you talk to bioinformaticians, they will say experimental scientists in India is not doing that, doing whatever has been done in the US and Europe actually. That's a common statement you got. So I am working I work in Chandigarh. That's a Institute of Microbiology. That's basically experimental biology lab actually. I was the odd man out who are doing bioethics actually. So they put me the challenge that you claim that your server or softwares are heavily used by the community in the world actually. And that is clear from the citations actually. Can you discover a single molecule which can be used in the real market? I take it as a challenge actually. I hired some PhD students who are biologists by background, and then work on this problem. So I decide I will design a drug delivery vehicle based on the self-penetrating peptide. So what we did in first step, we compile all the information related to self-penetrating peptide, and we created the two databases, CPP Site 1 and CPP Site 2, and both are published in database journal, one in database journal and another in NAR actually. Okay. After publishing these papers, the next target was to design a prediction method. So we develop a method, because how the prediction methods are designed, because we have the information here. We know this is the sequence and this is self-penetrating. This is sequence, this is non-self-penetrating. So how to drive the rules, how to develop the models actually. So for new sequence, they can predict whether it's a self-penetrating or not self-penetrating. That's a philosophy actually. And mainly we use the machine learning techniques or AI techniques, artificial intelligence techniques, to develop a prediction model. So we developed the prediction model, self-PPD, for predicting the self-penetrating peptide. So all three papers, they are all theoretical papers or maybe all web servers. So they are heavily used by public. So next step, what we did extra actually, we do the virtual scanning of all proteins in CISPROT. You know the CISPROT is a database of proteins which is well annotated database of the proteins. Okay, there we search which regions of the proteins can be natural self-penetrating peptides. Out of them, we only take the best 10 actually. We send it to for the synthesis of these 10 peptides and check on the different type of you call cells actually. And then we demonstrate that this one of the peptide IMTP8 have the better self-penetrating potential than any molecule so far reported in the literature. That's a big achievement to us, that experimentally we discover a molecule within 3 or 4 years, which is best in the world in the term of the self-penetrating peptide. We have a large number of peptide already, like TAT or pentene or that number of things. We patent it, we publish it. Then next we did the number of experiment actually. So delivery of cancer cell, we publish the paper in European Journal of Pulmonology. In all papers, they are all experimental papers actually. Here we demonstrate our peptide can deliver the drug inside the mouse skin actually. That's another paper we published. Then we also demonstrate if you take the chemical, means some MRSA, drug resistant bacteria, okay, which cannot be treated by this drug. In number of cases, if it is due to the membrane problem, then deliver it with our peptide and it can easily kill them, because these peptide make the membrane of the bacteria fragile actually, and then chemical can enter it, because sometimes resistance is due to that permeability change of the membrane of the bacteria actually. Then it will enter and then it will kill the, and then we demonstrate it and it is published. So now you can understand, within a span of 4 or 5 years, we publish many publications, experimental papers, as well as 3 or 4 patents. Idea here is, message is here, if experimental scientists and theoreticians or bioinformaticians, if they work together, they can discover wonder actually. The problem here actually, both community doesn't want to talk to each other actually. They don't have the trust or respect for each other actually. But I will tell you, in today's time, if experimental biologist thought that he or she can survive without using the bioinformatics softwares or the computation tools, then he's in some illusion actually. He doesn't understand the importance of the software. And same is true for the computer scientist. If they want to discover something in the real life, they have to work with the experimental biologist. That's a message actually. So this work demonstrates that in 4 or 5 years, we discovered the molecule which has not been discovered by MTech in last number of years actually. Okay. Anyway, so these are just example. one work we do normally that is called public service. Let's say Ebola come few years back, and if any biologist will work, he or she take a long time to find out the possible vaccine candidate, possible drug candidate, diagnostic primers, blah blah blah. So we work on that, because we are bioinformaticians, we use the different softwares and we predict the vaccine candidate, drug candidate, and potential primers for designing the prognostic, designing the PCR-based diagnostics actually. So anybody can use from this site. So this portal is freely available, so anybody is interested, they can use from there actually. So the idea here is how to help the community when they are fighting against some particular virus actually. This is an example of coronavirus. Different type of vaccines are there, and we propose certain modifications in these vaccines so that toxicity, toxic region, allergy can be removed actually. So we publish a paper, and that's called review, where we describe the importance of removing the toxicity from these vaccine candidates. This is the website where all the related information to the coronavirus is available, starting from drug candidate, vaccine candidate, genomics, proteomics, whatever the possible we can think, we have put it here actually, including the simple links, where is WHO link, where is the Indian sites, where is the Ministry of Health, so that person can contact to the right person actually. So rather than going here, there, you can get the information from a single source. That's the whole philosophy. And this is the information in this coronavirus portal is there. That's basically, it provides the information related to drugs, what are the potential drugs, okay, target structures, general information, diagnostics, PCR diagnostics, more primers actually, maybe some modification happen, then we should have the more primers actually, which can detect even the new strains of the virus basically. Immunotherapeutics, what possible B-cell epitope, T-cell epitope, what is the possible vaccine candidates. So we provide the complete comprehensive information here. As well as we develop some new tools, because in, let's say, interferon gamma, we already developed earlier tool for predicting the interferon gamma, because these are cytokines which are basically overproduced in case of the coronavirus, and which damage the different tissues or responsible for fever and other things actually. Okay, so we developed this method for predicting interferon gamma, but now we have developed a new method called IL-6 actually, because IL-6 is responsible for number of tissue injuries actually. Okay, so we developed a new method for predicting the IL-6 because there are no other method actually. So this is a method. If you are interested, just developed few months back, you can use them. Okay, thank you very much. Now floor is open for the question answers.
So this.
Okay, Hemant. Hello. Can you hear me?
Yes, sir. We can hear you, sir.
Yeah. If there is any question, please ask.
Yes. Tanbir Ahmed. He is asking, sir, what factors should be considered during developing a peptide in drug design?
Means question is basically means what factor? That's a too wide question. Let's say you want to first see the efficacy, then you have to, let's say you want to design the vaccine candidate. So it should be able to activate all arm of immune system, B cell, T cell, as well as innate immune system. That's the first thing. You should also check the toxicity, immunotoxicity, and other parts so that it is not harmful actually. It should be stable so that half-life is there, otherwise it will not survive. These are some factors.
So if anyone have any other question? So, sir's email information is available, and
Okay.
this talk is available on YouTube. And whoever want to ask anything, they can write it to sir. Somebody is asking that all these, but this is already explained. He's asking whether these software are freely available or it's commercial. So it
They
is
are
already
all
explained.
freely available.
Yes. Okay, sir. Thank you so much.
Okay, thank you. Thank you.
यूट्यूब में बहुत सारे क्वेश्चन थे ना, ये एमिनो एसिड्स और नॉर्मल यूट्यूब में।
Blah blah blah. So we work on that genome because we are bioinformaticians. We use the different softwares and we predict the vaccine candidate, drug candidate, and potential primers for designing the prognostic means designing the PCR-based diagnostics actually. So anybody can use from this site. So this portal is
Good morning, everyone. Welcome to the second day of MMTT 2021 International Workshop. Today, the first talk will be given by Professor GPS Raghava, who is a world-renowned.
should consider it as a vaccines design DNA or maybe carbohydrates or lipids. And
he has been listed in the world's most influential
Sir, good morning. You're muted, sir.
Yeah, okay. Can you hear me? Hello?
Yes, yes, I can hear you, sir.
Okay, so did I join the right link?
Yes.
Okay, so
Join the right.
let me just quickly get my Let's check. Anyway, I think we still have 10 more minutes, right?
Yes, we have, sir.
Okay. I was in some other call. I
Hi,
had to. Hi, Hemant. How are you?
fine, fine.
Sorry,
So
Hemant.
good.
I
So
mean,
there is no technical issue here. I think very
yeah,
good.
yeah, it's good to check.
Well organized office. Very nice.
Oh, my organized office. No.
Looks good actually.
Just busy.
So you got CRSI 2021. Congratulations. I have just
Thank you.
read your profile.
Yeah, thank you.
Very nice. Congratulations.
Thank you.
So
very much.
everything is coming day by day.
You know.
Yeah.
Anyway,
And you well deserve. No, you are working really hard, doing a lot of tough stuff, and then it is really nice to see all those things.
Thank you.
Good. So we have around how many minutes? 9 minutes now.
Yeah, I'm just trying to open the correct present. I worked on the presentation. Yeah, I have it here. See, these days it's becoming way too much. I'm like struggling to, manage my time and everything.
Yeah. So much of request for online talks now, no, these
No,
days.
not just that. It's just that, we have something called I mean, I see so many people on the call. Are they all hearing us?
No.
Okay.
They are not hearing.
Okay. Yes, I just saw that there are many.
So whoever is attendee, they will not be hearing you. But whoever is in the presenter, they will be hearing.
Okay, I see my student also there.
Yeah, because he is selected for hands-on.
Okay.
So hands-on selected people, they can interact with the speaker.
Oh, okay. Nice. Good idea. So how is Dr. Shastri? Are you in touch?
Yesterday he has given inaugural talk. It was really nice.
Yeah, I saw the YouTube link. I mean, I did not attend the whole thing or anything, but I still saw that.
Yeah, it was good actually.
Nice.
So we had inaugural session and then 1 hour break.
And
then we had his first talk. And today in the morning, GPSR has given this talk.
Oh, nice. He is a very good talk speaker.
Yeah. He has done so much, no?
Yeah. Not just that, the way he connects with the audience, no, that also is nice.
Yes.
Like, I don't think I will ever learn all that. So how is Professor USN Murthy? Please say hello to him.
He is not in the station.
I know, but just when he's
I will tell him.
Please say hello. Yes. Okay, now let's try. I have to go for the bathroom and come also. From morning 9 o'clock, I've been in meetings. That's what I told. No, these days it's becoming too much. Yeah, I don't know why.
Because 4 minutes are there. So you can turn off this video and audio, and in 6 minutes you can join.
Yes. Let's check this once, the thing that it's working, and then I'll share the screen.
Yes.
Okay. Are you sure you want to share? Okay, share.
Are you sure?
No, it's asked if I'm sure.
I think it is working. Yes, I can see your screen.
This is fine, right?
Yeah.
No problem.
Yeah.
This is fine, right? No problem. Yes. Hello, can you hear me? Hello.
Yeah. This is absolutely fine.
Oh, okay, good, excellent. So, okay, I will quickly go to the.
We'll turn off this and then.
Yeah, I'll turn off my video and then I'll be back in 2 minutes. That's enough. So everything else is fine, Hemant, with family and everything going good?
Yes.
Nice.
Everything is going on.
Say hello to your wife also.
Yes.
Oh, we should meet sometime. This year is so hard. I mean, last year.
This
Or I don't want to say this year.
Sure. Last year. Anyways.
But I don't see.
You are muted now.
I think someone muted me. But anyway, I'll mute and then I'll come.
Okay, fine.
Hello? Yeah, Hemant, I'm here.
So, good morning, everyone. Welcome you all after this first break. Now, this talk will be given by Dr. Deva Priya Kumar, who is an associate professor in IIIT Hyderabad. He was a visiting researcher in University of Strathclyde, Glasgow, Scotland from July 2018 till date. He is JSPS Invitation Fellow. He is also invited associate professor at Osaka, Japan. He worked as assistant professor from 2008 to 12 in the same institute, IIIT Hyderabad. He did his postdoc from University of Maryland with Professor Alex Mackerel. He did his PhD from Pondicherry University and IICT under the supervision of Dr. G. Narhari Sastry. And he got several awards in his academic career. That includes CRSI Bronze Medal, that is the most recent one, visiting fellow from Royal Society of Edinburgh, Distinguished Lecture Award from the Chemical Society of Japan, JSPS Invitation Fellow. He got INSA Medal for Young Scientist, that is very prestigious. Also got Indian Academy of Sciences Young Associate, and several other research awards and several other fellowships and so much of funding and all. So he is very productive, and let's welcome him for the talk. Now over to you, Dr. Deva.
Yeah, thank you, Dr. Hemant Srivastava. Thank you very much for the kind invitation and for having me here. So it's a great pleasure to be part of this workshop and be able to talk a little bit of how machine learning can be applied in healthcare. Okay, so today what I will do for the next 1 hour or so is to talk we'll take healthcare in general, and then we will also choose drug design as a specific sub area, and then we talk about how machine learning can be applied to these aspects. Okay, so now so as Hemant said, I am from IIIT Hyderabad. Okay, so in the last few years, right, especially recently, people have been talking about deep learning and machine learning and artificial intelligence in a multitude of contexts. If you take even the newspapers, or if you take read, if you listen to news, or if you talk to or look at conferences, for example, right, and then journals, papers, right. So you see that there is a large amount of or there is a sudden increase in the number of instances where one of these terms is actually used in several contexts, right? Sometimes people use deep learning, machine learning interchangeably, and so on, right? So, machine learning or deep learning is a part of this machine learning, and all these are artificial intelligence methods, right? There are other methods also come under this, but in science, for example, recently we have been looking at large of these examples, especially in deep learning and in the larger machine learning umbrella. Okay, but let's quickly look at in what context do we hear machine learning and deep learning and AI these days, right? Or where do they use now? Okay, so in science or in drug discovery that you guys are discussing, right? So we are talking about how this can be used now. Okay, so but then there are some areas where people have been using it for a reasonably long time, and of course they are also the efficiency of these methods are also evolving, right? So these are some of the areas. Okay, so for example, AI in travel, when you want to, for example, if someone books a flight ticket, let's say, let's say I buy a flight ticket. Okay, so if I look at the cost, let's say 2 days later or 1 day later, right, it's not the same a lot of times, right? Sometimes it's actually, most of the times it increases, but sometimes it also can decrease, right? That's like what is called dynamic pricing. Okay, and for example, in online e-commerce websites, right? So if I look at a particular product, if I explore a little bit, and then the website starts suggesting products that are similar to what we have been looking at, or it looks at our history and then starts suggesting new things for us to buy, right? And then social media, like social media sites, suggest friends or suggest videos for you to watch, or suggest news articles for you to read and things like that, right? So similarly, and of course, we also learn a lot about autonomous driving. Vehicles can drive themselves and so on. So all these things, right? All these applications, or the underlying technology is data-driven methods, or the AI ML based methods. Okay, so today we will talk a little bit about healthcare, right? So how ML, machine learning, can be used in healthcare applications. Okay, all right. So when we say this is just a schematic that I just took from this nice review of what are the different areas of healthcare that we can where areas of healthcare where AI ML has been used so far, right? Of course, in terms of the data, okay, so you use a multitude of datas, okay, and then on the other side, you have the task, like for example, disease stratification. I will give this as one of the examples, and then we also will move to drug design as a second example. Okay, of course, this is not just that. You can do image diagnostics, learn more about your genome. And for example, getting protein structures. Recently, some of you may have read about how deep learning methods have improved our capability of predicting or coming up with protein or modeling protein structures from sequence. Okay, and so on, right? So these are some of the examples. So when it comes to healthcare, right? So the idea is that if can we use a multitude of different kinds of data, multimodal data, and then use that to provide better healthcare, right? It could be in terms of preventive healthcare, or it could be diagnosing a disease, or finding new drugs, or finding new protocols for treatment, right? So it could be any of that. Okay, right. So for example, when we like there are a lot of image-based diagnostics, like for example the X-ray images, or CT scans, or fMRI scans, or histopathology images, and things like that. Okay, and then you also have temporal inputs, where a simple example could be like ECG, of course. But also, like we also nowadays wearables have become very, very popular, like Fitbit, or Apple Watch, or these kind of wearables, which constantly measure certain biomarkers. For example, it continuously measures heartbeat, right? Some of them actually can do ECG, right? Okay, and so on, right? There are a lot of sensors that people are have developed, and then there's a lot of research going on in terms of development of new sensors that can monitor our health, right? So basically the data from data such as that, and then you can also have genomic kind of information, right? You can go get the genome sequencing done much easier than even 5 years ago today, right? So so is it possible to combine these? In some sense, these are very, very different kinds of inputs. One is like an image, second is like a temporal data, right? Time as a property, a heartbeat as a function of time, and things like that. And then the other one is like the genome, right? Is it possible to combine all this in some way and then provide a better healthcare, right? So that's where the artificial intelligence comes into picture, where people say that, okay, it is, people have started to look at how this can be possible, right? So this is just a quick example of how the artificial intelligence based methods are actually making a big entry, or making a big difference in the general area of drug discovery, right? So for example, the number of startups at this time where they used AI for drug discovery is about 37 globally, right? And then in April, 2.5 years later, this number grew to about 230, right? A large increase given that it's only about 2.5 years, right? So, this is just another testament to how artificial AI or the machine learning methods have started to contribute to this particular area significantly. Okay. All right. So now first what we will do is try and understand what is the difference between a regular, like a typical computation that we do in a lab versus machine learning. Okay, so let's just try to, let's spend about 10 minutes or so in terms of just making some simple arguments to understand what machine learning is. Okay, of course we are not going to go into the math background, or we are not even going to discussions at a much deeper level, right? At one level of understanding of what is the difference between what a traditional computation is and then machine learning. Okay, so what is traditional computation? So basically what we do is we start with a computer program, right? So I assume that a lot of you would be doing hands-on exercises. For example, Gaussian, right? Gaussian 16, for example, is a program, computer program that can be installed on a computer. So what do we do with that? For a given input, right? Let's say for a given structure, I want to know, let's assume the properties of, properties of a given molecule, let's say. So what I would do, right, so okay, let's assume Autodoc is a program, right? What I want are the binding affinity of a given protein ligand structure. Okay, so I take the program and then use a computer, and then for a given protein ligand structure, I can calculate the binding affinity, right? That's what we do. If I take Gaussian 16, I take a Gaussian program, and then I take the Cartesian coordinates or the internal coordinates of a given molecule, and then I use the computer to get the output. Let's say energy, or or polarizability, or dipole moment, right? Or the orbitals, or ionization energy, whatever it is, right? So I get some output by using a computer program based on a given input, and I use computer for that. That's basically a traditional computing. So what is the difference between this and this then? The difference basically is that we have swapped these two, right? So I have taken the program and I put it on the other side. I've taken the output and put it on this side, right? So I have the output here, program here. Everything else is exactly the same as before. Okay, so now what we are trying to do is I'm giving the computer that, okay, for a given this particular input, this is my output, right? So like that, I give a large number of examples. Okay, we will discuss a little bit more on what that means. Okay, so I give large number of examples of input output pairs, and then I tell the computer, you figure out, or you write a computer program for me, right? So I ask the computer to give me a program which, taking a given input, produces that particular output. Okay, so that's the difference between the computation, or the traditional computation, and the machine learning. So now what do I do with this? So let us assume that the computer has actually figured out a program to do that. So now what I can do, I can take that and plug it in here, and then I can get the output for the future examples for which I do not have the output. Okay, so in other words, what we are saying, instead of having a program, I tell the computer examples of protein ligand structures and the binding affinity, and then the computer tells me, okay, use this particular program so that for a given set of protein ligand structure, you will get a given binding affinity. Okay, so that we don't call a program, we call that a machine learning model. Okay. Right, so let's just take a, as I said, we will, take some simple example and then try to understand what this means. Okay, so this machine learning framework, right? It involves two different aspects. One is what is called a training of a model, and then the testing of the model. Okay, So let's assume, let's understand what the rest of the things that are given here. So, we'll come back to this for a second and then do a, exercise. Okay. So let us assume that, let's assume that I go to a, let's say I go to a shop, right? And then, I don't ask anything. Okay. Let's assume that that particular shop's sells one item. Okay, so let's say like one, some sweet or whatever it is. Let's assume that's a small shop, and then they just sell one thing, like one kind of fruit or whatever. Okay, so I just sit and watch what is happening there. Okay, so let's say a person comes in. Let's say the shopkeeper is there. A person comes in, and then he gives 5 rupees and then gets one item. So let's say the second person comes in and gives 10 rupees and gets the second item. And then the third person comes and he gives, let's say 100 rupees and he gets, 20 items, right? And then the, fourth person comes and then gives 50 rupees and then gets 10 items. Okay, so now I ask you a question saying, okay, there is a fifth person coming in and that person is actually giving 200 rupees. So now I have to figure out, how many items this person is going to get, right? So this is a very easy problem. Just by looking at this, if this is, if these are the examples, based on that, I, tell that my answer is, let's say, 40. Right? No? Okay. So that's basically what I figure out. Right? So, but now, nobody told me that each item costed 5 rupees, right? So what all someone told was they gave this particular data and then somebody asked you to figure out what, how much will you get for 200 rupees. Okay, so on the other hand, so you figured out that you have to divide this number. Let's assume this is X and this is Y. So you have to take the number X and divide it by 5, and that is your Y. So this is what we figured out. Okay, so we, based on these examples that are given, right, I figured out that this is the relationship between the input and the output. Okay, so if someone gives you and then asks for a given, or if someone gives this example and then he gives this equation and says X equals 50, Y is what, right? So that is an example of a traditional computing. Okay, so instead, if someone gives, let's say, this. Okay, so let's say someone gives you this, and then you figure out what this is. Okay, that is machine learning. That's the only difference, right? So let's go back to this slide now and then try to understand what that is. So what we told in the next slide is that someone has given you a set of values of X and the corresponding values of Y. Okay, so now what is it we are trying to figure out? We are trying to figure out what this F is. We are trying to figure out a function which relates X and Y. That's basically what we are trying to do here. Okay, so that's what, as you can read here, given a large number of XY pairs, given a large number of these XY pairs, estimate F. We are trying to figure out this. How do I do it? By just minimizing the difference between the actual value of Y and then the predicted value of Y. Okay, so based on this particular F. So I minimize this particular value, somehow it could be a mean absolute error or the root mean square error, or there are several loss functions that we normally use. Okay, so let's assume that we have we take these two numbers and then minimize the difference. Right? If the difference is 0, that means your function is perfect, right? So if your difference is high, then the function is not good. It cannot explain this dependency, right? So that's basically the training, that you take a certain number of examples and then try to identify a prediction function that will take a value of that. It will take a value of X and gives you the value of Y, right? Because I know these X and Y pairs, I have the Y predicted using F, and then I have the original Y. I take the difference, and if it is close to 0, that means F is really good. If it is not close to 0, then I have to improve, or I have to change this function somehow, right? So that details we are not going into go into details of how that process is done. What is testing? So basically what you do is you take new values of X for which Y is known but was not used in this particular step, and then try to predict what is the value of Y given a value of X using the function that you came up at this particular step. Okay, so once that is done, so we can that's basically the testing, and if it works on the testing pairs as well, then that is what is your machine learning model that can be used on further examples. Right. Okay, so now what we are going to do, we are going to talk about, as I said, okay, so far let's just do a quick recap. So what we did was that we made some very simple arguments, right? And then we took very simple examples and then try to understand the differences between the traditional computations that we normally do versus the machine learning methods. Okay, so we'll go back here for a second. So this one, right? So the function, the prediction function, usually is a artificial neural network. That is like a universal approximation to the actual function that may have been there. Okay, so that's basically what we use. So now what we are going to do in the next 40 minutes or so is to take two sub areas within healthcare. Okay, so one is more like how to stratify. Let's call it risk stratification is one sub area, and then the second sub area that we will take is applications in drug design. Okay, so that's basically what we will do in the next 40 minutes or so. So let's understand what is risk stratification. So let us take the example of the current COVID-19 pandemic. Okay, so what we know from the data so far that we have collected in the last 1 year is that a lot of people get infected. Okay, but then only a very small percentage of these people who get infected are under risk of where the condition can become very, very severe, and some of them are a very small percentage of the total population does not survive, right? So that way, if we are able to stratify the risk, saying okay, I have 100 patients who are infected. Okay, out of these 100 patients, these 90 patients are of very, very low risk, right? They got infected, but then in 10 days, 2 weeks from now, they will be fine. And remaining of these 10 patients, they are likely to be high risk patients where the situation can become worse, and the situation can become worse such that some of them can actually pass away, or they do not survive. Okay, if we are able to do this, right? If I take the whole population and then classify them as, let's say, low risk and then high risk patients. Okay, if I'm able to do that, then I can direct, I can use all the medical resources, okay, to the people of high risk, and then hence minimize the human loss, right? So that's that's one way to go, but that's basically what is risk stratification. Okay, so here, I'm going to take two examples. Okay, both are very, very similar. Okay, the first one is using machine learning. Okay, so we have developed what is called a clinical decision support system for predicting COVID-19 mortality. So basically, you take simple parameters and then try to predict if that particular patient is in danger of not surviving or not. Okay, so that's the first one. All right, the second one is this is based on the population in from China, and then this is the second one which I talk about, which is specific to the Indian cohort, right? So basically, we develop risk stratification and mortality prediction models, to predict whether someone is at high risk or low risk, and then to predict someone whether this person would survive or not. Okay. All right. So basically, the first, as I said before, right? So let's quickly go to that slide once. Okay. So as we said here, right? So the ability or the chances of getting a better prediction function is heavily depends on, first, of course, the complexity of the problem itself. Second is the number of pairs examples that you have. What does it mean? You have to have a large amount of data to get, to do that. Okay, so the first problem, what is the first problem? That we are coming up with a machine learning model or machine learning method for mortality prediction. We need some data. Okay, so basically this is the data from China, right? So this is from the early infection rate, that data from China. So basically what we have is we have the hematological parameters. What do you mean by the blood parameters, like the neutrophil count, and then D-dimer, and then C-reactive protein, and all kinds of data during the time when they were hospitalized. Okay, so there are about 375 patients who are in this part of this data, and then out of which 201 actually survived after the COVID-19 infection, and 174 of them actually did not survive after the infection. Okay, so what do we do now? So basically, as I said, I just told before that once you have a data set, what we want to do is split them so that I can use some of them for training and some of them for testing, right? The data, especially such as the electronic health records, like the clinical data, things are not going to be very, very clean, right? For example, if I'm looking for 100 different parameters for every patient out of like 1,000 patients, let's say, right? There will be parameters missing. There will be parameters, with the quality is not so good, but then the parameters come from multiple, different days and things like that, right? So, you have to, there is a, lot of procedures by which you can, you have to do a, pre-processing, right? If there are some missing values, then you have to make a decision in terms of, completely ignoring the data. That's not going to be good, because we already have only for about 375 patients. If I'm going to delete data because some values are missing, I may not have much data to work with, right? So you can, for example, impute missing data, and there is a lot of pre-processing one should do before training a model, right? And then of course, you cannot use a large number of features. Basically, features are basically your x in that particular equation that I showed. Okay, so that's your x. So you have to be able to select a certain certain features. Okay, a certain small number of features for prediction. Okay, so for that we use what is called the XGBoost algorithm to do feature selection. And based on that, we use 5 different parameters: age, neutrophils, lymphocytes, LDH, and then HCRP. Okay, so basically these are the parameters using which we made the machine learning models. Okay, so now you have the machine learning models. You take the test data and then use the similar kind of procedure to find the missing values and so on, and then test it on this data and then see how well we do. Okay. All right. So let's show some result, as you can see here, right? So what is it that we are trying to? Yeah. Hello.
Yeah. Hi, sir. It's Suyash. So I
Yeah, go
have
ahead.
one question. Can you go to your previous slide?
This one.
No, sir. That one, no, where you are showing that what are the features and yeah. In the training set processing, you are using the normalization using min-max scaler. Now, in one of the case, if we don't have like active and inactive, like here, you are means you are trying to identify means what is the dependent variable and what is the independent variable, means what we are going to identify from this model.
Okay. I don't think I understood the question. So min-max scalar is we use so that the magnitude, some values, for example, the these numbers, right? All of them are in a similar range, right? So it doesn't, some of them is not in the order of 10 power 4 and some of them in the order of 10, right? So that, all of them are given equal importance. We do that kind of scaling, right? So what is the second question that you asked?
No,
independent variable.
My question. Yeah, my question is what we are going to identify from this model after getting all this data: age, neutrophil, lymphocyte, and LDH, HCRF. These features actually. So,
okay.
This model, but it will differentiate. Means.
Basically, it's a classifier, right?
Classifier.
So basically, yeah. So I don't have the confusion matrix, but anyway, so the task, and that's what I was going to explain. Sorry. So basically, what is it we are trying to do? We are developing the machine learning models that will the same thing that we started from here, right? So I take all patients that are there, and then be able to classify them. This is not there, okay? So classify them or as whether low risk or high risk. In the first project that I'm showing, whether that particular patient will survive. Will likely to survive or likely not to survive. That's the classification you're doing. Is that okay?
Yeah. That's what actually I was asking. So my next question is, if we don't have a data set, means we have a data where high risk means survival rate is very less, means patient with high risk is very low, like we have only 10 patients with that, and with low risk we have around 100 or 200 patients. In that scenario, how we will balance our data set?
Okay, there are two issues here. Good question. So one is, so basically what the question is that we started with an argument saying like out of 100 patients, we know that only a very small percentage of them are at high risk, or only a small percent of them do not survive. Right? If I go just take data, collect data randomly, of course, I'm going to approximately preserve that particular ratio, right? In terms of high risk is to low risk, right? So one of the things that we do is, like maybe I'll show the next example. Second project where when you collect data, you make sure that you have serious a reasonable balance, right? For example, in this case we have two classes, and then both these classes are very well represented. Okay, so maybe you are in a situation where there's absolutely no way that you can achieve that kind of balance. Then there are ways to do it, right? There are ways to handle data imbalance, but they're always not so great. Okay,
yeah,
so that's a very good question.
In most of the cases, we found that this is the case where we are looking for the high risk and the low risk. But in case of like small molecules, most of the time we got a higher number of molecules which are not active and a low number of molecules are active. So how to balance the data set? Whether we should remove less active molecules from our data set and make it balanced, or is there any algorithm or what to use in that case?
Mm-hmm. So there are multiple ways to do it, right? So one, if the total data set size is very large, then then basically what you have is like when you have two classes, you have two different distributions. You have a class of molecules in one distribution, which is, let's say, active, and a second class of molecules which are not active, which is too big, right? It's very well highly represented, right? So if the total size is big, then it's okay to do this. As I said, there are other ways to handle, right? So maybe we can discuss later, right? So I don't want to do it now. Yeah. Okay, so now what is that saying? Yeah, so basically, as there's good questions. Thank you. So what we are trying to do is like we are trying to classify the group of patients into either survived versus did not survive, right? So that as you can see that in terms of the AUC score or the F1 score, we do reasonably well when we choose neural network as the neural network for the classification task. Okay, all right, so that's the first example, right? So and then the second question is that we do know that the way people different populations respond to different ways. Different populations respond to the COVID, SARS-CoV infections differently. Okay, for example, if you take the mortality rates in India, it's very, very low compared to, for example, in the US or Europe, for example, right? So what we are saying is that a model, for example, this one, right? That this one works reasonably well for the population in China. So now if we want to apply the same model to the population in India, that's not going to work because we respond differently to the infection. So we did this study on taking the Indian patients, which again this also is available on medRxiv, even the other one also. So basically can you take propose a new model that can be used for the Indian cohort or the Indian population? Okay, so now. So what do we need to do? Then you'll have a data set involving the Indian patients. Okay, so we collaborated with Max Hospitals, CSIR, IGAB, and Intel, and so on, and then we came up with we got data from about 544 patients, right? So with reasonable representation of both the survived and did not survive, and then high risk, and then low risk. Sorry, this is high risk and this is low risk, and so on. Okay, and then we did a similar exercise, and then of course this is the confusion matrix corresponding to using the Wuhan model and then predicting on the Indian data set and then see how well we're doing. Obviously, we are not doing so very well. That again shows why it's why you need specific models for different cohorts, right? So the approach is similar, where we start from a data set of 544 patients and then develop machine learning models for the risk stratification in this case. Okay, so we did both risk stratification, that is whether high versus low risk, and then also the mortality prediction, again patients who survived versus did not survive. Okay, so as you can see, we do reasonably well, not as well as in the other case, but then we do reasonably well. Here, for example, this is the F1 score corresponding to the test set, right? So the other thing that we want to do is like there is no point predicting that someone is not going to, or someone is high risk at a point where you have to give, put them on ventilator. So you want to be able to predict well in advance, right? Let's say someone is infected. And then if we can take the blood and then do some tests, okay, so let's get the complete blood picture and then the inflammation parameters and so on. And if you're able to predict if that particular patient is going to be at high risk or low risk, let's say 10 days later, right? That will be the most useful thing to do. So that's basically here. So that's the number of days to outcome, right? That's the number of days to outcome and how the models perform with respect to moving towards the day of the outcome, right? So this is the good news that actually the model performs better in the later days, right? again, why such behavior? Please do look at the reprint. Okay, we do understand why it drops down later. Okay. And not only that, we also kind of try to understand several things in terms of why the mortality rate is possibly low in India and things like that. Okay, so now let's move on to the next sub area. How much time do I have? 11:56. It's about 40 minutes or so that we started. No. Okay, the second topic that we are moving on to is, again, I'm just going to take a few examples again from our own lab in terms of how machine learning methods can be used in drug design. Okay. All right. Right. So, the use of drug design methods, right? It's not so very new. Sorry. The use of machine learning based methods. It's not very new in drug design, right? So, for example, this is a paper from 1973, right? So where what they did was they used the perceptron model, a 3-layer perceptron model, right? And then to classify a class of molecules called dioxanes based on their pharmacological activity, right? So basically they took with respect to different R groups in this particular molecule, right? So they use this network to classify whether it is pharmacologically active or not. This is about 50 years ago, right? So this is not so very new, especially in the area of drug design. If you consider, machine learning applied to sciences in general, maybe not so very common. But in drug discovery, drug design, of course, in the QSAR and things like that, like the SVM, random forest, and these are methods that are very, very commonly used in drug design. Okay, right. So, so this is a slide that you may have seen already in this workshop or otherwise as well. This kind of or a variant of this particular slide. So this basically gives a schematic representation of the drug discovery process. Like, for example, starting from a disease, right? How do you get to a drug that will be marketed? Okay, so that's the process of that's basically the process of what do you call discovering new drugs, right? In terms of identifying a target, validating a target, and then trying to identify hit molecules, and then improving, find lead molecules and propose some candidates, and then show that it works on realistic systems. It could be biochemical assays, cellular assays, or tests done on animals, for example, right? So once all of this is successful, you go to the different phases of clinical trials, and then you come up with a drug molecule, right? So basically, this is something that we understand well, right? So the same thing, slightly coarse-grained or with much less detail. So you have a disease. First is you do a target identification. Once you identify the target, you design a molecule that works, and then show that it also works in the experimental lab, and then do several phases of clinical trials, and then give this particular drug could be marketed depending on the depending on the data which the regulatory authorities go through and then decide whether to approve or not. Right. So now, as you can imagine, okay, as you know already, some of you or most of you, that this process involves a large number of experts or a large number of people from different areas of science and technology. Okay, so you take, for example, biologists, they spend a lot of time in terms of understanding the disease, in terms of the pathways, in terms of, proposing new targets, right? And then understanding the life cycle and things like that, right? So once that is done, they try to come up with a target, right? Again, this involves people from bioinformatics, proteomics, and so on. And then so if you and then go here and you involve people from computational chemistry, medicinal chemistry, organic chemistry, pharmacology, formulation, and all kinds of areas, and then and then it goes here, it becomes much more medical based, right? The idea is that there is in this whole process a large number of expertises are involved in terms of the area of science, right, on technology, and also in terms of the number of people who contribute in this particular process, right? So let us assume, or let's take some examples of some papers that have been published in the last 2 years, not even 3 years, right? Less than 3 years. Okay. So there was this paper on machine learning. You can see the title: Machine Learning Approach for Drug Target Identification. Okay. Again, I'm just selectively showing some papers, especially the high that were published in Nature, Science, and so on. Okay, so you can see improved protein structure prediction using potentials from deep learning. So what is it that they are saying here? Once you have some networks, or based on whatever data that we have and whatever data that we generate in experimental lab, use that data to identify the target automatically. And then once you identify a target, you take the sequence of that particular protein and then use this deep learning method to get the protein structure, right? So for example, here, once you have a protein structure from here, you can take it and then do a de novo drug design using deep, in this case, reinforcement learning. I'll also talk about this a bit. And then once you have the molecules proposed by this algorithm, maybe there is another algorithm, AI-based algorithm, which can help you plan the chemical synthesis. Okay, so once that is done, maybe you have a robotic platform that can take the input from here and then actually do a synthesis of the chemical compounds that were proposed here. Okay. So maybe once you have this, maybe there is some AI algorithm that can read the spectra that was generated, or like for example the NMR spectrum, and then do the compound characterization, and so on, right? So and then you can also have machine learning models that can help you predict the clinical outcomes. So basically what you are showing is that the AI or the ML based methods can contribute to every aspect of this particular process, whether in terms of analyzing the biological data here, all the way to your marketing or the market needs in terms of, let's forget about the marketing aspects, here in terms of predicting how, in terms of selecting cohorts for doing the trials, or predicting whether a given clinical trial will be successful or not, and so on, right? If the chances of success is high, then you invest in that particular direction. If the chances are very low, then you'll have to see how you have to go back, or you keep so the idea is simple, that in the whole process of drug discovery, that's the the idea of this workshop, right? So it is possible that the machine learning methods actually could contribute at every single step. That's what the last 2 to 3 years are kind of showing us, right? Okay, so what I'm going to do now, again, it will be nice if someone tells me how long I have. Based on that, I will plan the rest of the talk. Hemant or someone.
Sir?
Yeah, go ahead, please.
You have more 40 minutes, sir.
40 more minutes, huh? Okay.
Yes,
All right. Maybe I will plan for another 25 minutes so that there is some time for discussion. Is that okay?
Okay, sir.
I mean, I can continue speaking, but that's not nice. Yeah. Okay. All right. So, right. So let's. Okay. So let's start from this one, right? Okay, so one of the fundamental exercises in molecular modeling is to be able to predict properties. Okay, so don't worry about what is there in the slide now. Let's discuss for 30 seconds and then go to this slide, right? So if you take molecular modeling for like some for any kind of applications, including drug discovery, right? So one of the major tasks in molecular modeling is to calculate the energy of a given molecule, right? The energy, the potential energy function, and then the energy has a lot of implications in terms of calculating the other properties. Okay, so for example, energy is the guiding principle for getting to the stable structures also, right? Starting from getting a proper structure of a given molecule, right? We need to worry about the potential energy of a molecule. Okay, so what I'm saying is energy is an important property which we need to calculate as accurately as possible, right? Practical, as whatever practically allows, such that that is extremely useful. Okay, so now going back to this, how do we do that? So one straightforward, accurate way of doing is to use quantum mechanics or the density functional theory methods to calculate energies of molecules. What is the problem? The problem is the applicability of these methods are not so great when the number of atoms in a molecule grows, right? For example, doing a density functional theory calculation on let's say 100,000 atoms, right? It's not possible today, right? So what do we do when the size increases? We start using approximations which are not so great, and then hence the quality of the calculation goes down, right? When the number of atoms or the number of particles, including the nuclei and the electrons, are small, right? This should move to the right a bit. So are small, then you can use these kind of methods and then calculate energies which are very, very accurate. But then if the system size grows, then we introduce further approximations, and then the quality of the energy goes down. So beyond a certain point, it's not even possible. So what do? We resort to methods like molecular mechanics. So molecular mechanics is simply again, I'm assuming, or I hope that you would be doing as part of this workshop as well. These are interatomic potentials, right? For example, if you take charm force field or amber force field and things like that, right? So molecular mechanics is our empirical potential energy functions with empirical parameters that will help you model molecules, right? So one of the major criticisms of molecular mechanics is that in terms of the accuracy, they are very, very low, right? So the objective, or one of the things that people have been trying to do for quite some time now, is to see if we can come up with what you call machine learning based force fields, or potential energy functions based on artificial neural network. Okay, so that are as good as quantum mechanics in terms of the accuracy, but in terms of the computational time, they are possibly close to molecular mechanics. Okay, so that's the idea of this, right? So we cannot do quantum mechanics is highly accurate, and then we cannot do it on really large systems, and then hence we do molecular mechanics, accuracy of which is lower. The idea is the hypothesis is that if you can come up with some machine learning model which can reproduce quantum mechanics or quantum mechanical energies, but then take almost as short time as molecular mechanics, that's the good way to go about. Okay, so this is one thing. It's been a while that we did this. We published this last year, right? So basically, we started. We came up with something called band NN, right? Band stands for bonds, angles, non-bonds, dihedrals, NN stand for the neural network. Okay, so we let's take let's start from here. Okay, so let's take this is a small, extremely small molecule. Okay, so for any computation that we want to do, right, we have to represent the molecular system on a computer. For example, Cartesian coordinates is a way to represent a molecular system, right? So for machine learning, it turns out that the Cartesian coordinates are not so really great for some of the reasons. For example, a same molecule with exact same internuclear distance, you can have multiple Cartesian coordinates representing that. Okay, so you want the representation to be agnostic to, for example, rotations, translations, and things like that. Okay, so we came up with this particular representation where, for example, if we have a small molecule, right? So how do we represent, right? No, if it is for every atom in a molecule, we have a 4-dimensional vector which indicates what atom that is. For example, H means it's 1000, C means 0100, and so on. Okay, so that's the first 4 dimensions. If I go to the second 4 dimensions, that captures the connectivity. In force field terms, we call this atom type, right? That defines the environment. For example, if I take this particular H1, which is this, okay, because it's a hydrogen, it's 1000. That's from here, and then this captures the environment. For example, this particular H1 is connected to 1 carbon and hence 1 here. And then it is not connected to hydrogen, it's not connected to oxygen, it's not connected to nitrogen. That's why 000. So maybe if I take this one, it may be clearer. Because it's nitrogen, that's this N4 here. Because it's nitrogen, that's 0001, that's coming from here. And then what is it connected? It is connected to 2 hydrogens and 1 carbon and 0 oxygens, and hence 2100. Okay, so similarly, so we can define each bond in this, in a given molecule using this particular representation. That's the 8 dimensions coming from here for the first atom, second atom, and then the actual bond length between these 2 atoms. Okay, that comes to 17 dimensions. Similarly, angle, non-bond, and dihedral. So you define every molecule as a list of bonds, angles, non-bonds, and dihedrals in this particular form. Okay, so now what do? We have Cartesian coordinates, let's say, that basically means that I have the positions of all the nuclei and then the atomic number of all the nuclei in that particular n-atom molecule. So now what do? I make a list of all bonds, angles, dihedrals, and non-bonds that are there in that particular molecule. Okay, so once I have that, what do I want? I want to predict the energy. So I want to predict the energy. Okay, so like for example, in the first place where we said, right? So this is my X, okay, and then this is our Y, right? So what is my F then? And what is here is my F, right? All the way up here. Okay, so that's my F. So basically, what is it we are trying to do? Given pairs of X and Y, okay, large number of examples of X and Y, we use the artificial neural network to emulate the function that connects these 2. Okay, so that's basically a simple idea of how this is done. I'm not going to go into details of the data set and so on, and just quickly give some results, right? Okay, so let's take this example. Okay, what we have in this slide are representative C11H22 isomers. These all of them are C11H22 isomers, right? And of course, randomly selected. At the same time, the examples represent reasonable diversity, right? Like the branched or linear or 4-member ring, 3-member ring, 5-member ring, 6-member ring, and all that. Okay. So what we have here, that's the energy, that's the DFT energy, that's the ground truth. Okay, so now we use this band and our machine learning model to predict the energies, and as you can see, the agreement is really nice. right? So as you can see, the agreement is very, very good. This is just to compare with some other semi-empirical method. That's the numbers that we get for the semi-empirical method. Okay, so as you can see that this is doing very, very well. And of course, this is much faster than both of them, right? Okay, so now, we can also show multiple aspects, okay, you are saying that, okay, it gives the energy of a given structure reasonably well, but then can it also reproduce the potential energy surfaces? For example, again, there is a comparison for bond stretching of different molecules, right? So the bond stretching using the ground truth and then the neural network one and so on is another set of bond stretch. That's the bond bending. That's like you take an angle and then bend it, and then you see how the energy changes. And again, there's a reasonable agreement, right? And then now, beyond that, we want to apply this to multiple problems. For example, I want to calculate reaction energies of molecules, right? These are some of the reactions, okay, common reactions from organic chemistry. And now you calculate the delta E for all these processes, and then that's the DFT numbers. That's the machine learning model that we have, and then that's the AM1 method. You can see that it's even getting the sign wrong sometimes, and then the magnitude is just half of it, and so on, right? So it's too bad. Okay, so that's just one example of predicting molecular properties. So in this case, what I have shown is prediction of energy of a system, right? The density function theory energy of a system, right? But similarly, you could apply it to multiple other molecular properties. It could be, it could be binding affinities. It could be, anything that you want for your purpose, any molecular property. Right. Okay, so the second example that I'm going to show is prediction of solvation free energy of or drug-like molecules. So let's say I have a bunch of drug molecules, or let's assume I'm designing a drug molecule, right? So we do all know the importance of the pharmacokinetic properties. For example, whether that molecule will get absorbed into the system or not. Whether it has the right solvation properties for the distribution, or whether that particular molecule can undergo what do you call can they pass a lipid bilayer membrane and so on, right? So all of these things, solvation free energy or the hydration free energy, is a very important property that affects all of these things. Okay, so in this case, in this method, what we have proposed here is to be able to predict solvation free energies exactly, and not only that, and is it possible to make this model chemically interpretable? So what does it mean? If you take any or most of the machine learning models we have today in terms of predicting something, right? So it's not so they are not explainable, right? So the fundamental premise of in science, right, we always want to understand things, right? Not just getting the answers correct, but then the process by which we get the answers correctly, right? So in science, that is very, very important, right? We want to understand things. If we understand things, maybe we can make it better and so on, right? So machine learning models, the problem is most of them are not interpretable. You cannot really understand much, even if they are very, very efficient in terms of getting the answers right. Okay, so we wanted to see if we can come up with something which is at least chemically interpretable, at least show that the model actually is learning some chemistry, right? Okay, so as you can see, we also have this model can take any generic organic solvent and then can predict the free energy of solvation. Okay, so we use what is called a graph neural network, right? We will not go into the details of this architecture, but just to give a brief brief description, right? So we have the solute, right? Let's say that's an organic drug-like molecule, and then you have a solvent, any solvent. It could be water, it could be ethanol, it could be hexanol, right? Any acetonitrile, DMSO, it could be any organic solvent. Okay, so both of them are represented as graphs. Okay, and then we use the Massey passing and then use that to generate the features. Or combine the features from solute and solvent and then generate what is called an interaction map. It could be dot product of these two matrices and have the interaction map and then use all this information, the interaction map, solvent features, solute features, all of them, and then predict the solvation free energy. So that's basically what is being done here, right? So what is this interact? This is the one, of course, of course, when the training is done, it's completely end-to-end training, right? So all the weights, all of these matrices have weights, and then all those are adjusted, or all those are optimized to get the free energy of solvation. So now what we will discuss is this quantity, the quantity that we are predicting, and this one here, the interaction map for the chemical interpretability. Okay. All right, so first is this. Okay, so that's basically what is plotted here. That's the predicted free energy of solvation. That's the experimental free energy of solvation. And as you can see that we do extremely well, though the number of solvents are like close to 90, right? 90 different solvents, and then we do very well in terms of predicting the solvation free energy, right? Okay, and then the interaction map. So that's basically this here, right? So if we take that interaction map and then analyze what it is that it is saying, right? If we look at the numbers in that matrix and then see that actually perfectly explains in terms of how these two molecules would interact. So let's say that's your solute and that's my solvent. That's 2-aminoethanol and then ethanol, right? So if I look at the matrix corresponding to these interactions, so what this would mean is these are the 4 atoms of this molecule, and these are the 3 atoms of this molecule, right? So and then this would, for example, correspond to how strongly these 2 oxygens would interact. This would tell how strongly or weakly this carbon, that's this, and this oxygen, that's this, would interact. Okay, so these numbers, if you take them, they explain, or they capture the normal chemical wisdom. For example, if you take O and O, of course they can form hydrogen bonding. They have hydrogens here, and hence the interaction is very, very strong. If I take C, I mean, it's there's not, the polarity is very low compared to O and O, and hence the interaction is not so great. Okay, similarly, if I take N here and O, that is again high, right? So this particular interaction map explains the predicted solvation free energy trends very, very well, and we have several examples by which we show that it actually works. Okay, so let me see. I think I have 2 more examples. Let's just take one of them. Okay, we'll take this and then stop the next one. All right, so the next example that I'm going to show is again drug design, right? But what we call the de novo drug design. So what do we mean by de novo drug design? The problem is simple. So normally when we do computations, right, we always start from a molecule and then do a computation or do an experiment and then calculate the property of that particular molecule or measure the property of that particular molecule, right? But what is in reality, what is it that we are doing? We are asking the other way around. They're saying what I want is a molecule, right, with a certain property. So for example, let's assume, right, start from here. So let us assume that I start a problem saying, okay, I have this the SARS-CoV-2 main protease, right, and I have the main protease and I want to design a molecule, or I want a molecule which binds strongly to this particular protein, right? So the question I'm asking is, I want a molecule, right? That's my requirement. I want a molecule that will bind to a given protein very strongly. But then when I go, what is it that we are doing? We are taking a molecule, or we are assuming something and then calculating a property, right? The actual question we are asking is this: I want a molecule with a certain property, right? That's what is called a inverse problem. These are what is called inverse problems, right? Or inverse design. Or we want to answer this question directly instead of asking the reverse question, right? So we say that I want a molecule with a certain property, and is it possible to get something which will do that? Okay, so that's basically the question. Okay, so this is a paper. This I showed in the initial slides. Okay, so this is a method called reinforcement learning in machine learning, which helps us do it. Right? That's one of the methods. There are also other methods, right? So we have two machine learning models here in principle here. Okay, so one is what is called a generator, G, right? Generative model, let's say, and then second one is a predictor or a predictive model. So what does this generator do? Generative model, this model basically proposes new structures. Okay, that's what we want, right? What I want this model to do is to propose new structures. That's exactly what this is capable of doing. But then I put a constraint, or I want a bias. What is the bias? I want molecules such that they satisfy a certain condition, that this particular property that I'm looking at has to have a certain value, let's say, right? Okay, so basically, the reinforcement learning, helps you do that. You have the generator and then the predictor, and then it generates the molecules, and then the predictor model predicts the property of the generated molecules. And then if the property value is close to what we want, then it gives a reward to the molecule to the model. And if it's not what we want, it can give a negative reward or does not give a reward. And based on that, this particular molecule will slowly start learning to generate molecules with a specific property. I'll show an example. Okay, so let's I'm talking about two properties here. One is the log P that I'm assuming most of you would know. Log P is a measure of the partition of a given compound in two layers. One is water, second is in one n-octanol, right? And that value helps us decide whether a given molecule will cross the lipid bilayer or not. Okay, so typically this value has to be between 0 and 5. And second is, when we generate new molecules, one should be able to synthesize that molecule in a lab, right? So this score called as the synthetic accessibility score, right? That, it's an empirical score which lets us, assess whether it is easy to make a molecule or difficult to make a molecule. Given a structure of a molecule, it will tell whether it's easy to make or difficult to make. Okay, so that's the synthetic accessibility score. Okay, so now, what we did was we tried to use these two properties and then say, right, so what we have is the log P value, right, and then what we have here is this synthetic accessibility score. So if the log P value is between these two blue lines, that is, let's say between 0 and 5, then the molecules can enter or can permeate the lipid bilayer or enter a cell, right? So if it is above 5 or below 0, then they cannot enter the cell, and hence, we don't want those kind of molecules. Okay, and if we do a normal generation without any constraints or without any bias, this is how the distributions look like. Okay, and similarly, this is the synthetic accessibility score. If it is here somewhere, then it's very almost impossible to synthesize those molecules. And if it is here, then it is easy to synthesize those molecules, and hence I want all the molecules that are being generated to have a lower assay score value, right? So this is how the distribution looks when there is no bias. So now I start giving rewards for generating molecules in this region, or giving rewards by minimizing this number, let's say, right? So that's how it looks like. That's how the distribution changes. Okay, so at some point, as you can see, all of the molecules that are being generated are in this particular range. And similarly here, as you can see, all the molecules that are being generated are having a lower assay score value. So that's basically de novo drug design. So again, you can change your objectives to multiple other things, and then you can also change your reward functions in ways, to suit, to, increase the probability of generating molecules with desired properties. For example, log-P, or the number of benzene rings, or the melting point of molecules, and things like that. It's in principle possible to apply this to a multitude of other objectives, right? Okay, so as I said, that's just one particular, what do you call, one particular example, right? So just to quickly summarize, the modern machine learning methods, right, so that are being developed recently, are fundamentally changing the way we do computations, or the way we do applied, or way we do apply, theory, or the theoretical knowledge in the, in general all areas, but especially, we took example of healthcare by taking the risk stratification of one example and designing molecules as the second example, right? And the last 3 or 4 years have shown that these methods have a great potential to make the design and development process very, very efficient, right? So with that, I will stop. Right, people who are interested in working on ML for drug design, please do feel free to write to us. Just want to discuss, or to do long-term internships, or PhD and postdoc fellowships. All of these are possible. Okay, so with that, I will stop, and then maybe take some questions if there are any. Okay, so with that, and I should also thank for all your attention. It's been a long lecture, actually.
Thank you so much, Dr. Deva, for a very informative and very nice talk on machine learning and artificial intelligence and deep learning. This is all very useful because this is the future of drug discovery. So if anyone present here, they want to ask any question, they are welcome.
Hello, sir.
Hello, hi.
Sir, this is
yeah.
Sir, this is Banu Rekha from Naipur Gahoti.
Yeah, please tell, Banu.
Sir, what is the major difference between the data mining and machine learning?
What is the difference between data mining and machine learning? So data mining, see, there are, once you have a large amount of data, right, there are multiple other things, multiple things you can do to generate new knowledge, right? So one is, for example, I have a large dimensional data, let's say, right? So there are many ways I can look at the data, like just to visualize this data, for example, right? Because when we are all used to maximum of 3 dimensions, right? If I can plot something in 3 dimensions, we can look at the object and then understand multiple things, right? If there are 5, 6 dimensions, maybe we can do like first 2 first, and then we can do multiple things. Or we will resort to dimensionality reduction like PCA or TSNE and things like that, right? What if you have 100 variables, or 1,000 variables, or 1,000,000 variables, right? Your y is not a function. We discussed about x and y, right? So what if y is a function of not just one of them, one x? It's like x1, x2, x3, x4, all the way up to x million. Okay. There is absolutely no way that we can perceive or comprehend how the function y, or how the output y changes with respect to all these different dimensions, right? So that way, machine learning, algorithms help you recognize such kind of patterns in larger dimension, right? So, that's basically, that's the, major strength of machine learning, that it can process data. One is large amount of data. Second is complex in terms of how many variables that thing is dependent on, right? The complexity of the data itself, right? So that way, it can learn from the existing data. Data mining is slightly different, and of course, this overlap also. It's look like visualization of the data, or you have classical ways of looking at data and learning from them, right? So recently also we have submitted some paper on how one can use the, represent the protein ligand complexes in form of graph, and then, use data mining techniques to, look at, graph transactions and things like that to, learn the protein ligand interactions and then be able to design new ones. So in both, machine learning are much more, efficient in terms of looking at the large dimension data. Okay, so data mining is a little bit more classical.
Sir, thank you, sir. I have one more question.
Yeah, please. Yeah.
Sir, what are the tools or algorithms used in machine learning?
Tools. I mean, there are some, some libraries that people put together just for drug design kind of purposes. For example, Molecule Net or the Deep Chem and things like that, right? There are some which are available that one can use like in a modular kind of basis, right? But then because of the availability of large number of these libraries, right? So if you start from PyTorch, that would be the easiest way to go. It gives a lot of flexibility, one thing. The second one is, it's more like Python, right? But then, the programming or the coding that we talk about today is very, very different from what we spoke about coding even 5-6 years ago, right? Today, the coding is you don't write the code. Somebody has already written the code for you. You just have to put things together, right? So that way, it's reasonably, not so difficult to start from, Python and then move to PyTorch and then start doing things, right? Yeah, actually, if you go to our lab GitHub page, there are at least 2 problems where for which the if there is a data set, there is a solved exercise, and then how to do it, right? For example, the first problem that I discussed, if you take structure and predict energies, okay, so that kind of problem, if you're able to solve that one in our GitHub page, right, you will be able to do. And then second example that we have is, I think, to read a protein sequence and then, do a secondary structure prediction. That example also is available, solved example with a lot of, headers and everything. You must be able to, look at it. In any case, I mean, there are people to help also, right? If whoever is attending, if you want to, spend some time here, of course, not at this time, but maybe later during the year, yeah, spend some time here and then learn things, right? And all of that is possible. Yeah. And nowadays, finding help.
Our students will be interested to do that. So, so any other question? Anybody have?
Hello, this is Ronita from IIT Kharagpur.
Yeah, please go ahead.
So the equation y equals to fx, there x are the descriptors, right? Molecular representations.
Yes.
So is there any limit as to how many descriptors we can use, or is there a correlation as to the number of descriptors to the number of compounds that we are using in the data set? Like statistically, will it make a difference?
Yeah, I think I got the question. So good question again. So, how do we choose the number of descriptors, right? So let us assume that, like the first example, let's take that first example, right? That we do a risk stratification. No. So there in the first project that I spoke about, we chose 5 descriptors, right? 5 quantities or 5 parameters for the machine learning model. So why we chose only 5? Because there were 375 patients, right? So, we had to choose a small number of descriptors. Okay, so in the second problem, that the band NN that I showed, right? So we didn't even count the number of descriptors. So in that case, all the list of bonds, angles, diagonals, non-bonds, all of them are descriptors, right? So we didn't even count. Why we didn't even count? Because the data set was like 22 million compounds in the data set, right? So however number that descriptors was, it was not going to matter because we will never reach that kind of limit, right? So there is a correlation between the number of descriptors you choose for the modeling and the total data set, right? If your total data set is small, try to minimize the number of descriptors, right? Yeah.
Okay, and suppose we are using 3 or 4 descriptors different. Now, what if the descriptor 1 and descriptor 2 are correlated? Won't it overestimate the Y value?
Yeah, so yeah, absolutely. Yes, that's also again a valid question. These are considerations that one has to keep in mind while choosing the descriptors, right? If I have 2 descriptors that are perfectly correlated, right? If they have let's say a perfect linear correlation, there is no reason why you should have both of them, right? It's enough if you have only one of them, right? So this is what we call the pre-processing. You have to be able to check the interdependencies. Sometimes it's actually useful to take a ratio of a couple descriptors than to take them individually. So one way you are actually reducing the number of descriptors. Second way that your model is going better. Like we again, that same problem we found several examples, and others also have found, right?
What kind of tools do we use to screen these descriptors or to find out if they are correlated?
So there are these methods like how do we
if you want to use some commercially available tool, you can use Codesa to screen.
Okay. For the molecules, Codesa, of course, yeah, that one.
Then you can screen the
Codex. Can you repeat the
CODESA? That is a commercially available program package where you can screen your descriptor interdependency, and you can also do artificial neural network calculations and all. But free tools, I am not aware currently. Okay. Of course, if you go to Professor Deva's laboratory, he will give you a code and then you can do that, but like he is doing. But if you want tools and available tools, then I am not much aware. But Codesa, I am aware. Codesa is doing that. But we can write our own code to do that.
Okay, thank you, sir.
Any other question? So many people are watching on YouTube and they are writing some questions. So most of them is not very clear question. One question that the way what he's writing, what way we can use AI and machine learning in drug discovery practically in our laboratory. So probably he's having some lab where he has some patient data and all, and he want to use machine learning and AI practically.
I don't know what you mean by practically, but so I mean I don't know what the I don't know what was the thinking behind that question. I don't seem to understand. But then okay, let me answer a very generic kind of reply for this. What do we mean by practically using in someone's lab, right? So we nowadays I'm sure in 10 years or already there is a lot of systems, right? Whether it's equipment or computational systems, right? A lot of these systems are already equipped with at least one level of AI-based, approaches, right? So this is only going to, increase in the next coming years, right? So if the question is about you want to use AI for a completely new thing, that's as I said, we have at least couple completely solved exercises in our lab's GitHub page. Please go through that, right? For that, you don't even have to download and run. There are links to Google Colab on which you can run the codes directly, right? You just by clicking, you can start from click the arrows in the Google Colab and then see what is going on. And then next step is to understand the code, line by line. It's not so very complicated, so you must be able to do, right?
So we have 1 more minute, and then there is one question which is very general question. Is there any limitation for machine learning?
Oh, absolutely, a lot of limitations. Yes, so of course, when especially in the healthcare domain, right? So there is a large number of concerns, starting from the privacy or ethical issues, and then fairness, right? For example, let's say the algorithm that we develop for risk stratification, right? If it will be useful only if we can show that it works on all kinds of populations, right? So let us assume, because this data comes from a specific hospital, right? It should not work only on the patients of that particular hospital, or let's assume that data comes for population from North India. Does that algorithm work as well for the South Indian community, or the Northeast, or the East, or the West, right? So does it work on all of them, right? So without confirming all that, we cannot, for example, use that in a hospital, right? So if we do that and it works only on one population, that means we are already not fair to a set of population, right? So, and then second is like, if we take data of a given patient, for example, right, we have to be like super sure in terms of, protecting the identity in all forms, right? So privacy of that particular patient, right? Because they, volunteer to give this particular data, and that should be only, the best that we can do is that this does not affect them, right? So they should not go, it should not affect them in any negative way, for sure, right? So these are some of the higher level concerns, right? Like the fairness, ethics, explainability, transparency, right? All this, especially in healthcare, right? Because I don't want anybody to know what my health complications are, right? I don't want anybody to access my health records, right? So that I'm sure all of us will feel in some way or other, right? So if I volunteer to give it to, let's say, you, to say that, okay, you think it's useful for your research and your research is useful for the community in general, okay, I will volunteer to give. So, but then, it's the person who is taking the data's responsibility to absolutely be super safe about all that, right? So, in that sense, a large, there is a, there are a lot of issues we still are understanding, right? In terms of what, issues and what, what implications are out there, right? For this, especially this area, okay?
So is it okay if I take one or two more questions, or your time?
Yeah, please, no problem. Yeah, it's fine.
Okay. So many people are writing. So one person, Shubham from Naipur Mohali, he has written, "Sir, can we apply machine learning for toxicity study that arising from drug metabolism?"
Yes. So given that, especially for toxicity, there is some data available, right? We can predict. Maybe I didn't show a couple other examples. So one of them that we have done recently, which I hope we will submit soon, some point, is drug-drug interactions. For example, when we are designing a new drug, right? One of the most important things that we think about is how strongly this drug will bind. And then the second thing possibly we think about is the toxicity, right? The drug-drug interaction is something that we analyze post facto, very, very later at the stage of drug design, right? So if we have, if you're designing a new drug molecule, and then if you're able to use AI or any method, for example, for that matter, and then take that drug and then see if it would interact with any of the existing drugs in the market, right? And if it does interact, what are the kinds of interaction that it may have? Like, is it pharmacological, or does it, does it positively work towards, like, give an example, right? You have aspirin, is a drug, very old drug, we know. And then warfarin is another drug. Aspirin, if I take aspirin as a analgesic, warfarin as a blood thinner, okay, that's not so good, because both of them can work as blood thinner, and hence can cause intestinal, internal bleeding and so on, right? So if we can identify these things during the development of the drug, right, then it's useful. We have to consider all aspects, not just only binding, right? Binding is one aspect, a very, very important aspect, but still it's just one aspect. So in that sense, toxicity can also be predicted. So now I think a lot of studies are there. I think someone is also going to talk about it later, that you can predict, can classify molecules as whether they are toxic or not, if they are toxic, in what way, and things like that. Yeah.
So the last question will be, in interaction map, could it able to consider external factors? This is what it is written. Could it able to consider external factors?
Yes, so that's a good question actually. So in the interaction map, what we are doing is we are trying to use that block to see if that particular block is explaining the interatomic interaction between solute and solvent, right? So now I can go back, now this thing works for solid solvent. I can go back and then change the solvent to a protein. Right now, I can look at the interaction between a drug and a protein. Right. So for a given drug molecule, I have a protein molecule. I can get the interaction map, and then now instead of asking, whether it is understanding, now I can ask by looking at each of those matrix elements. Now I can ask saying if I have to increase one particular element and then decrease some other element, right? So how can I change the drug molecule such that I can increase or decrease, right? So those kind of things can be answered. So what I am saying is that this method can be applied to a large number of, we see that this can have a great potential for application in, especially in protein ligand interactions, and then optimizing lead molecules for making the molecule stronger, as stronger binder. Yeah.
Okay, so I think that we will not take any more questions. Oh, okay, fine. Okay, you can ask question. Yeah.
Good afternoon, sir. This is Abha Mishra from Naipar, Guwahati. My question is, how does deep learning contrast with machine learning?
Okay, deep learning is a subset of machine learning methods. Okay, so it's like see, there are several things which I did not tell. So you remember we spoke about. Maybe I don't know if my slide is still shared.
If you can share, it will be visible.
Okay, I think it's being shared already. I'm sharing it. Maybe you can. I don't know if people are able to see. I'm trying to go here. Can you see this slide?
Yes, sir.
Okay, so let's, this you remember, right? So basically what we said was I start from Cartesian coordinates and then I'm predicting the energy. And then I'm using this as my function, right? Yes. But then we are doing, I did not mention this part, right? So this I said is my X, and this I said is my Y, and this is my F, right? But why do we need this something in between that is not part of any of these, right? So one of the problems that the deep learning methods address is the need for this. Okay, so in traditional machine learning methods, what we do, this is what is called feature engineering. So this is my original input, right? I do some kind of engineering and I get some features out of here. So in that sense, these are my actual X. This is something else. Right, I have something, let's call them, I don't know, Z or something, right? So this is my, this is my original input, but then I have to extract X from here. So I'm extracting X from here, and then, looking at this relationship like this. Okay, so that's the traditional machine learning that we do. So deep learning methods say that, okay, you don't need all of that. You don't even need this. Okay, I don't need this. You just tell me what the output is. This is my X, this is my Y. I will figure out the in-between part. You don't have to do anything, right? So that's one aspect of deep learning where it can, you don't have to do any feature engineering, or, you don't have to apply chemical knowledge and things like that in between, right? And then another difference that people talk about deep learning is that the networks that we use, the artificial network that we use, are much deeper, that the number of parameters involved are very high. And hence, for deep learning methods in general, the data that is required is very, very large compared to the traditional machine learning methods. For example, if you have a small data set, the best is to stick to the regular, shallow networks, right? If you have large amount of data, then deep learning methods will, deep neural networks will help you achieve better accuracy. I hope I answered that question.
Yes, thank you, sir.
Thank you. So Hemant, I think you are muted.
So yeah, it was a very nice talk, Professor Deva. Thank you so much. And we could understand that so many questions are there and some very good discussion has happened. So thank you so much for all this. Now we are going for a lunch break, and after that there are so many requests from participants that they want to learn Python more. So instead of Hajme Hirao's talk, which will be postponed to some other days, we have requested him and he agreed for that. So we are going to have hands-on on Python at 2:15. So all hands-on participants, they should join by 2:15. And now we are going for lunch break. Thank you, Professor Deva. Thank you for your.
All right, Hemant. Yeah. Thanks for having me. Yeah. Take care, everyone. Bye bye.
Good afternoon, everyone. Welcome to the post-lunch session of International Workshop Molecular Modeling Tools and Techniques, MMTT 2021. So in this session, it will be a hands-on session on Python scripting, where Dr. Anvesh Pandey, who has already taken one session, he will take one more session and explain some scripts in Python. So Anvesh, this is up to you now.
Thank you so much, sir. Is the screen visible to all the participants? Can somebody confirm?
And put it on presentation mode.
Yes, visible.
Yes, sir.
Okay.
Okay. So good afternoon, all the participants, and it's my pleasure that Hemant sir gave me again an opportunity to share with you a few Python scripts in data mining using Python and in PDB files only. We'll be doing today itself. I have already mailed a few of the Python scripts that we'll be doing today. So I think let's start, and then during the session onwards, we will have a conversation, and then we can see what is the progress. Accordingly, we will move on. Okay. So let's start.
If anybody is parallelly doing it and having any difficulty, they can always interact, and sometimes they can share their screen and show if they are getting any error.
Yeah.
That is the meaning of hands-on. So any of you, whoever is doing it parallelly, you are allowed to share the screen, if required.
Thank you, sir. Okay, so today I have selected around 33 PDB files. So let's check. Meanwhile, we will also be getting am I audible? A list of all the files. So as you can see, we have 33 PDB files that we will be exploring. I already mentioned yesterday that it could be possible that some information is missing in one PDB file while other information is already present in some other PDB file. So we will be retrieving those informations using our Python scripts. Today we will be doing the scripting in two batches, like in two types of scripts are there. First of all, we have solo scripts, like for pH, we have one script. We have a similar script for resolution, a similar script for chain A, chain B, and then we will be grouping the scripts and making a bigger script. To bring out more and more data using a single script. So scripting from one information to two information to a handful of information to all the possible information at once. So let's start. So as we can see, the total count of the PDB files as shown is 33. So we will start with one PDB file, one script, and then move on. Okay, so starting with chain A. First of all, let us visualize the script and try to understand the structure of the script before moving on to how it works. Yesterday itself, I demonstrated that the very first line is import sys. Import sys, it imports the system module of the Python and interacts the user's language, what user has written inside the script. It tries to interact that with the computer. So whatever you can say, it's like it acts, this line tends to be as an interpreter between the user side and the computer side. And accordingly, we will see how this works in the further lines also. In the second line, we have defined a system argument. System argument as one means whatever there is written in first line, it has to call it. So every script, it does some function. Here it is doing calling the first line. The third line means its F is open function and read. R stands for read. Yesterday we saw a script that we were calling a function W. That W stands for writing in a text file. Here we are reading the FN. FN stands for the files that we will be calling. We have not imported the globe module here. We'll be doing this in the later part of this session. While as the session progresses, we will see this more in detail. Then we have defined a variable CHNA. This CHN stands for the chain length of A. Can everyone see this CHNA variable? Coming to the loop part, while one, what it has to do, it has to read every line. It has to read every line, and what it has to find, it has to find a pattern. See, it is very important to notice that in all the scripts that I have already shared with you people and that we'll be discussing in today's session, we will always try to find a pattern. If that pattern, we will define that pattern in our script. If the script matches our PDB files with that pattern, it will extract the data of the mentioned lines. So we are not doing nothing, we are just using Python to write a script for matching first of all a particular pattern, and if that pattern matches, we will extract the data out of it. It is very simple. Like suppose if you it's like recognizing somebody, whether the person is short or tall, whether the person is thin, like this. So first of all, it will try to match the defined pattern. If the scripts find something like that in the description of the PDB file, then only it will move forward. If it doesn't find any pattern like that, so it won't produce any result, it will produce an error. We will today demonstrate some errors also as to what if the pattern is not found in a PDB file. In that case, how the script will behave. So let's first of all execute this script and then move on to what are the possibilities. Okay, since we have not called the globe module inside our script as already mentioned, there is a possibility that we can define all the PDB file at once or one by one. So we will do it both the ways. First of all, we'll be calling a single PDB file. Suppose we have called 3bsa.pdb. So it will bring the information only out of 3bsa file. What if we want to find out all the information from all the PDB files? Simply, we have to put star.pdb as an extension over here. And now let's see. What if somebody wants that, somebody is confused that whether all the PDB information is displayed here or not. So first of all, let us count the total number of PDB files, which can be done using the following command. So as you can see, there are 33 PDB files. Now let us check whether the chain a.py script was properly executed to all the PDB files or not. Both the times 33 informations, 33 outputs were displayed. There were 33 PDB files and the script read all the 33 files. Whether or not chain A information is present or missing. It didn't mind whether the chain A information is present or not. It will execute the script to all the PDB files because during the execution of the script, we have mentioned star.pdb. It has to read all those files which end with the extension PDB. Now let's move on to chain B and then club. First of all, let's look at the structure of this file. So as I already mentioned, it's doing nothing but finding patterns, and instead of A, it has now found a pattern. It has to found a pattern called B. For the chain sequence B, it has to find B. It has to append it. Appending means storing the information in a temporary variable and then finally displaying that using the command print. Let's read the program and try to understand it. Import is importing the system argument, calling the system through the argument 1 defined in line 1, opening the function, that is the PDB files, reading, R stands for reading those PDB files. Then we defined a variable CHNB for reading the information chain B information, and then the loop. While the system 1, while this condition holds, what it has to do, it has to read lines and such that the lines should have this information 0 to 6 SEQ RES and 8 to 10 as 1. And in that case, if this matching pattern is found, what it has to do, it has to extract the information present between line 11 and line 12 and append it into a temporary variable named chains. Okay, what we did in this line, 9th line, 11 to 12 information was extracted and sent to variable names chained, and later on this chain was transferred to another variable chain B, which we have already defined. We can omit this line, but it is easier. We have already to look at patterns. And later at the last line, it has to print the function, whether what is the name of that PDB file and the variable that we have already defined for chain B. Now let's execute this script and see how this is different with chain A script. So we can see that while executing the chain A script, all of these PDB files had chain A information. It was very rare that any of the files do not have chain A. It has to be there. Mostly they have chain A information. But we can see chain B is missing in some of the files. Like in the second one, there is no chain B information. In this 4BUZ, there is no chain B information. Similarly, over this file, and this file. But the script is well operated on all the PDB files. See, all the 33 PDB files that are present in the current working directory were well executed by the chain.b.py script, whether or not the chain B information is present in them. So whether they have chain A or chain B or both or either, the script will always execute. If the information is present in them, it will display on the terminal or in the stored file whatsoever. If the chain A information as well as chain B information simultaneously and bring them out. So let's see a script first of all. Okay, this time what we have done, we have made a slight change in the script. We have used a else if command, and this else if is used in such a way that if chain A is present, display chain A information. If chain B is present, display chain B information. If both are present, display both the information. If none is present, display none of the informations. So let's first of all execute it, and then we would get to know how this works. See, so let us execute A and B alone so as to make a comparison. This is A, this is B. So chain A is present in all the scripts. Chain B is present in chain A is present in all the PDB files. Chain B is present in a few of the PDB files and is missing in few PDB files. So individually these two scripts were operated so as to find only chain A or chain B information. But when we combine the two scripts, it displayed simultaneously displayed both the informations, that whether chain B is present or not, or chain A is present or not, or both. So as we can see, like in the second one, only chain A is present. Rest of the files, mostly chain A and B both are present. Again, we have only chain A information, B is missing, chain A, chain B is missing, A, B is missing, and likewise. If suppose some of the PDB files have some other information missing, accordingly a blank space will be dedicated to them and no information will be printed at that very particular place. Is there any confusion to anybody in these three scripts as far as the execution and is concerned and visualizing the information is concerned?
What difference we will see if we write W, like write in place of read or R?
Okay. Yeah. Who is this? Please, I would request all the participants to first give their names and affiliation, and then obviously you can proceed with the questions and queries.
Sir, I'm Sanjana Roy Chaudhary from Naipur, Guwahati.
Okay. Thank you, Sanjana. So Sanjana asked, what if we want to write or not? It is very easy, Sanjana. Like yesterday, we had seen a few PDB files in which we called import globe and displayed. Let's see a script and then we will. Sanjana, can you see the third line?
Yes, sir.
So in this, what it does, it creates a file, text file to save the output. Okay. We don't want to do this as of now. However, since you have put a query, also this file putting this arrow and writing some file name. Suppose I write A or b.txt. So now it will store the information in a file named a or b.txt, which can be visualized by VI editor. See. So there are a handful of ways, whether you want to do it through terminal or you want to do it through Python or you want to call it or not, or you want to display it or not. It all depends upon the user and the problem that user is addressing. Is it clear, Sanjana?
No, sir.
Not yet?
No.
Okay. So let's write that and see if we can clear out Sanjana's doubt.
Sanjana, what is the query?
So like in place of R, if we write W, so what difference we will see in this?
So that is read mode and write mode, Sanjana. R means read and W means write.
Yeah, so if we write the W mode, like if we write the write, then what changes we can do and how will we do that?
So whatever information, it will be printed on the terminal, no, that will be saved in a file. So instead of printing it, it will save in a file.
Yeah.
It will create a file, whatever file, and it will write the output in a file.
Like in a separate file, it will.
In a separate file, whatever file name you will give, it will write into that file. Okay?
Okay, sir.
So we did a few chain A and B scripts. Now let's move on to see whether the experimental technique, what experimental technique was used in order to create that PDB file. Again, we are what we are doing is we are importing the system module. We are reading the first line, that is through system argument. We are again reading, as Sanjana asked, we are only reading. R stands for read mode. The file has to do it has to read the PDB file. If we were to call a W, that will be writing output in a pre-called file and saving that file. So we are just reading as of now. We are not writing and displaying the output on the terminal itself. Again, this time the script has to look for a pattern 0 to 6 as exp data, and it has to extract the line 8 to 30 and save the data into a variable defined as exp. So let's see how this file works. So we can see that all the PDB files were, the crystal structures were obtained through X-ray diffraction studies. So this was the data that we were to obtain. Is there any confusion with this program? Any confusion? Anyone?
Hello, sir. This is Monica.
Yeah.
Sir, now you extracted this one experimental data, right?
Yeah.
So you didn't use append. So, like, we use it or we don't use it, we get what we want, like in a printed form.
Yeah. Sir, it's a good question. Sometimes we'll be using append, sometimes we don't use append. It all, see, I said it's a very good code is that code which has the least number of lines written in it. Okay. The less the number of lines you'll be writing in your code, the less it will take to execute, and the less if there is some error you have to debug. Like suppose you wrote a code in C language and it took you 30 lines. You write the same code in Python, it will take you, like say suppose 3 or 4, maybe 5 lines. Is there if there is some other possibility, if there is somebody else who can write the code in 2 lines. So likewise, it is user to user, I would say an experience thing. Other than that, there is no such, I don't think so. There is anything else apart from this. Whether or not you can use the append or not, and then the code should work. If it doesn't work, you can include it. If it works, it's good. Like in the previous script, we didn't use the append thing. Other than that, we are using it, and both the ways the code is working fine. So this is another lesson for you people, another good point for you people. So as to test I have already shared a few codes, so as to test whether the append thing works with it or not. If it works, it's good. If it doesn't work, you can include the append thing and then move on with the scripts. So always remember, a good code is that code which has the least number of lines. So because less time in the execution, less time to crack the errors, and accordingly it will be easy to write and understand also. A 30-line code is difficult to write, difficult to understand, difficult to debug. A 10-line code is comparatively easier for all the aspects. A 5-line code is much easier in all the aspects. The less the number of functions called, the less the number of variables called, the more the number of information displayed, the better the code it will be. So this is the only difference, coding to coding.
The append method adds an item to the end of the list.
Yeah.
And if you want to add some item to the end of the list, you use append. Otherwise, you don't use it. Anyway, so you explain the code.
Yes, sir. Thank you, sir. So let's check whether this code works or not. This time, we are executing the code to bring out the gene information from all the PDB files. See. So what we did was, we had to extract data from 17 to 57th line. If something is present, it will be displayed. As I already mentioned that whether or not some PDB files could have a data, some PDB files might not have data, the data in that very particular section. So it is very much possible, it is very much likely that you might be displayed with some data, or you might not be displayed with some data. So all that we are doing is to extract the data from the PDB files. As in the chains we saw, whether they have chain A or chain B or both or none, accordingly the information will be displayed or can be saved in an external file, text file, or whatever with the extension of the file that you want to save in. Similarly, if the information is present, it will be displayed. If it is missing, none of the information will be displayed. So as we can see, in the mentioned lines, no information was present for these files, whereas there was some information present for these files, and accordingly the extracted information is displayed. We will now try to do a few fast scripts and then move on to see whether we can execute data in batches or not. So let's bring out ph. So let's look at the PH scripts first of all. What we did, again, system import system argument 1 with functions open, that is with the file which is PDB extension, open the file, R go to read mode and read each and every line of that file. This dot R read line stands for reading the file line by line. It could be writing, it could be reading. So what we are currently doing is, since we have to match a pattern and extract the data, this is our prime target. So we have to read it. First of all, we have to read so that the computer could find the pattern. What pattern? The defined pattern. And what is the defined pattern? 0 to 6 should be remark and 12 to 14 should be PH, capitals. See, it is very important to keep in mind that it is case sensitive. It is very important to keep in mind. PH should not be kept in small. If there is a colon, whether there is a dot or hyphen or space before the word, after the word, whatever pattern is exactly there, that only we have to look for. And accordingly, it has to extract information between 45 to 48 lines and then store that. Let's look at a PDB file as to how see, can everyone see the highlighted line? So we can see the resolution is 1.60. What is the pattern? 0 to 6 should be remark. 14 to 23 should be resolution. And it has to what? Extract from 50 to 53. So likewise, we select the pattern. Similarly, in case of resolution, it has to find a pattern from 0 to 6. There should be remark. From 12 to 22, see, resolution ending with a dot. Resolution not ending with a dot would result in the extraction of some other information which we are least interested in. This PDB knows. So this is to differentiate in this kind of, for the user's easy point of view, they have put a dot towards the end of it, so as while the extracting the data, while retrieving the data, the user would easily put a dot towards the end of it, and eventually it will give us the resolution information. Similarly, pH information. Similarly, any other information, whether the whatever be the user interested in. Let's look at the resolution file first of all. 0 to 6 remark, 11 to 22 resolution with a dot. As I mentioned, resolution ending with a dot. And what it has to extract? The data that is in between 26 to 30. Why? Because we have called a system. System has to operate and as the argument number 1. It has to open. Open means open the file, specified file that is defined by function. It has to read. Read what? Line by line. It has to split the entire PDB file into lines and read the entire PDB file line by line. It has to then look for the pattern. What is the pattern? 0 to 6 is remark. 11 to 22 is resolution. Resolution must end with a dot. Resolution must end with a dot. Let us do one thing, deliberately make a spelling mistake in the resolution word and see if this script still works or not. So what I am doing is, I am writing some, say, I am putting another dot over here, and let's see if what happens. See, no data is extracted. Why? Because it did not find any such pattern. Now let's correct it and see what information does it present. See. So it is very, very important to keep the pattern exactly the same as seen in the PDB files. If the pattern doesn't matter, the data displayed will be very irrelevant. It could be null and void. It could be misleading. So it is very important to look for the pattern. It is very important to look for the number of lines. It is very important to look for case sensitivity and likewise. Now let's use 2 more scripts and then we will move on to batches. Likewise, all the previously discussed scripts, this script, what it does, it again imports the system module. It reads it has to do is import the system module. It has to open the function file. It has to read the file. We have defined a variable known as syst. Then it has to read line, that is, it has to open the PDB file. Each and every PDB file has to be opened and is to be read line by line, split into lines and read line by line. Why? And it has to look a pattern. What pattern? 0 to 6 must be source. 11 to 29 must be expression underscore system with a colon. Expression underscore system with a colon. And what data it has to extract? The data present between 30 to 47. Then appending, as sir mentioned, it has to display towards the end of the file, towards the end. So append we use to display the data, the output towards the end, and finally printing it and displaying it over the terminal itself. So first of all, let's see how does this work. See, excuse me. So all the data is for Escherichia coli only. In some PDBs, the data is missing, whereas in some PDB, the data is present. So as I have already mentioned, whether or not the data is present or missing, it has to bring out of it. It has to bring the data out of it. If the data is present, it will display the output. If the data is missing, it will leave it blank. So it will read all the PDB files in the mentioned lines, specified lines. It will bring the data out of it if present. If absent, there will be nothing to display. It will be blank. Let's look at another script. This time we are interested in looking at the title. So just a few minutes ago, a candidate, one of the participants asked a question, what if we write R instead of W? So this script's answer that question. What we did was we imported the globe. I already mentioned yesterday that globe module, what it does, it brings out the path. It is used to call the path. It is used to call a particular extension of the file, that is PDB or log file or CSV file or MEGZ file for Mastro or whatever be the kind of file that you are dealing with. Globe module is always used in order to call the file in batches. Like this script can be executed in 2 modes. We can either mention this commented line to display the output on the terminal itself, or if we uncomment the line, we can display the output in a text file named title title.txt because it has to write, that is W. Since this line is presently commented, so we will look at this script in 2 ways. So let's look at the script in the way it is kind of presented as of now. See, since we have commented the writing part in a text file, the data is displayed on the terminal itself. What data? The title data. How the title? It has been extracted. How it has been extracted? As we have already seen in the previous scripts of chain A or chain B or simultaneously chain A or B or gene or the experimental procedure or the residue pH or the system or the resolution or anything. So whatever we did up to now, in the similar manner, we brought out the data out of it, extracted the data and displayed. This script can be executed in 2 ways. One of the ways is already mentioned in the terminal, that the data is displayed on the terminal itself. Another way is we would uncomment this line and now the data would be displayed in a file named title.txt. See, no data is displayed. Whether there is title.txt file? Yes. What information does it hold? The same information as previously displayed on the terminal. So Sanjana, are you online?
Yes, sir.
So did you get your doubt clear as to what is the difference between the 2 modules, W and R?
Yes, sir. Yes, sir.
Earlier, only read and display the output on terminal. I told that as the session would progress, we would tackle each and every problem. But this time, what it did, keep following. Again, let's comment this line and see what happens. See, so the same file, the things are done in two ways. If you want to write in a particular text file and store the information, you can use the uncommented version. If you doesn't want so, you can omit it. It all depends upon the user's preference. So again, I am reciting the functions in the script. Import globe. It has to, what it does, it calls all the mentioned extensions, whether PDB or log or whatever be the extension that the user is interested in finding, extension or path name or file name, we use the globe module. Why do we use it? Because suppose if we have data in bulk, if we have 100 files, 200 files, 500 files, 1000 files, how do we do it? How do we write the star.pdb, or how do we 10,000, for mentioning the 10,000 PDB files? It's a very difficult task if you have to do it manually. So what do? We just simply import the globe module, and we mention what do we mention that globe has to do? It has to read all those things that are ending with the extension PDB. If somebody is interested in reading Gaussian log files, they should write it star.log log. If somebody is interested in reading the Gaussian com files or GJF files, they can mention their favorite extensions, whether com or PDB or log or whatever instruction they are interested in. Now it has to read what? The called file. This was done already in previous all the scripts. It has to read all the called files line by line. This read line means read all the files line by line. Why do we want to read all the files line by line? So as to look for a particular pattern. What is the pattern? That is 0 to 5 must be title. And if the pattern is found matching, it has to extract the data information present between lines 10 to 80, append the data in the variable named dt1, and then display it. So now, if we want to print that information, the extracted information in the terminal itself, we would simply comment this line. We would simply comment this line and the data would automatically be displayed on the terminal. But if we want the data to be displayed, saved in a file, so what do? We write another line, and it has to open what? A text file. Why? Since the text file is open, it will write now. Write whatever be the instructions given over here. So it's a very systematic procedure. You can see globe is calling the particular extension, system is interacting the system with the Python, then creating a text file, opening the text file, writing inside the text file, writing whatever is present in the PDB files, all the PDB files, because the globe module has been called. Now what to read? Since the function stands for the PDB files, has been termed as function. R is to read all the PDB files, dt is the variable defined, then we have the read line, split all the PDB files into line by line and then read them line by line. Read what? Read the particular pattern, read for a particular pattern. What is the pattern? 0 to 5 must be title. If such pattern is found, what to do? Just look for another data extraction between lines 10 and line 80. Extract the data between line 10 and line 80, append it and then print it. Since we have already opened a file named title.txt and we have to write inside it, so this time whatever the data that has to be printed on the terminal, instead of being printed upon the terminal, it will be written in the title.txt file, and close means save the file. So we opened the file, we wrote inside it, we wrote what? All these instructions, output obtained from all these instructions, and then at the last we close the file. Once we close the file, we would execute it. So again, let's execute it. Instead of displaying the data on the terminal, it will display the data in the file name title.txt. Fine. Now let's make some scripts for batches. So I would remove these solo scripts. Okay. So till now we have seen we were bringing the information one by one. That is starting with chain A, then with chain B, then with moving with chain A and B simultaneously, then moving with pH, then resolution, then system, then gene information, then title information, and so on and so forth. Now what we would be doing, we would be clubbing the scripts. The better script is which can there must be a single script and it could bring out all the informations. Writing separate scripts, smaller scripts for each and every kind of action that we want to perform, we would prefer that we would write a single script and that script should bring out all the information instead of bringing out writing different scripts for different operations. So let's first of all look at one such script, a small script, and then we would try to enlarge the content of the script by adding more and more informations into it. So the smallest script into this is pH resolution and experimental data. See, earlier the scripts they were displaying, since no globe module is called, so we will have to mention the extension of the file. Okay. Now what we did earlier was we were giving it the instructions to do actions one by one. Like we had a separate script for pH, we had another script for resolution, we had another script for experimental data. But this time what we did, we joined all these three scripts at once and made the Python to interact with the computer so as to read the PDB files and look for three patterns at once, look for three informations at once, look for three data's extraction at once. That is, if 0 to 6 is a remark and 12 to 14 is pH, extract the data between 45 and 48 line numbers and save it in a variable named pH. If the 0 to 6 is a remark and 11 to 22 is a resolution with ending with a dot, extract the data present between lines 26 and 30 and store it in a variable named res, that is for resolution. If the information is 0 to 6, between line number 0 to 6 is exp data, that is for the experimental data, that is what was the experimental procedure to bring out that PDB file. If such pattern is found, so what it has to do, it has to extract the information present between lines number 8 and 30 and store it into variable named exp. Now let's see how this script works. See, as mentioned, the output will be displayed in following order: pH, then resolution, then experiment. Another freehand that we get is in whatever order you write the commands, the instructions, the whatever order would be mentioned in the print command, that only in that order only the output will be generated. So in this we have first written pH and then resolution and then experiment. If somebody wants, they can change the order too. Let's do it and see what the output we get this time. Suppose I want resolution, experimental data, and then I want pH. What it did, the very first column refers for the function, this for the PDB files. This refers to, this refers to the resolution of the PDB files. The third column refers to as to how the experimental procedure was done in order to retrieve that PDB, and this time we were tend to display the pH information towards the end. We can see that pH information is null. Null means no pH information is available in that very mentioned lines. So it is all up to the user also that in what order you want to display. In whatever order you can mention your commands, you can mention the instructions, but finally the output will be displayed in that order only in which you have mentioned it here. Suppose we again want to change the order. If some user is not interested in this order and wants the order to change, what he has to do is, suppose I want the experimental data to be written first and resolution to be written at last. See, the order has changed now. So it is all up to the user as to how you bring out the information. Now let us try to incorporate the gene information also. Let us try to incorporate along with the pH, resolution and experiment. We would now include the pH, experiment and gene information also and see how this script looks. Import sys, it has to interact with the system module. What it has to do? It has to look for system argument. Now how many arguments are there? There are 4 arguments. Which are those? The pH, the resolution, the experimental data and the gene. There are 4 variables or 4 you can say arguments are defined. So as many arguments you define it, you mention it in the system argument section. Again, the same thing it has to do. It has to open the function, that is it has to open the PDB file. It has to go in the read mode, that is it has to read the PDB file. Read for what? Look for the patterns as mentioned in line numbers 17, line number 12, line number 15, line number 18 and line number 21. The mentioned patterns are remark with pH, remark with resolution, experimental data and gene information. Again, I would say the same thing, that the output will be displayed in the order as preferred by the user. It is the user's choice as to in what order he wants the information to be displayed. So let's check how this script works. We saw that in the gene expression that the gene information was missing in a few of the files, so that is not displayed. First column refers to the pH, the second column refers to the resolution information, the third column refers to the experimental procedure through which the data is retrieved, and the last refers to the gene information as to what gene is involved. So likewise, you can increase the number of information to be extracted in a single script instead of writing single scripts for each and every information. So we began with 1 scripts at a time, and then we are now what we are doing is chain A, then chain B, then together, then all the chains, then along with all the chains pH, resolution, experimental data, gene information. So this in this way we can increase the information that we have to bring out of a PDB file. Let's now look at, instead of this, we are now trying to look at the residue information also. So what this scripts look like? We have called allinfo.txt file. Let's rename this. So what we are doing is, again, system module, it will interact the Python with the system of the through the kernel of the computer. Globe, it has to read a particular extension. If we uncomment this line, what it does, it will write the output in a file named information. Its extension is text. What will later on it will look for all the PDB extension files. It will open all the PDB extension files in read mode. It will split the lines and read each and every file line by line. It will look for the mentioned patterns, that whatever information is mentioned is all these patterns. It will try to find out these information in these patterns in this manner, and later on it will print. It will print the output in the same order as mentioned in the print inside the print command. Since we want the information to be displayed in a file named information.txt, so we have created a information.txt file and we have written inside that file. And finally, towards the last, we have closed that file by saving the information written inside it. What information is to be written? Whatever information that is brought out after the execution of these many commands. That is, whatever was to be printed after this print command, instead of displaying the output on the terminal itself, it will now display the output. It will now write the output in a information.txt file and it will save it. So let's check this Python script in two ways. First, we will display the output in a text file and display the output in a file named information.txt, and later on we will display the output in terminal and see how it works. So let's check information file. So we can see that in the information.txt file, all the information is saved. So it is for the benefit of the user that whatever information you are getting, you can either display it or you can save it in a text file, and later on you can use it for whatever purposes you have performed the experiment. Again, we would use the same thing, but this time we would display the information on the terminal itself and then see how this works. So the difference is clear from the two sections, two parts of the command. If we want to create a file and write the output inside it, we can do that also. If we want to display the output on the terminal itself, we can do this also. Now let's look at all the chain information. Earlier, what we were doing, we were looking at the chains one by one, whether or not the chain is present in that PDB file or missing from that PDB file. We were looking at the chains one by one. That we started with chain A, then we moved on to chain B only, then we moved on to chain A and B simultaneously. But suppose there is a PDB file that it has more than one, more than two, maybe 5 or 10 or maybe 26 or maybe a number of chains in it. How to retrieve the data? Let's see whether this file can retrieve the chain information from all the PDB files or not. Very nice. We can see that the very first file has chain A, chain B, chain L and chain M. Whereas the second file, it has chain A and chain L. There are some files like this one, it has all the possible chains present up to X. So it is up to the user that you can retrieve the data one by one, chain wise, like you can write a single program for chain A, chain B, chain C up to chain Z, or you can combine all it into one and write only that information which is present for the particular file, and accordingly you may or may not display it on a terminal by writing out the calling a text file, writing inside the text file and saving the information. So in this way, it is a balance that the user has to maintain as per the requirement and the nature of the work. Now what we are going to do is today's script for all the possible information that can be retrieved so far that we have done in today's session, that is pH, experimental data, genetic information, residue information, system information, title information, chain information, everything I have incorporated inside a single PDB file named as allinfo.py. First of all, let's try to have a look at this file, and then we will try to execute it and then see how it works. So This is a very nice file. I would persuade all of the people, all of the participants to have a look at this file and practice the things for themselves also, and they can reach us for the doubts and anything they have, if they have any query or doubts and anything like that. So again, I would restart from the very beginning. Import sys means it has to interact the system with the kernel of the computer, that is user has to interact with the Python and the kernel. Import globe means it has to read a file of a particular extension. Here the mentioned extension is PDB. The user can, if the user wants to read another extension file, say Gaussian input file, so they can mention it in as GJF. If they want it to be as COM file, they can mention it as COM. If they want it to be log file, they can mention it to be log or CSV or MKV or JPG, whatever be the kind of information you can deal with, you can mention this. It is free to up to the user that what kind of file or what is the path or what a particular file you want to read through this globe module. Then we have opened a file named allinfo.txt. We have to write inside that file. This line means this globe module is looking for all those files that are ending with the extension PDB. Since this script has to extract chain information, so we have created variables from chain A till chain Z. Along with that, we have taken variables for pH information, resolution, experimental data, gene, and residue. And now we have tried to impose the conditions. What are the conditions? That if there is 0 to 6 is remark and 12 to 14 is pH, extract information present between line 45 and 48 and save it in variable named pH. If the information between 0 to 6 is remark and information between 11 to 22 is resolution, extract the information present between lines 26 and 30 and save it in, append it in variable named resolution RES. If there is a data named XP data in between lines 0 to 6, extract the information between 0 to 30 and store it in a variable named EXP. Likewise, we have done it for gene, for the residue information, and now look for chains. Else if command means if chain A is present, look for chain A. If chain B is present, look for chain B. If both are present, look for both. If none is present, look for none. So else if is a conditional clause statement that whatever be out of the 26 chains possible, whatever chain is present in whatever file, you just display it either on the terminal or store the data in a file, and accordingly if it is missing, just keep it blank. If it is present, show it that this information is present inside the file. So we can display again the same thing in two types. So first of all, let's see how this file works on the terminal itself. See, it is very confusing that so much information is being displayed inside a file. So what we would do at this time is we would open the file and we would now create another text file inside it named as allinfo.txt, and we would write whatever be the information that is present inside this file that has to be printed on the terminal will now be stored where? In the opened file. What is the file? Allinfo.txt. And later on towards the end, what we have done is we have put another command. This command will direct the open file to save with whatever information that is stored inside it. Let's now check the difference. So this time we have stored the information in terms of a text file, and that text file is later can be visualized by using any text editor like VI or Nano, or in case of Windows you can use the text file like Notepad or maybe Notepad++ or any whatever be the user's preference. Is there any doubt?
Okay, so if anybody have any question, they can interact. So nobody have any question? Okay.
Hello.
Yes.
Yeah.
Hello, my name is Vedin. I'm from Naipur, Guwahati. I have one question. Like, we are using Python to, we are writing Python file name and .py to run our program, but you are writing P3 for why?
Okay. Yeah. Actually, P3 is instead of writing Python3, I have created alias in my bash so as to make the process faster and easier.
How to create it? How to
It is,
create it, sir?
yeah.
How to create it?
No, so this will be explained. This will be explained to you later because now we don't have any time. This is break time. Thank you. Anybody else have any other question?
Thank you, sir.
Thank you.
Okay, so good Anvesh. I am happy to see that you have made a very good presentation and then explained very well.
Thank you so much, sir.
Thank you so much. It was really nice hearing you.
Thank you so much, sir. Thank you so much for giving me this opportunity, and sir, thank you for teaching me Python and all these basic scripts, sir. Thank you so much, sir.
No, thank you. Yeah. So anyway, so let us go for the break, and once we come back at 3:30, then there will be a talk by Dr. Madhvi Sastry, who is going to talk about effect of water restructuring mutations on ligand binding to human carbonic anhydrase. This is a very interesting title, and then I think all of you will be very happy to hear her. Let us meet at 3:30. So break time now. Yeah, Suman.
That is correct as well. This is just minimize the PPT.
Are we in the right position? This is the PPT. Are we in the right meeting?
Yes, yes, ma'am. Come on, sir.
What should I do?
Talk, we can hear you.
Hello? Hello? Can somebody hear me? Everybody is muted. Sir is also there.
This is the second one. Sir, Hemant sir?
Hello?
Yes, yes. Yes.
Can you hear? Yeah, great. So next.
Yeah, so we can hear you, and your video, if you can turn on.
Yeah, we'll just turn on.
Yeah, your presentation is also I can see, and your video also I can see. So everything is working fine. Thank you.
Okay. So I can just start. Let me know when. Is that one over? Hello?
In another 4 minutes, we will start, ma'am.
Okay, I don't know how it works, so I'm just asking.
Fine.
So it is, sorry, yeah. So how will we know that it starts?
Good afternoon, everyone. Welcome to the after tea session of MMTT 2021 International Workshop. So in this session, the first talk will be given by Dr. G. Madhvi Sastry, who has obtained her PhD in theoretical chemistry from School of Chemistry, University of Hyderabad. And then she did her postdoctoral research at the Fritz Haber Center for Molecular Dynamics in Israel. And she also worked with Professor Thomas Bali. She was involved in developing software to analyze results from quantum chemical calculations. She then carried out her independent career as a DST young scientist at Pondicherry University and later at IICT Hyderabad in the Center for Molecular Modeling, which was headed by Dr. G. N. Sastry. There she worked on a range of topics from homology modeling to artificial intelligence algorithm applied to proteins. Later on she worked with the GVK Biosciences, following which she joined the scientific team of Schrodinger at DE Shah India Software Limited. She is currently leading the scientific team and involved in research projects related to drug design, development and collaboration with industry and academia in US, India and whole Europe. She has published so many research articles, they are highly cited, and she has a lot of experience in coding and in various other research topics. So let's welcome Dr. Madhvi Sastry, and it's up to you now. Over to Dr. Sastry.
Hello everyone, thank you Hemant, and thank you for the invite for this conference. So at the outset, I thank the organizers, Dr. Hemant Srivastava and Professor S.N. Murthy and the entire organizing team to give me this opportunity to present some of the work that we have done at our firm. So, and a very good evening to everyone. So I'll be talking about some of the work that I have done in this. Okay, can you all see me? Is it okay? Is the audio okay? Hello?
Yes, it is. Yes. It is okay.
Yeah, okay, thank you. Yeah, so I'll be talking about a certain project that I was involved in a couple of years back. And then to begin with, I will start off with the theoretical part and how the whole algorithm works and why is it used and for what. So keeping that in mind, I'll just talk about the system first and what we are going to do in the whole of what I'm going to present in the whole of this session. So first of all, I'll be focusing on human carbonic anhydrase, which catalyzes the reversible hydration of carbon dioxide. And it is associated with osteoporosis and renal tubular acidosis. So defects in this enzyme lead to these type of diseases. And so we would like to understand the role of water in the binding process, analyze the water networks, and also quantify the water energetics. So in general, normally most of the time when we do analysis, we just look at the location of waters and the hydrogen bond interaction, so on and so forth. But in this presentation, I'm going to show you that how we can calculate the thermodynamic properties of each of the waters present in protein cavities and focus on carbonic anhydrase and a few other examples. So basically, why is water important? It's everywhere in biology. Protein sites are filled with water, and water competes with the ligand and substrate on binding. And this is basically the major point that we'll make use of in designing ligands using displacement of water or replacement of water or similar such analysis. So displacement of unhappy waters, when I say unhappy, it just means that they're unstable with respect to the bulk water. So the waters in the protein cavity which are unstable with respect to bulk. So since displacement strategies of those waters, and what is very important is just by looking at the structure, the energetics cannot be determined. You can't just find delta G values of whether the water is stable or unstable just by looking at the structure. So you need some theoretical method to calculate those. and there is no way other than a theoretical method as of now to do this work. So I'm talking about this algorithm which is implemented in WaterMap, which we call it as WaterMap. So here is the, I'm going to give an example of the pocket of thrombin. This is a protein binding site which has several water molecules. And the green water molecules that you see here in the pocket are those which are stable with respect to the bulk water. So it just means that these waters are happier to be in the protein pocket rather than in the bulk. And similarly, the red ones, we denote them as unhappy waters because basically they are unstable with respect to the bulk. So they would have been more stable had they been in the bulk, but they ended up in the cavity. And these are the waters which have high positive delta G values. So delta G is the Gibbs free energy expression, which is equal to delta H minus D delta S. So we will be calculating those, and I'll show you a few things related to that. Now, for example, this is the thrombin packet. How are these important? This is a nice example to illustrate how the waters play a major role in protein cavities. So this is the picture I showed you in the earlier slide, where are 3 stable waters and 1 unstable water. So now if you put a ligand here which displaces the stable waters, it's not a very good scenario because the waters are already happy to be in the pocket, but you're asking them to leave or forcing them to leave by putting a ligand in the pocket. So this ligand has a IC50 value of 10,000 nanomolar. Our ideal situation would be to displace the unhappy water, the water which is not wanting to be in the pocket and rather prefers to be located in the bulk. So you just modify the ligand a bit and you see a halogen group which is protruding towards this unhappy water, and then you see there's a very minor potency gain from 10,000 to 9,010 nanomolar. Then we further design the ligand in such a way that the ligand moves closer to the unhappy water, and again you see the gain in potency of 654 nanomolar. Then keep on modifying the ligand till it finally displaces this unhappy water, and you get a huge gain in potency of 3 nanomolar. So you're basically starting from 10,000 nanomolar and by minor modifications to the ligand, you have brought down the potency or increased the potency, brought down the IC50 values to 3 nanomolar. And this is a significant change. So this can happen provided the binding energetics are based on waters alone. We are considering that the rest all interactions with the protein are constant. Now, the delta G of binding of a ligand basically involves several processes. So initially we have this ligand which is solvated, and the protein, the apoprotein, which doesn't have the ligand in the cavity, also solvated. And after the solvated ligand gets into the bioactive conformation, the protein also gets into a ligand-induced conformation. Then the ligand gets desolvated. Similarly, the protein binding site gets desolvated, and the ligand gets into the protein pocket, and the protein ligand complex is formed, protein ligand water complex. And the delta G of binding is a sum of all of these individual delta G processes. And WaterMap is going to consider this part, the one which is in the red box, for delta G. So these results can predict the binding affinities accurately, provided this part of the whole thermodynamic cycle plays a major role in the binding thermodynamics. So the solvated protein in ligand-induced conformation. So in this situation, this is the part of the scoring that water map contributes to the binding of a ligand to the protein. Now what happens? Now for any hydration site within the binding cavity, there are 4 choices. One is when you are designing a ligand, you can displace a hydration site. So I showed you in the earlier thrombin pocket, the ligand is throwing out the water molecules, whether they're stable or unstable. So you can, the ligand can be in the same space as the hydration site, and as such the hydration site gets displaced, or the water gets displaced from the pocket. And such interactions are mostly hydrophobic. So this is one scenario. What happens when a ligand is in the pocket? The other scenario is replacing. So when we position a ligand atom in the same space as the hydration site or the water molecule, then if the ligand can mimic the functionality of the interactions of the water, then we call it as a replacement. So the water is having certain interactions, and when you design a ligand and the part of the ligand which occupies that hydration site mimics the same type of interactions. So we call that as replace. Interaction happens when we decorate a ligand or modify the ligand with a group that can favorably interact with the waters occupying the nearby hydration site. So these are basically the polar interactions. And there are certain times regions in the binding site which have very highly stable water molecules. And these are the regions which we have to avoid while designing our ligands in the pocket. So the thermodynamics of the individual hydration sites and the response to the changes in ligand structure are used to guide us between the options wherein we can fine-tune the ligands to come up with ligands with better potency. So it's worthwhile more in terms of lead optimization. To give an example of displacement, so a hydration site has both delta G and delta H. If they are both greater than 0 kcal, you know delta G is a summation of delta H minus T delta S. So let's say a delta G and delta H are both greater than 0 kcal. It is generally favorable to displace it with an atom from a ligand. So the water is already having a delta H of which is positive. That means there are no favorable interactions because delta H is positive, and delta H actually quantifies the interactions. So it's in such situation, rather than focusing on the interactions, all you need to do is just displace of that water. So all you need to do is, basically such waters are trapped in hydrophobic regions. So you need to design a ligand which displaces that water. So sometimes we see methyl groups or halogens. You see some magic methyl effects, which are because of this type of displacement strategies, having waters which have high positive delta H and delta G. Otherwise, these magic methyl effects cannot be explained theoretically. Replacement strategies here. So these are hydration sites where delta H of the water is less than 0 kcal. It just means that the water now being having negative delta H with respect to bulk. It can have negative delta H provided it forms interactions, some sort of interactions with the protein or with the ligand or whatever the case may be. So delta H is negative, very favorable, but delta G is greater than or equal to 0 kcal. In such situation, you can make a modification on the ligand to displace that particular water. Okay, so a suitably chosen polar functional group, if it is positioned carefully, can make some strong enthalpic interactions with the protein. So the ligand should have some polar group here which displaces this type of water, which can form an interaction with the protein. So you see this blue line, it's actually, you can see that it can form interactions with these nearby residues. So if you have a group like that, then a replacement strategy works. Similarly, we have the interaction strategy where some hydration sites are clearly conserved like this, and displacing or replacing them is very difficult. So these sites, and then every water molecule is clustered spatially. A spatial clustering happens, and the location of the highest water density is identified. And for each spatially distinct peak, we place a hydration site as a for which the delta G values are calculated. So the waters are spatially clustered, and then thermodynamic properties are calculated for each of the spatially distinct peak in the water density map. So this is the general algorithm, the way it works. Then we can go into details of that. So I said that a simulation of 2 nanoseconds is done in an explicit solvation. So that's what is written here. Waters are spatially clustered and analyzed. Entropy and enthalpy are computed for each hydration site. That each clustered waters are represented by a single hydration site for which the entropy and enthalpy are computed, which are relative to the bulk water. So these are calculated, and the entropy part is calculated based on the inhomogeneous solvation theory by Lazardis and Karplus, which is published in JFIS Chem in 1998. Delta E is taken from the force field from the non-bonded interactions, and the entropy is from a local expansion of the correlation functions. And I will explain that in the next part. So I told you when we are doing a molecular dynamics simulation, we are restraining the protein and doing the simulation just over the hydration water molecules. And so this is a majority of the system is heavily restrained, and only a few terminal hydroxyls are allowed to move during the simulation. So restraining the system prevents any rearrangement polluting the values calculated by water map. And it should be understood that while calculating the waters in the pocket, we are using an EPO structure. And an EPO structure can be a very high energy system, and it may tend to collapse if the structure is not restrained. So this is one of the reasons why most of the protein structure is restrained. I'll also talk about selectivity, which also helps in keeping the protein structure restrained, so which can sometimes give us intuition or intuitive design protocols or strategies for getting or designing selective ligands of homologous proteins. So clearly the restraints are needed to give a particular protein ligand complex water map meaning. So the problem, of course, is that even a slight change in the structure can really reorganize the waters heavily. So if a slight change in the structure, and one would end up seeing some hydration sites disappearing and some new ones appearing. So that's basically the complication. But the same complication can be used as a advantage too, which I'll talk about later. So because it is very sensitive to small changes in the structure, there's a question of transferability of the results across different ligands arises. So generally what we have found through our experiences is that the results are broadly in line with the regions of protein which are reasonably rigid. So wherein you don't see much changes in the structure. But whenever there is a mobile region, highly mobile region of the protein, especially in the binding cavity, near the binding cavity, we always recommend a fully dynamics-based approaches like FEP, the free energy perturbation protocols. So it is very important to understand the advantages, disadvantages, what can be tackled and what not of a particular algorithm before we apply it to any of the systems. Now, what do these delta H, T delta S and delta G correspond to? So, as I told you earlier, they correspond to the average excess enthalpy entropy and free energy that a water molecule located at a hydration site would possess relative to the bulk. So you have a bulk, and then you have this water, then the water enters into the cavity. So there are some water hydration sites into the cavity. And then when you displace the waters, they go out back into the bulk. So these yellow colored sites are those sites which enter the cavity. So this basically means that a hydration site with a negative delta H is making stronger interactions with the protein, and a hydration site with a positive delta H is making weaker interactions with the protein. So that's what one needs to keep in mind. And similarly, every interaction with the protein has a corresponding entropic penalty. So one thing should be noted down, that whenever a water molecule gets into a protein, it gets restricted. So there is an entropy loss, there is an entropy penalty by default for all hydration sites. So it is favorable to displace a water molecule from a hydration site whenever there's delta G greater than 0. But whenever delta G is less than 0 of that particular water molecule, it costs some energy to displace that. So the displacement is brought about by the ligand binding to the protein. And in this slide, we can see that delta G values calculated for APO water map correspond to ligand-induced protein desolvation energies. So this part of the whole delta G cycle, thermodynamic cycle, is basically taken care of. So why do we see hydration sites with delta G greater than 0 at all? I mean, why are water molecules even getting into the pocket and losing their free energy, or probably gaining in free energy? So one frequent point of confusion results in the presence of hydration sites with greater than 0 delta G. So many people feel that they should spontaneously leave the binding cavity. But actually they don't, simply because we are in condensed phase. And if the hydration site is vacated, we are left with a vacuum. And the penalty for a vacuum, for a single water-sized vacuum, is about 6 to 8 kcal per mole. So there is a 6 to 8 kcal per mole bias holding the delta G greater than 0 or the unhappy water molecules in place. So only when the environment becomes more unfavorable than 6 to 8 kcal per mole, we see a evacuation of the binding site. That is, the binding site in those regions gets devetted, or they are also called as the evacuated regions. Now, I was telling you we calculate the delta S and delta H is from force field, but the delta S is from the inhomogeneous solvation theory. So this is something which I was telling earlier, and this is how the equations look like. So basically, when you look at these equations, they are orientational distribution functions with the Cartesian and Euler angle coordinates. So the entropy is calculated from a local expansion of the spatial correlation functions. And basically the integrals, what they mean is they are just trying to calculate the excess, assuming that for a uniform distribution of waters, delta S being equal to 0, whatever is the deviation from the uniform water distribution is calculated by these terms. So the deviation from the uniform distribution and the orientational distribution functions mixed quaternion Euler angle methods are used for more easier mathematical simplicity. For example, a correlation function that we are talking about here. So divide the pocket into subvolumes of hydration sites. And then what is calculated is the probability of finding the center of a particle a given distance from the center of another particle. So let us say we are interested in one of the hydration sites here. If you look at the spheres, we look at how the water molecules are distributed around this particular water molecule. How are the other water molecules distributed? So, and then we get a distribution curve. So the pair correlation function, so first you have to pick, so each water molecule is picked up at a time, and then you pick a value of dr, the distance that you want to evaluate the density distribution. Then you loop over all the r's, that is what the integrals that you have seen in the earlier slide means, you're summing up all of them. So you count each particle you have in turn, and then count all particles that are at a distance between r and r plus dr from the particle that you are considering, from the center particle that you are taking care of. Then you divide your total count by n, the number of reference particles, reference water molecules that you are considering. Then divide this number by the volume of the spherical shell that you consider, and then divide this by the particle number density. So by this, when you do this, and then this ensures that the distribution, the correlation function with and is equal to 1. And in other words, if you just had an arbitrarily spaced spherical shell of inner radius r and outer radius of r plus dr, you will find a distribution, a rho star v particles inside, and where rho is the number density and v is the volume of the shell. So basically what you are doing is calculating the distribution of waters from a particular water molecule and looking at the deviation from a bulk distribution. After you calculate those, we calculate the delta G for each of the hydration sites using this function, where delta hydration site is delta E minus T delta S. So as I said, it is all hydration site is with respect to the bulk fluid. And then RCO in these equations is a distance at which a ligand atom begins to displace a hydration site. And then EHS in the equations is the system interaction energy. And SE, hydration site, is the exocentropy of hydration site. So these are the theta, these are the Heaviside step function. So the values are equal to 0 or 1. Whenever these are lesser than the cutoffs, those terms don't get added up. Whenever they are more than the cutoff, the terms get added up in calculating the delta G pi. So this particular methodology has been tried on several type of proteins and activity cliffs, wherein subtle changes lead to huge change in activity. Subtle structural changes lead to huge changes in the ligand activity have been studied. So these are just examples of some of them. I mean, in serine proteases, trying to pick kinase selectivity, work on GPCRs, falcipanes, PDZ domains, kinase binding, and many examples are out there in the literature. They are but a very few initial ones, which were attempting at a totally different type of studies that one can use WaterMap for, allowing our creativity to different domains, stretching the imaginations. So here is, for example, one example from Kung et al.'s paper, where they started off with compound 3, which had a 22 micromolar IC50, and then they came down to a compound which looks like this of 15 nanomolar. And if you look at the strategy, how WaterMap could have helped here is throughout the paper, they designed strategies, making modifications, and then finding out the experimental IC50 values. And WaterMap cleanly explains this whole process of how it could have assisted the design. So here there is a water molecule. There are two water molecules in the pocket. One is the water 1A and water 2A. And water 1A has a 3.57 delta G. You can see that enthalpy delta H is positive here in both the waters. Delta G of this water molecule, this red one, which is beyond this halogen, has a very positive delta G. So it's a very good candidate to displace. And similarly, this is another candidate, that water that can be displaced by modifying the ligand. So when you look at it, this is the complete observed SAR, this is the scaffold, and these are the groups. And you see that as you go down, when the OCH3 here at R3 in R2R1Cl, the compound 7 has I see a Ki of 0.14, whereas compound 6 has a Ki of 0.35. So what is happening here? So what is 7 and what is 6 doing? So this is where. So basically, 3 hydration sites, 3 clusters have been identified in the pocket. Water 1B, 2B, and 3B. 1B is about 4.7 kcal, this one. This is 0.33 kcal, and this is 4.42 kcal. So all the 3 hydration sites have very unfavorable entropies. And experience has shown that picking out one water molecule from such a chain is difficult because they can form a hydrogen bonded network amongst themselves. But what one could somehow design is removing all the 3 water molecules. Removing all of the hydration sites is one possibility which can lead to a huge gain in potency. So what they have come up with is they have switched a pyrimidine moiety here, which you see here, to a pyrrolopyrimidine here. It's a very intelligent strategy which displaces all the 3 water molecules. And one is replaced because the interactions are retained. And so through a series of simple modifications, they came up with a compound 17, which had a very low IC50 Ki values and high potency. So this is one of the examples that you see here. And these are the different substitutions on the scaffold and the R2, R1 groups and the corresponding Ki. And you see 17 is 0.015. This was what I was showing in the earlier slide too. Replaces, but 16 has 0.03 because it misses one of the water molecules, whereas 17 is 0.015, which displaces all the water molecules. Now, the method can also be used to see druggability. So if you have Gleevec, ABL kinase is a very highly druggable target, and if you look at the binding site, there's so many high energy waters. All these reds are high energy waters. So these actually provide a very useful indication of the shape of an ideal molecule, as well as it actually, by looking at the continuous water map, it shows an almost unbroken red region through the binding site. So this immediately tells you that this is a highly druggable site because it has so many high energy waters sitting in the pocket, which can be displaced to the bulk. Similarly, there's another one, PTP1B, and there are many stable water molecules here. So this naturally limits the drug likeliness of any ligand. And so the large core of binding site contains stable water molecules. So hydrophobic atoms are detrimental to binding in this particular region. So only powerfully ionic atoms are capable of replacing. So powerfully ionic, you again have problem with log P and other related parameters. So this naturally limits the drug likeliness of any ligand. So a druggable site can also be, you can analyze a site for its druggability based on these hydration sites. Then as I was telling, selectivity. So when I told you water map is very, very sensitive to slight changes in the structure. So when you have highly homologous proteins, which have similar structure but minor changes, and you want to design ligands which hit one of the proteins but not the other, selectively bind to certain but not to certain others, this becomes a very key method to actually apply. So there are many, however, subtle differences because water map is going to show the difference with subtle structural changes. So these small differences can make a surprising difference to water map of these proteins. And these differences can be exploited to give useful levels of selectivity. So using this technique, so sometimes we have been looking at some undruggable targets, which no methods were really working enough to design potent inhibitors. So this is, for example, so how could WaterMap help is, this is an example where for a target, it started off with 1.3 million molecules and then looked for ACC1, ACC2 potencies of 8,000 nanomolar compounds. And then from these 8,000 came up with new ideas, 5,000, and then synthesized 175 compounds, and which actually got molecules of potency less than 10 nanomolar. And from these again enumerated them and got 10,000 molecules, synthesized 225, and then got to less than 1 nanomolar molecule within a matter of 12 to 42, 16 to 42 months. And then these are the milestones achieved. Binding mode confirmation through X-ray, in vivo target engagement, in vivo efficacy in 16 months, and the safety profile, first human clinical trials in 42 months. And this, I think, is a very big jump from the traditional way of doing drug discovery. So wherever there is a chance that one cannot quantify empirically interactions between protein ligand or protein ligand water, a desolvation or WaterMap calculations can give a totally different scenario and intuitiveness to design ligands which can be more potent. So here I'm going to explain about some work that we did on human carbonic anhydrase. So far we have been talking about its applicability and the methodology by itself. So how we wanted to examine the effect of water rearrangement in one of the experimentally well-defined model system, that is human carbonic anhydrase. And then we had sufficient experimental group collaborations to actually see how the results are correlating with each other, and if not, why not, and trying to understand the whole process. So I'll talk about residue mutations and Hofmeister ions in particular in the protein. So before we start off any calculations, we need to prepare the protein that we download from the protein data bank and make sure that the hydrogens, etc., are properly added, and the tautomeric states of some of the protein residues and flips, hydroxyl flips, or charged residues are properly represented. Only if they are properly represented can we get results which are meaningful. And as I said, for WaterMap, it becomes more important because these type of interactions, changes can drastically affect the results. So preparing a system for a WaterMap calculation is of key importance. And I would say that we published how to do these sort of calculations and methodologies in this particular paper in 2013, which now has about 2,075 citations as of today. Now, the human carbonic anhydrase has many residues in its cavity, obviously, and using some sequence analysis techniques, methods and all, certain mutations were designed. And then ITC and other calculations were done. So there's a valine which was mutated to isoleucine and threone, and then there's a phenylalanine mutated to tryptophan and tyrosine. Similarly, L198A mutation was considered, and two ligands, the sulfonamide ligands, benzene sulfur, were considered as for binding when the protein residues were mutated, and looked at the effect on the binding thermodynamics. There are many other residues, also mutations that we studied. This was published in MMTT Chemie, the results, and I'm going to present them here. As a result, we deposited many crystal structures in the PDB, which I didn't list them here, but they are there. So we calculated, we did the same exercise here. And then we have the results from experimental data as well as from the water map calculations. The hollows, the empty spheres are results from water map calculations. And then what we see here is these are the results from the ITC as compared with the water map, and you see the trends look very similar all throughout, and which actually means the thermodynamic properties of ligand binding are majorly influenced by the hydration sites in the pocket, which can be obtained using the water map calculations. So these are the different mutants which were all for which we have tried doing the doing experimental studies as well as theoretical studies. So for example, if you look at the leucine 198A example, so if in the wild type, this is a leucine, and when you mutate it to alanine, the cleft obviously enlarges and the water network expands. And then what we notice is that when the HCA, the BTA, the ligand, sulfonamide ligand binds it, the waters undergo an enthalpically unfavorable rearrangement. That is, once the ligand binds, the waters are now enthalpically more unstable. So why is the ligand binding? If it results in enthalpically unstable waters, it just means that it's binding because the whole binding process is now driven by the entropy, the change in entropy, which is dominating the change in enthalpy. So even though there is an enthalpically unfavorable rearrangement, the entropy is taking precedence here and leading to the binding of the ligands. And we also did it with the other, between the two ligands, the TA and the BTA. BTA has this benzene group, so it's a larger ligand. So in the wild type, the two ligands look like this, and there is a hydration site here in the wild type with the TA ligand, but when BTA ligand is there, this hydration site is gone and the water network breaks. Now let's do a mutation of leucine to alanine and phenylalanine to tyrosine, and then we see a similar thing. So now you have a TA and a BTA, and you see this high energy water here, and when BTA binds, the benzyl ring forces an enthalpically unfavorable rearrangement of its constitutions. So there is an enthalpically unfavorable rearrangement. Again, even for this double mutant, the resultant is hydrophobic effect, which is entropically driven. So based on all of, I have presented only a couple of mutations here, but the paper has a whole lot of mutations, and a lot of analysis has been done to come up with strategies to enhance the affinity of the ligands. So the strategies are presented here. So enthalpically favorable rearrangements within the binding pocket could probably perhaps get us to ligands which can be more potent. So for the present ligands, we are finding due to binding there is an enthalpically unfavorable rearrangement. So if we can find out or look for mutations which lead to a favorable rearrangement, that's what is required. Mutations that enhance the enthalpic benefit of rearranging water will enhance binding affinity. And then while one might expect free energy enthalpy to be correlated, the nature of correlation in this system and our ability to attribute it primarily to the influence of mutation on local water networks is unique. So what basically we are doing is we are looking at the influence of mutations on the water networks and coming up with strategies of designing ligands. Mutations that decrease the hydrophilicity of the amino acids, especially near the non-polar surface, are likely to increase its affinity for ligands by reducing the enthalpic cost. So this is as far as the residue mutations in the pocket are considered. We also studied the effect of Hofmeister ions. The Hofmeister ions in the pocket are two types, cosmotropes and chaotropes. And the cosmotropes tend to stabilize the folded proteins and cause proteins to precipitate in aqueous solution. Strongly hydrated, and they're very small ions. The larger ions are chaotropes, which can promote denaturation and enhance the solubility of proteins in solution. They are weakly hydrated and large. So by hydrated, we mean free energies of hydrations, not the water map type of hydration sites. So earlier studies on ions in bulk water suggested that the effect of the ions on water structure is limited to the first hydration shell. And here we have found that it actually extends far beyond that. And again, as I said, one can use these results and really enhance or take the creativity and intuition to different levels. So we did MD simulations, which suggested an unanticipated effect of Hoffmeister ions on the structure of water with concavities on the protein surface. So these anions, upon associating with zinc, trigger rearrangements of water beyond the first hydration cell. So it's going much larger, something like 8 angstroms or further. This amplification is consistent with the notion that water within concavities on proteins suggest long-range structure, and these rearrangements influence the ligand binding. So, and ion-induced structural perturbations are likely to be sensitive to local environment influences like the electrostatics topography in the protein cavity. So we studied the effect of these Hoffmeister ions, which again trigger rearrangements of water, and these rearrangements, which was earlier thought to be the ionic effect to be only to the first hydration shell, we found that they actually go much beyond that. So if you look at iodide here, it actually replaces these two enthalpically and entropically unstable waters. And then you see the first hydration shell and the longer hydration shells which get affected. Again, we tried to calculate the delta G values for different anions, and the distance you see around 8 angstroms. The effect of these anions on the water network is seen up to 8 angstroms. So SE, an example, triggers rearrangements of water that extend up to 8 angstroms from its surface, and this was the details are published here. And then later on, we studied also ligand modifications. I said that these can be used very well to understand how the ligands can be modified with the, using the water networks and the thermodynamics of water, and if the mutations can help. So this was in process, still undergoing. And a few PDB crystal structures have been deposited and many more submitted to PDB with the mutant structures as well. And we did a very preliminary FEP calculation, very preliminary I should say, with wherein we could separate out N alkyls and C alkyls could be separated in terms of their free energy of binding. So delta G bound minus unbound comparison between experimental and predicted values are shown here. And the values are taken at 9 angstroms because we know at that point the effect of the ions, if there are ions, would be more or less stabilized or uniform. And the predicted delta G values are well able to distinguish between N alkyls, C alkyls. And remember that all these ligands, the range of delta G is 1.24 kcal per mole. And within that 1.24, if you are able to distinguish C alkyls from N alkyls, that's a very big result. So I would say that the FEP calculations were able to literally pick out and cluster these two group of ligands that we calculated. So in conclusion, I should say there is an important asymmetry in our system, and all of these mutations we noticed tend to make delta H less favorable and T delta is more favorable. And a reversed form of HS compensation could plausibly result from mutations that weaken or shrink networks of water in the APO binding pocket. Then many mutations most likely to cause an effect. So in this particular, since we took human carbonic anhydrase as a model system, The two mutations which are most likely to cause such an effect are these. We identified the mutations are this asparagine to leucine and phenylalanine to tryptophan. And these could weaken the networks of water solvating it. And by excluding molecules of waters from declivities in the nonpolar wall could reduce the size of the water network, leading to entropy-driven binding thermodynamics. And then ion-induced structural perturbations are likely to be sensitive to local environment influences. And as I noted earlier, that the influences go beyond the first hydration shell. I think that's where I stop my presentation and leave the forum open to questions. And I thank the organizers and my colleagues at Schrödinger, my collaborators, investors, and everybody who is associated with this work, and also with my team. Thank you. Namaste.
Thank you, ma'am, for such a detailed and informative talk. It was really very interesting. We could learn a lot of things. And now I want all the participants, whoever want, they can ask questions. So the session is open for question and answer. So before asking, you have to unmute. It's very low voice.
I see one question in the chat.
Yeah, please, you can answer that.
Yeah, by Omkash Tanwar. So the question states that how can we identify the most important waters from the PDB structure? Is it wise to delete all the waters during protein preparation? Okay, so there are it's a very tricky question, and there is no general rule that you can say you should remove all the water molecules from the PDB structures. So one way, what I would do is, if there are similar protein, there are many structures of the same protein, one way would be to identify conserved water molecules. The other way, if and see and try to retain some of the water molecules. Another way of doing this is you could obviously run water map, which also predicts the hydration sites and calculates the thermodynamics, and then decide on whether you want to retain the water molecule or not. That can be another. And there is one thing that people have to be very careful about is that in most of the drug design strategies, if somebody if somebody finds a bridged water molecule between the ligand and protein, they think that particular water molecule would play a major role in the interactions, but it's not necessary. So when a water molecule is bridged between a protein and ligand, how many hydrogen bonds, one should realize that there's a huge cost, entropic cost, at the water molecules and assess whether they are important to be retained or not all the time. So the thermodynamics need to be assessed, number one. And number two, we need to be very careful of the entropic component of delta G, which most of the time we ignore. And that can play a major factor.
Hello.
So, Yeah, I don't know if this has answered your question, but if not, you can please ask me more. Clarify. You can ask for clarification.
Hello.
Yeah, please.
Yeah. Hi, I'm Suyash from Naipar, Kolkata, and my question is how the binding free energy computed by water map is different from binding free energy computed for protein ligand complex by MMGBSA. Is there any?
The first major difference is MMGBSA is using an implicit solvation, and it uses an empirical way of calculating the electrostatics and the cavity, especially the cavity terms. The electrostatics are done by either Poisson-Boltzmann equation and all, and it doesn't really take into account the water explicitly. And whereas water map is trying to take water, it takes water explicitly. So let's say, how do you identify which water is forming a hydrogen bonded network or hydrogen bonds with the ligand or the protein through MMGBSA? Because it uses implicit solvation. So the directionality of hydrogen bonds are so important. The which water is forming hydrogen bond, it's important. All this MMGBSA doesn't account for. MMGBSA, on the other hand, tries to account for the interactions in an empirical way, protein ligand interactions and all, which actually water map doesn't directly involve, it doesn't calculate. As I showed you the whole thermodynamic cycle, water map takes into account one part of the thermodynamic cycle of the ligand binding, and MMGBSA is the complementary term. It doesn't take into account the desolvation terms the way water map does, but it definitely takes into account the interactions in a very empirical way. So they are very different methods. Actually, I would say both are complementary to each other.
Thank you, ma'am.
So the next question is how one can track down the
mutation. We also have a way to combine both of them, MMGBSA and water map scores, so that you get an overall binding and free energy estimates. Yeah. Thank you.
So the next question is how one can track down the mutation. This is asked by Vishal Yadav.
How can one track down the mutation? In what sense? To identify the mutation? Or
Vishal, can you write answer?
Can you be clear? What is the exact? In what way do you want to track down?
Okay, so then the next question is that in heme enzymes, namely like P450s, water usually easily get displaced with the entry of substrate. Does that mean that it must be having high delta G value?
Possible, very. It's a very good possibility. Some of the waters can have high delta G, or even if the delta G is slightly zero or something, if the ligand coming in brings in more worth in terms of interactions or anything, then it can still throw out. But yes, that's a very high possibility. I would. I haven't done a water map on any of the heme proteins, though I have worked on heme proteins a lot earlier, but that's a very high possibility. One can check, for example, bromodomains. They have these water networks at the base of the pocket, and they are highly unstable. When we ran a water map calculation on them and see, I mean, they seem to drive the binding affinity a lot. These the specific waters in bromodomains, the bottom of the cavity, and in all cases when we tried, we saw that these waters, hydration sites, end up to be having delta G as positive with respect to the bulk.
Okay. Anyone have any other question?
Okay, so I'll answer that question. How one can track down the mutation, to identify the mutations. These can be done very easily using bioinformatics tools like sequence analysis and a structure analysis. So you look for conserved residues and unconserved residues, and residues which are conserved in most of the homologous proteins, but in some they are unconserved, and then see what happens if you mutate. So you can get a very intuitive idea of what residues to mutate using these tools. and they're very fast. And also the structure. Sometimes you need to look at the structure also to see, though the mutations sequence analysis predicts that possible one can zero in with some mutations, there can be some residues in the structures which are probably not near the binding site or probably closing a channel or something. Yeah, could be. There are many scenarios. Yeah.
Okay. Anybody have any other question? If not, then let's thank Dr. Madhavi Sastry for a very informative talk. Thank you so much. And now we will move to the next session. Thank you, ma'am, for your time.
Thank you, Hemant, and thanks for organizing this seminar. Thank you very much. Bye-bye.
Yeah, thank you. Thank you. So next session is hands-on session on Schrodinger, which will be given by Dr. Sudarshan Pandian. Dr. Pandian, you are online?
Yes, Dr. Hemant, I'm here.
Okay, you can turn on your video.
Well, I'm going to share my screen.
Before you share, I want to have interaction, because if you share the screen, the video will not come properly. So
okay, so
you turn on your video and then we will interact for a minute. Then
yeah, sure.
Yeah.
I hope you can see me.
You can unshare your presentation.
Well, okay. Yeah. Yes, sir.
So good evening, Dr. Sudarshan. So thank you for joining. Dr. Sudarshan Pandian obtained his PhD from University of Sauve, France in the year 2009. He had 6 years of postdoctoral experience in various universities in France, Germany, Belgium, and Luxembourg. He has 15 years of experience in using molecular dynamic simulation for various industrial applications. He is mainly involved in providing scientific support and training to the customer for the simulation of software from various industries, government research centers, and academic institutions in India, Middle East, and Southeast Asia. His recent research interest is developing sophisticated molecular models for pharmaceutical formulations and understanding their behavior at the molecular level. He joined Schrodinger in the year 2016, where he presently works as a senior scientist. He is going to take a demo. Let's welcome him. Thank you. Now
Thank you. Yeah. So I'm going to share my screen. So I believe so all the registered participants for the workshop, 24 of them, got their login details from my colleague Kishore. So if you haven't received the email, please raise your hand now so we can fix it now. So we have shared it this morning. So in case if you haven't received your login details for the cloud-based online workshop, please do let us know. So we will be.
This workshop was started early in the morning. So since it was shared in the morning, probably participants did not get chance to download and install.
Well, it was they don't need to download anything. They don't need to install anything. Just they have received a username and password. So now I'm going to give them a link for correct using that username and password. So I'm going to put it in the chat window. So you can look at it in the chat window here. So I have shared a link. Using that link with a username and password shared with you this morning, you can log into your virtual machine. So these are cloud-based machines. It's located in Google's cloud platform. So we are going to use Schrodinger software in Google's cloud platform. So please kindly check the link in the chat window. Click on the link, and the username and password has already been shared with you in an email this morning. In case if you haven't received the email, please do let us know. We can provide you a username and the email address, username and password in the chat window here. I believe there were 24 participants, and to all of them we have shared the details.
So participants are saying that they have not received it. So can you share this in the chat window now so that they can get
Sure.
this user ID and password?
I'll get it done. Yeah.
So if you see the chat window,
There's a link to connect.
It's asking for a username and password.
Yeah.
You send this username and password to my mail. I will forward immediately to the 24 selected participants.
Yeah. I'm sending it to you. Right now I'm sending it to you. So it shouldn't take long. I just have to find it.
So all of you should check your mail in a minute for user ID and password so that you can go directly to Google platform and then work with Dr. Pandian. So
yeah,
when he will share, I will forward.
Subject line read cloud login details. So no, we have shared it this morning at 10:30. So in case if you haven't received it, I'm going to share it in a minute. Just give me a minute. I'm going to share it in a minute. But if you have received it using the link which I shared in the chat window, click on the link. It would be asking you username and password. Just enter the username and password which we shared with you. In case we haven't received it, give me a minute. I'll share it with you in a minute. Dr. Hemant, I just shared a PDF with you, so which has all the login details for all the participants, and also we sent an email to everyone again. So we have already sent an email to everyone. One more time, and also I have shared it with you.
So till now I did not get the mail.
Okay, I just sent it to you. So probably give it a minute. So there's a PDF, login details.pdf. I shared it. So with you, So there are 24 usernames and passwords, and we have shared the same in the same order. Basically, the same email order which you have given us. So the same order in which we have sent it
so it is different user ID and different password for each one of them?
Yep, it will be user 1, user 2, user 3, like that. So up to 24 users, they will be getting the 24 username and passwords.
Okay, fine.
Yeah, we have shared an email already with them also, second email.
I got your mail.
Yeah, so we shared login to all of them also. Okay, sure. How many of them connected already?
So I forwarded to all of you. Please check your email for the credentials and log in.
So yeah, we also shared an email with them with the individual username and password for them. And so far nobody logged in.
Okay, I'm just calling.
So you can pick up one of the username and password. Also, you can try logging in using your computer.
So I'm using a Mac system. It will work right there.
That doesn't matter. Any system, you just click on the link which I gave in the chat box and use the username and password given in the PDF document. You'll be able to log in. Sorry, come again. So you should get to this page. So just copy that link and paste it in the browser, web browser. So you should be able to get into this page, storing your virtual workstation. So in here, you can copy paste your username and password. So I'm using username 25. So here is my user 25. And the password. So user and password. Just once you are in this machine, in this page, you have to enter your username and password. Just give login. You should be able to connect to the machine.
I think a few of them already logged in.
Yep. So let's give them a minute or two. So let all of them log in.
If any of you having any difficulty in logging in, please let us know so that we can start it. And whoever is not able to log in, they should see the presentation and try it later on. So we will continue with the session within 3 minutes. So 3 more minutes to log in.
I believe there are 7 people who already logged in. So in case if you logged into this page, click on virtual workstation. So click on the link called virtual workstation. So it will prepare a virtual workstation for you. Okay, so click on the virtual workstation. It would open a virtual workstation for you. So this is a cloud-based computer which is located in Google's cloud platform, and we are going to use this computer through this, browser window. So once you click on that one, it would take a moment. It would prepare your interface there. Okay, so in the interface, you will have Linux environment in the background, and you will have Studio software installed already ready for you. So from there, we can launch the Studio software. We'll be doing the demo of QMMM.
So, sorry, sir, can you turn on your video?
Oh, haven't I? I'm sharing my screen actually. You don't see my screen?
We can see your screen, but your video is not working.
Oh, okay, sorry. So that's for the bandwidth. I switched it off.
Now it is coming, sir. Thank you.
Yep. So just for the sake of bandwidth, I switched off the video because I wanted to make sure that we are able to get workstation properly going.
Okay.
Okay. So we have about 12 people connected to the machine. If others having trouble, please do drop a message or drop a chat message. I hope Om Prakash.
No issue. I think we can continue, and this video is available, so they can do later also. So now we will continue.
So video is going to be recorded, and you can do it later also, not a problem. So once you are in the interface, so there is a materials icon. So there is a material science icon which is at the left hand bottom side in the virtual workstation. So you can double click on that virtual material science workshop icon. So it would open up the material science interface. So there is a material science icon on the left hand bottom corner or close to the left hand bottom corner in the virtual workstation. You can click on that one. It would open Schrodinger's material science suite interface. Okay, Schrodinger's Master Material Science interface, which would look like this. Okay, just click on that. Yeah.
Sir, this is Ronita. Sir, my virtual workstation is still showing that please be patient, your desktop is.
Yeah, if your internet connection is slow, it's going to take some time.
Okay, yeah.
So,
okay, now it came.
It came, so it depends upon the internet connection. So you have to have minimum of 5 Mbps speed to get it connected, which you normally get it in the mobile phones nowadays. Okay, so here is the materials icon. Double click on that materials icon. It would open the materials interface. Okay, shouldn't take long. So we are going to do things inside the materials interface. So today we are going to do QMM calculations. So QMM stands for quantum mechanics and molecular mechanics. So when we have a drug molecule or a ligand molecule inside the binding pocket, so sometimes we have to do treat them with the QMM approach. So where we have to do this QMM approach and why do we need QMM approach? That's what we are going to discuss first before we start on the actual tutorial. Okay, so when we do the docking, we use force field based approach. When we do the force field based approach, so we rely on force field parameters to be right and then give us the binding energy of the ligand in the binding pocket. But when there are metal atoms around the binding pocket, when there are situations where the binding pocket is kind of relatively flexible, and then there are a lot of things which are happening, or as Dr. Madhavi Shastri was showing, there were potential bridging interactions between water molecules, or there are potential water molecules which are easily coming in, going out. So in that kind of situations, we really want to make sure whatever the value which you are predicting in the binding pocket is going to be the right value. So in those situations, what we can do is we can use this QMM approach, where the ligand is going to be treated, or ligand plus the local environment around the ligand is going to be treated with a quantum mechanical approach, and the rest of the molecule in the background are going to be treated with the molecular mechanics approach. So with this approach, we are making sure that using quantum mechanical calculations, we are calculating the binding energy in a more accurate way. Okay, so that's exactly the point of doing QMM calculations. In the second situation scenario, where there is a chemical reaction that is happening, that means there is a covalent binding that is happening in the binding pocket, or there is a covalent reaction that is happening between the ligand and the one of the residue, amino acid residue in the protein backbone. So we want to understand how the reaction happens, what is the transition state energy looks like, how much of energy is required to make the reaction go in the right direction. So to understand that one, we can again use QMM approach to calculate these energies. So these are the two situations where we can use QMM approach. In Schrodinger, we have QSight, a tool called QSight, which is used for this kind of approach, QMM approach. Okay, so as I said, that QMM has both QM as well as MM. So we need two different engines which are going to treat the molecules in two different ways. So QM engine here is going to be Jaguar. Jaguar is our QM engine, which is kind of very well documented DFT code or a QM code. So this engine runs much faster than any of the conventional codes which are available in the market. And then for the MM part, we would be using macro model. So the macro model would be giving us the background energy or background calculation, energy calculations. It should be doing the rest of the molecule, whichever is not in the QM region. So these are going to be treated in the MM approach. So these MM molecules are going to be, or MM residues are going to be treated using macro model, and their energies are going to be calculated from the macro model. So using OP-less force field. So once we have them combined together, we are going to get the entire complex's energy. So to do that one, so now we are in the interface. So I hope you are all able to connect to the interface. You are all able to open the Nestro interface, Nestro Material Science interface. You are in the interface now. In case if you are not able to do this one, don't worry about it. The video recording is going to be available to you. The server is going to be open to you till the end of the day today. Okay, so it will be closed by 9 o'clock this evening. So the
hello, sir.
Yes.
Sorry for the interruption, sir. Can you go little bit slow?
Okay, I'll go slowly now. Okay, sorry for that.
Sir.
Okay, so yeah, so if you're not able to follow, please stop me in between. Okay, we'll discuss that point and we'll go slowly, as slow as possible. Okay, so yeah, so I hope you are all able to open this interface. You are in the interface now. In case if you are not able to open it, don't worry about it. This GCP Google Cloud platform is going to be open till 9 o'clock this evening. Okay, so this video recording is also going to be available to you, and whatever we are going to do here is already available for you within the interface itself. Okay, so you can do it on your own. Just listen to me, follow the instructions here.
Dr. Pankaj, I have a request.
Yeah.
So please allow the access till tomorrow, so that whoever participant is missing, they can do that.
Well, they can come back to us, Dr. Hemant. We can give it, because for the GCP we really have to pay money for it. Actually, for every hour it's going to cost us. So that's why we have it for the day today.
Okay.
If they want to really learn more in that front, they can always come back to us. They can write to help@studingo.com. And we will be able to help them, providing an evaluation license or providing a local license to their machine. They can practice these things also, not a problem. But today, this
whoever is not able to do by 9 p.m. today, they should contact Schrodinger. Okay, but yeah, I request all the participants. This is a very good opportunity to learn QMMM, which is a very useful calculation. So please do your best. Do your best and learn whatever is possible.
Yeah, if you are not able to do it by yourself, if you can listen to me and follow these instructions, you can do it later also, not a problem. Okay, as I said, this next 1.5 hours or 1 hour, we are going to give you the step-by-step instructions which are already pre-installed in the computer, in the GCP. So you can do it later also. So if you go to help, tutorials, so the help is there right in the top. You can just follow my instructions here. Later you can go into your tab and do it. Okay, so if you go to the help and tutorials, in the help and tutorials, there are show tutorials by product. Then the product, there is all. If you drop down on the product, you'll be able to get QSight. Click on QSight. Click on QSight. So it has 2 tutorials, and we are going to use the second tutorial, QSight tutorial. Okay, just click on the QSight tutorial. Click on the view button in the left-hand corner. Okay, so in the left-hand corner, you have copy button. Next to copy button, there is a view button. Just click on the view button. It would open up in the web browser. Okay, it would open up in the Mozilla Firefox web browser, a stepwise guide to run QMMM calculation. Okay, QSight quick start guide. That's what you would be getting, a page like this. Okay, in the QSight quick start guide, click on the link QSight quick start guide. It would open the first page of the tutorial. Okay, so the first page of the tutorial, you have contents of the different stages of QMMM calculations. Okay, please let me know if you are not able to follow me, if you are not able to reach this place. I'll be redoing this one.
Please redo this once. I have not reached that page yet.
Yeah, sure. I'm going to redo this one. So I'm going to close this one. Okay, so let me go back to master interface.
And people are having a bit slow internet. So if you can go a bit slow, it will be useful for them.
Yeah, so I'll do that. Yeah, so if you go to help tutorials, so help tutorials. So once you are in help tutorials, in the product drop down, you should be able to see Q site. Take your time, do it slowly, not a problem. Okay, in the product there is Q site. Just open the Q site. You would be having two different tutorials. Take the second tutorial, click on the second tutorial, it would be highlighted with a yellow line. So once you have the second tutorial selected, there is a view button at the left hand corner next to the copy button. So we will be doing the copy button also, but that's later part. First, let's click on the view button, which would open a page like this. In that page, there is a quick Q site quick start guide link. Click on that one. You have step-wise procedure which is going to be available in this quick start guide. Take your time, do it slowly. If your internet is going to be slow, then it's going to be taking some time, but patiently do it. If you are not able to do it, don't worry about it. Let's wait and do it slowly. Just follow the instructions. I hope all of you are able to reach this page.
Yes, sir.
So some of us should interact so that we understand whether you are fully following or not.
Yeah.
Yes, sir, we have reached.
Yes, sir, we have reached the page.
Very good. Thank you. So now let's go to the first link, copying the files for the exercise. Okay, so we are going to copy the files for the exercise. Okay, so what does it say? The step one, again, if you go to help and tutorials, there's a copy button, right? If you go to help and tutorials, there's a copy button before the view button. So if you click on the copy button, it's going to copy the files to a location. So you can see copy to. So this is a location in the
Hello, sir.
Yes, ma'am.
So, how to go back to this section where we can?
So just click on the, okay. So in the bottom you have this material science icon. Click on the material science icon. It would come back to material science automatically. So in the bottom you have master material science icon. If you click on that one, it will come back. So in that one, just go back again, help and tutorials. So we go on to help and tutorials. Okay, so you can do a copy. Tutorials. Okay, so you can see by default it's going to copy it to the temporary directory. Okay, in case if you want to take this data, you can take this data also. So for that one, you can browse and put it in a location where you have access. Probably put it in the desktop. Okay, create a new directory, call it QMMM. Okay, just choose it and copy the files to that particular directory. Okay, you can copy it in any directory. Okay, but here we are copying it to, I'm copying it to QMMM directory. If you by default copy it, it would go to the temporary directory. I hope you are all able to copying it. If you're able to copy it, just let me know.
So can you tell me once more that how to check if it's copied or not?
Yeah, so to check, you can go to the directory. So again, if you click on the bottom left hand corner, there is a button. Okay, click on the button, it would show the default desktop. Okay, so once you are in the desktop, so you can browse through your home, or see, for me, I copied it to the QMMM directory in the desktop. You can see I got two files copied here, one tpb.mae or qsite-1tpp.01.mae. This is the input file, that's the output file in case, we are going to visualize the output file from here. So that's why we have input file, we have the output file. Both the files, two files are copied into that particular directory. Okay. So I put it in the QMMM directory. You can put it in the keep the directory in your name or some other name, but put it in a location where you have access. I kept it in the desktop. Okay, copied it. Got it?
Yes, sir.
Yes, sir.
I hope all of you are able to copy it also.
Yes, sir.
I take that as a yes for everybody. So we copied it now. Okay. So now we are going to create a working directory. So where we are going to be setting up the calculations. Okay. In Schrodinger, what happens is you can close down the tutorial panel now. So Schrodinger, what happens is by default, everything is going to go into a temporary directory unless we change the directory path and specifically show that it is in this directory are going to run all my calculations or all my simulations or all my preparation. So otherwise it's going to go into a temporary directory. So in order to do that one, we are going to change the working directory. So how do we do that one? File, change working directory. File, change working directory. I'm going to go to the desktop and select my QMMM directory as my working directory. So I got selected the QMMM directory, just click the choose button. So now I changed my working directory to QMMM directory. Please do it slowly. Once you've done it, just tell me that, yeah, we have done that one.
Done, sir.
Thank you, ma. Okay, next we are going to save this project. File, save project as. Okay, file, save project as. Let's call it again QMMM. So I saved it as a QMMM project. So you can see in the top QMMM.prj. So it's saved it as file, save project as, and then save it. It's going to automatically save it inside that working directory. So we have saved that project now. I hope you are also able to do it.
Yes, sir.
So now we are going to import the file, input file, file, import structures, file, import structures. See 1tpb.mae, open it.
Hello, sir.
Yes, ma'am.
Sir, actually I can't save the project. Like how to do? Can you tell once?
So file, save project as. Okay. So inside the directory where you copied the files, you can save it as. See, I saved it as qmmm.prj file. So just save it. File, save project as. So if you are trying to save it in the desktop itself, it won't work. Put it inside the folder. So this one TPB file. So that's what we are going to import. So once you save the project, you can import the file. File, import structures. In the import structures, okay, browse for this one TPB file .mae. Import structures. 1tpb.mae. Okay, so this 1tpb.mae is a pre-prepared structure for us. As you know, the PDB structures which we are going to take from the PDB database would not have the hydrogens. Okay, so it might even miss some of the loops. It might miss some of the terminal residues and stuff like that. But here we have a pre-prepared PDB structure. In case if you are working on your own structure, you have to use protein preparation wizard to prepare your protein. Okay, so which is not going to be done today because that's not in the context of today's workshop. But if you are interested in those things, learning those things, you can always reach out to Schrodinger. Schrodinger conducts frequent workshops on how to do docking, how to do the protein preparation, all those things. We can train you on that one. Okay. Even for NIPERS, we have done it many a times. If you are new student, if you haven't heard of Schrodinger, if you haven't done any Schrodinger workshop, you can learn those things from our regular workshops. Okay, how to do the protein preparation. But today, that's not the context. So we are going to do a QMMM calculation. For that, we have taken a pre-prepared protein structure. Okay, so once you select this 1TPB.MAE, this is a pre-prepared protein structure for us. Okay, so I just opened it. When I open it, I have that structure imported into my workspace. This block space is called the workspace. Okay, I hope you are all able to do it. Yes. Are you here with me?
Yes, sir.
Are
yes,
you at
sir.
this level?
Yes.
Okay.
Yes, sir.
Good. Thank you. So in case your computer is slow, if your internet connection is slow, do it slowly. Not a problem at all. And that the tutorial which we opened has all the stepwise information. If you go back to the tutorial, it has all the stepwise information. You can see change the working directory, saving the project, importing the 1TPB complex. All those informations are given in a stepwise way. Okay, in a stepwise procedure, it is given. Yes, ma.
Sir, I'm using the user ID 31. But in my directory there is no PDB file there is no PDB file, so I downloaded that one. Then after that you said it's the pre-processed protein.
Yep.
So what should I do now?
Okay, so if you have downloaded by yourself, okay, so what you have to do is, if you click on, again, go back and click on tutorials, help tutorials. Okay.
Yes.
Select the Q site. Okay, in the Q site second tutorial, just click on the copy. So once you click on the copy, it would automatically copy those two files into your directory. But please make sure you're copying it to the right directory. Okay?
Yes, sir.
So if it is not the right directory, please browse and change it to the right directory.
Oh, yes, sir.
Okay, so once you copy it, then the files are going to be available there. You can import the input file. So once you import the input file into the interface, just press the L key in your keyboard. No, just press the L key in your keyboard. It will take you to the ligand. So there is a ligand in here. Wow, it's because of the internet connection for me also. It's taking time to, take it to the ligand.
Sir, does it come as a blue highlighted part?
Yes, the blue highlighted part. Yeah, it's the turquoise blue is the one which is. Yeah.
Okay.
I'm going to
excuse me, sir.
Yes, ma. Yes, pa.
So is it possible by any chance that one user ID is being used by more than one person?
Well, we have given, if you use the one which came to your email, then it is every user has a separate user ID. So in case if you feel that your user ID is used by somebody else, what is your user ID?
User 2.
User 2. Is anybody else is using user 2?
Because I see things happening by itself, so I think somebody else is also using.
Okay, so then in that case, close that session, go and open a new session. You can open a new session. You have user ID up to 30 in the PDF document which we shared with you.
Okay, okay.
So you can open a new session. For you, there is going to be 4 more hours to do these things, okay?
Okay, thank you.
So 1 TPB, once you press L, so you have ATP molecule in your interface, right? So you can see, so that's a ligand molecule. So this ligand molecule, we are going to set it up in the QM region, and the rest of the protein, we are going to set it up in the MM region. So how do we do that one? That's what we are going to see now, okay? So all the information, step-wise information is already available for you in the tutorial. In case if you are not able to follow here, you can read what is there in the tutorial, follow it step by step also, not a problem. Okay, so that's why we open the tutorial also. So in case if you are not able to do it with the instructions followed here on live, you can follow the instructions in the tutorial do it step by step by yourself. Okay, and if any doubts, just stop me. We will discuss on that point. Okay, so here is a ligand. Okay, so this ligand we are going to run it on QM mode, and the rest of the protein is going to be in MM part. Okay, so once we press the L button, we are going to see the ligand in the center of our workspace. Do you see that?
Sir, how are you zooming in on the structure?
Yeah, so let's go back out. Just press the L in your keyboard. Okay, it automatically zooms into ligand. So anything that is not amino acid residue, it considers that as a ligand. So it zooms into ligand. So in this case, it is ATP molecule.
Sir, when I'm scrolling up with my mouse, I think some additional parts are coming in the protein, and when I'm zooming, like scrolling down, it is going away.
Yeah, it is because we are zooming into that particular binding packet. We are zooming out of that particular packet. So the background is, so in 3D, they are coming closer or going away from one another. When you scroll down, scroll up, it means you are zooming in or zooming out, right? That means you are getting closer to this binding packet or going away from that binding packet. When you look deeper and deeper, so the residues which are further away, they are going to be visible at some point. When you zoom it in closer, they are going to disappear in the background. So that's why they are disappearing and coming back. Okay, it's just a zooming in effect. Okay. Yeah, so here's the ligand. So now we are going to open the panel called Q site. Okay, it's there in the right hand top corner. Click on the tasks. Okay, browse all. Tasks, browse all. Okay, in the application section, there is a last option, other applications. Click on other applications. In the other applications, in the first column, 6th row, Q site. Okay, it's there under browse all, applications, last option, other applications, first column, 6th row, Q site. So click on Q site. It should pop up a window like this.
Sir, can you please repeat this?
Yeah, I'm going to repeat it now. So tasks, right hand top corner. Okay, you have materials, and below the materials you have browse all. Click on the browse all. You have all the programs which are listed on the left hand side. Right at the bottom, there is an option called other applications. Click on other applications. Inside other applications, in the first column, 6th row, called Q site. Click on that. Got it?
Yes, sir.
Yes, sir.
I hope all of you are able to get this to this page. If you're not able to get to this page, please let me know. I'll repeat it. Okay. Otherwise, it is there in the tutorial also. Okay. It is there in the tutorial also. So step by step, you can follow it. Okay. So importing the TPB, setting up the display, and then okay, you can go to the Q site panel. So this is the Q site panel which you are going to do it. Okay, in which you are going to do it. Okay, in the Q site panel, you can see in the top there are 8 different tabs. Right? Do you see that? 8 different tabs. The first tab is called QM settings. So in the QM settings, we are going to set up what are the atoms that are going to be treated in the QM region, and how we are going to pick up that QM region, and also we are going to select the QM basis. When you want to run quantum mechanical calculation, you have to give them the basis set and basis functionals. So we are going to select the basis set and basis functionals for that QM region. Then the second option is called potential. So the potential is we are going to be treating the rest of the molecule which are not going to be treated by QM region by molecular mechanics, right? So for molecular mechanics, we have to define what is the potential for that molecular mechanics, for which we need a force field. we are going to be using OPLS 2005 force field, okay, with a constant dielectric. Okay, so this is to consider that the background effect. So we are going to be using this potential. Again, we don't need to change much in here, this potential part. Then constraints. In case if you want to constrain your residues, I don't want my constraint to be my residues to be relaxed. I want my binding, residues around my binding pocket to be rigid. So then you can use a freeze atoms using the constraints, which is not usually recommended because the molecule should evolve into a real life scenario. So for that we should not apply any constraint. We should let it go. In some rare cases, when there are metal atoms which are coordinated and the metal regions which are highly rigid, so we can apply some rigidity there. Otherwise we can simply ignore this constraint also. Then QM constraints. In case if you want to freeze some of the portions of your molecule which is going to be ligand molecule which is going to be considered for QM region, we can freeze those atoms also. Basically we can freeze their distance, their angle or dihedral. Okay, so in case if there is a molecule, okay, so ligand molecule, in the ligand molecule I don't want this particular angle between three atoms to be not to be changed during the minimization, then you can set it up here in the QM constraints part. Again, these constraints are not always used. In very specific cases we might need to use these constraints, otherwise these are not used in general. Okay, then when we go to minimization method, so how we are going to do that? The parts which are not going to be treated in QM region, the parts which are treated in region to be minimized. So we are going to look for the energy convergence criteria, where we are going to see the energy and the gradient, the energy change between the two next neighbor conformations to be less than 0.01 in the gradient correction. Okay, the energy change should be 0.1 kilocalorie, less than 0.1 kilocalorie. So then when they are in this region, that means the energy is converged. So we are going to stop our minimization. So when it comes to QM region, we can do three different types of calculations. Okay, one, we can do the minimization calculation. That means we are trying to minimize the energy of the QM region as much as possible, QM energy minimization, or single point energy calculation, where we don't want to do the minimization. We simply want to calculate what is the energy of the given ligand molecule in that binding pocket using QM approach. Third, we can do a transition state search. So when you want to do a transition state search, where there is a chemical reaction which is happening in the binding pocket, for example, a covalent docking or covalent binding is happening between the molecule and the protein. So there is a bond which is being created. So then we want to find out what is the energy required for that bond to be created. So we need here some kind of input in these cases when we want to do the transition state search. So either we can do for linear synchronous transition, where we need to give a reactant structure as well as the product structure. That means we have to tell how the reactant would look like before the covalent binding and how the product would look like after the covalent binding. We have to give two structures. Once we give these two structures, the engine would try to optimize the transition state and come back to us with the transition state structure. So why do we need this transition state structure? For every chemical reaction, there is an energy barrier. So we have to find out what is the energy barrier. The energy barrier is within the range in which the reaction can happen. Then this reaction is going to happen, the covalent binding is going to happen. If the energy is really high, that means that reaction is not going to happen. Okay, so to find out whether the reaction is going to happen or not, we can, we have to calculate the transition state. So in case we think this reaction is not going to be a simple linear reaction, this is going to be a very complex reaction. In those cases, we can give our transition state guess also. So we believe this could be the transition state also. So that in that cases, we need a transition state guess. That's called quadratic synchronous transition, QST approach. Okay, in here we have to give a TS guess structure. So when we want to give the TS guess structure, we can discuss with the organic chemist. So here is my amino acid residue, histidine or cysteine, which is interacting with my ligand molecule, and they are forming a chemical bond between them. What would be the transition state structure would look like? So the organic chemist can give you a kind of a schematic diagram. So this is how it would look like. This is how I believe that they are going to react between them. So then we can build up a structure for that one, a TS guess structure, we can provide that guess structure as a guess entry to calculate the transition state structure. So in this case, for the tutorial purpose, we are not going to do the transition state. We are going to do a simple single point energy calculation, because within the context of the time limit of this workshop, you won't be able to run anything else. If you run minimization, it's going to take some time. It's going to take like 4 to 5 hours. Okay, when you run a transition state calculation, it might take even overnight or up to a day. Okay, but when we run a single point energy calculation, that is for the given structure, what is the energy that we want to calculate, so that won't take more than 20 to 30 minutes, which within the framework of this workshop we can complete. Everything else would take really a lot of time, but in real life scenario, we would be running minimization calculations. Okay, but in here, within the context of this workshop, we won't be able to run the minimization. Okay, so that's why we are running a single point energy calculation. Got it?
Hello.
Sorry, come again.
Yeah, sir, what can be interpreted from the minimization calculation, means how it is different from single point calculation?
Yep, single point energy calculation is for the given structure, what is the energy? So that's what it's going to calculate. When we do the minimization, what does it do is it allows the degrees of freedom to be relaxed in the binding packet. Okay, so that means the molecule can slightly tilt or rotate or even change its conformation a little bit, so that it can get into a better position and dock itself into a more comfortable way. For example, you are sitting in front of a teacher, and then when the teacher is gone, you are going to be relaxed, right? The same way, when you are crystallized, you are in a crystallized position. That means you are kind of crystallized in that position, right? You are frozen in that position. So when I give you energy, or when I give you let you go, so how you are going to behave in that binding packet, in that zone, in that empty space, or in that space of local interactions, how you are going to adjust yourselves so that you can have maximum interaction with the protein. So that kind of small relaxations are going to be allowed during the minimization, because of which we are going to calculate what is the exact interaction energy there. Okay, which is going to be very, very key in case if you have a metal atom in the binding packet's vicinity.
Is this method can be used to refine the cryo-EM structures where the metal ions are there? Means minimization. Yep. Okay.
Yes. Well, if you have a right kind of PDB structures, yes, we can do that also. Yes. Okay. So I'm going to show you a paper here. Okay. So I'm going to just show you a paper. Okay. I believe this paper. So here is a paper. Okay. QMMM docking and its applications to metalloproteins. So here is a list of metalloproteins. Okay. So you can see, so these are all the different docked ligands. Okay, all these docked ligands are having one or many metal atoms close in their vicinity. You can see. So here, so this is the docked pose. This is the pose which is coming from QMM. You can see the bending here, right?
Yeah.
So this kind of relaxation would happen when you allow them to do the minimization. When you do a docking and do the single point energy calculation, you would get the energy of this docked pose only. Or you can take a crystal pose, you can try to do a calculation only for the given structure. But when you are doing the docking, maybe they might even relax like this, right?
Yeah.
Because of metal atoms attraction. So this kind of things has to be captured. For that, we need to do the minimization.
Can we use to minimize our docked means like I have prepared some metal complex, means ligand and metal complex. And after glide docking, I'm thinking that the pose which I'm getting is not very satisfactory. Again means I can refine my structure using this Q-site minimization method.
Exactly. So in that kind of situation, only we have to use this one. So in that kind of situations, okay, if you are using glide, so glide tend to produce, with the XP docking, you should be able to produce very close to what you're going to get from QMMM. In case if you believe that is not a right one, so what you can do is there are two other approaches after that which you can take. There is something called quantum polarized ligand docking. That's before QMMM. Okay, so if you do quantum polarized ligand docking, what it would do is it would try to re-optimize only the partial charges in the ligand, so that it can do the right kind of re-docking of that molecule. Okay, so quantum polarized ligand docking. In case even if you are not satisfied with that one, definitely you can do a Q-site approach, QMMM approach, in which you would be able to explain why that is coming out. because you can see this kind of changes also. You can see this is a visible change, right? When you calculate these RMSDs, they are going to be completely different. Okay, so that's why this is going to be key. And they had done this one for really 9 different proteins, which are having completely different metal atoms. You can see, so 4 different metal atoms. They were able to dock it, and then they were able to do the QM dock, as well as they are going to do, they had done the QMMM approach. So with that one, they were able to reproduce a lot of results which are very close to experimental values. Okay, but you can see Glide produces very good results also usually. Okay, but in some cases, when Glide results are kind of not up to the satisfactory level, then in those cases, you can definitely try QMMM approach.
Thank you, sir.
Okay, but in my experience, what I have found is Glide tend to give very good results actually. Okay. In case if they are not giving it, then you can definitely do it. Okay. And also in some cases where people believe there's an electron transfer which is happening, or there's a charge transfer which is happening between the protein and ligand, right? Okay, there was one of the paper in which people wanted to really understand. Okay, so what kind of charge transfer is happening between this group of atoms? So that means you can calculate their properties. What happened? So we haven't set it up yet. So once we set it up, we can set up the properties. So once we set up the QM region, for the setup the atoms in the QM region, we can calculate some properties. What kind of properties we can calculate? We can calculate the HOMO-LUMO orbitals. So how the HOMO-LUMO orbitals are looking like. So in case if you are interested in how these interactions are changing my ligand molecule, so you can calculate ligand molecules HOMO-LUMO on its own outside in the gas phase or in the solution phase when you put it in the water medium. So the same ligand, when it is in the binding pocket, how its the HOMO-LUMO orbitals are changing because of the local environmental changes. Okay, why that is going to be key? If you look at certain drug molecules which are used as fluorescent materials, the materials which are going to emit some kind of light, or the materials which are going to have some kind of specific electronic interactions with the ligands. So in those cases, they want to understand how the HOMO-LUMO energy looks like also. So that can be calculated during the course of QMMM calculation, which is not possible in any docking, which is not possible in any QPLD also. Okay, whereas in QMMM, you can calculate that one. Okay.
Okay, sir.
Yeah. So next, the scan part. In the scan, so in case if you want to put the molecule inside the binding pocket and move it closer to a particular residue or move it away from that particular residue, you can set up the distance or angle or dihedral within the atoms in the QM region. So then you can do the scanning. Okay, when you do the scanning, so that means I'm pushing it closer to that metal atom. So how how close it can go, how the energy changes when I push this ligand closer to the metal atom, or when I push it away from the metal atom, so how it is going to go. So you can reduce or increase the distance between these two atoms, atomic centers, and calculate the energies. The same way you can calculate the angles. So I want a particular group to be rotated. So how this rotation is going to impact my binding energy. So that can be calculated using the scan option. Okay, inside the binding packet. These all things can be done inside the binding packet. But here, for the sake of the workshop, we are going to do simple single point energy calculation. But in case if you believe this is going to be useful tool for you, you can always come back to us. We can give you an extended training. So this is just to show how to set up QMMM calculation, how to visualize the results. Okay, that's all we can do in 2 hours. Okay, so when you want to really understand more deeper understanding for your particular problem set, we can discuss your specific problem set. We can try to see how this particular tool can be very useful for your particular problem set. Okay, but that's not a discussion in a public forum. So that's why I'm offering you, if you feel that this tool is going to be useful for you, you can always come back to us by writing to help@schrodinger.com. Okay.
Thank you, sir.
Yeah, so here's the QM settings. Now in the QM settings, we have to select atoms that are going to be in the QM region. So you can see QM region. So what are the atoms that are going to be in QM region? Okay, so here you can see the drop down. You can select 4 different types of waste the residues or the ligand into the system. Side chain, that means a protein molecule has plenty of amino acids and amino acids have the side chains. You can select only the side chains of these amino acids into the QM region. Or entire residue, that means the entire residue, entire amino acid residue can be selected into QM region. Or free ligand or ion. So when there is a free ligand in the binding pocket, you can select that free ligand into the QM region. Or hydrogen cap. So the hydrogen caps are going to be used when we want to select a region, and that region has to be treated in QM. The rest of the molecule is going to be treated in MM. So when I say the region, it means it includes a ligand and its surrounding. Let's say the first layer which is surrounding the ligand molecule, that's going to be treated using QM. Rest of the molecules are going to be treated MM. But this first layer of amino acids, they are going to be connected to the rest of the molecules, rest of the protein structure through a single bond or through a chemical bond, right? So, but we can't cut down a chemical bond and take the molecules alone or the fragments alone into the QM region. So in order to avoid that one, what we do is a hydrogen capping. So we can cap that single bond by capping a hydrogen. That means, so we are going to put a dummy hydrogen atom by breaking that bond, so that we can treat the ones which are close to the protein, ligand atoms by QM region, by applying the QM method. Rest of the molecules are going to be treated in the MM method. So for that, we can use the hydrogen cap approach. In this case, we are going to use free ligand approach. So where we are going to select the free ligand in the interface and run that free ligand in the QM region. So now I selected the free ligand approach. Select the free ligand. By default, I think it should be the free ligand. Once you select the free ligand, you can check the box pick. Okay, so if you check the box, the pick, now you come into the interface, click on this blue molecule. You can see it gets highlighted with the red dots. So once it gets highlighted with the red dots, that means it's by default selected here. You can see. So it's a ligand which is having 16 atoms. So now that is going to be treated in the QM region.
Sir, could you please repeat this part?
Yeah. So I'm going to repeat this part. Okay. So in the QM region, now there are 4 ways to select the fragments which are going to be treated in the QM region. So I selected the free ligand option. Then you can check on this pick box. That means we are going to pick it in the interface. So once I select the pick or not tick mark, come inside the interface, click on that blue molecule. You can see that is going to be highlighted with a red color dots. That means this molecule is selected now for the QM region. If I go back into the Q side panel, you can see, so that is already selected. So here if I look at it, 16 atoms which are going to be treated in the QM region. Okay?
Yes, sir.
Yeah, okay. So now I'm not only interested in this ligand, I'm interested in the residues nearby also. So how do I select the residues nearby? You can see we can select the side chains only, or we can select the entire residues. Okay, let's change it to side chain first. Let's click on the side chain, go back in there. So we can click on this side chain. You can see, so this residue side chain is now selected. Do you see? There are these 5 atoms which are selected, right? So that's the side chain of this particular amino acid residue. So that is selected for the QM region. If we go back into the Q state, sorry, come again.
How to select the side chain? Can you repeat what?
I'm going to repeat it. Okay. So let's select the side chain. Here you say I select change it from the free ligand to side chain. Again, check the tick box. Come back into the interface. Click on the side chain that is closer to the amino acid residue. Okay, here is a serine residue. In this serine residue, I picked on this side chain, so it picked the side chain. So if I go back into the Q side panel, you can see the side chain is selected. So I deleted my free ligand. Let me go back and pick up the free ligand also. Okay, so you can see, so the free ligand and the side chain, both of them are selected. So if I want to select the entire residue, okay, so how do I select the entire residue? Change it to entire residue.
Sir.
Yes, ma'am.
Sir, in my case, a side chain of, I think, lysine has been selected. So how do I delete that and select the correct one?
Well, it's okay, ma. It doesn't matter. Like, this lysine selector, you can select the lysine, not a problem. Okay. You can there's a delete all button here or delete. So you can select that entry, just delete it. It will delete it.
Okay.
Okay. So if you check the ligand, delete, it would delete it also. Okay. So you can select it. So now I'm going back and selecting the ligand. Okay. And then go and select the side chain. You can pick up any side chain, doesn't matter. Okay. So now let's pick up again the same one. Okay. So here's the side chain which is selected. Okay. So the same way you can go back and change it to entire residue. Come back in here and click on the residue. Oops, okay, it's already exists in the interface. Okay, so here I selected another residue. You can see, so this particular residue is been entirely selected now. If I go back to Q side, you can see, so these are the entire residue. Okay, 12, 13, 14. So that particular side chain is been selected. Okay, so entire residue is selected, then side chain of the cerium is selected, ligand 2 is selected. So total I have 47 atoms in my QM region. Okay, entire residue. So we don't need the entire residue because it's going to, you increase the number of atoms in the quantum region, it's going to delay your calculations. Why? Because in the QM region, we are going to solve each and every electron in the system. So once we increase more number of atoms, it's going to take some time. But yeah, we can go up to 100 atoms also. Again, for the time period, if you want to close the session at right time, we should not select more atoms. Let's stick to 47 atoms. I think it should be fine. 47 atoms for a single point energy calculation. Then we can visualize the results. Okay, so this is our QM region. Are you able to select a QM region by yourself? If not,
sir, could you wait a minute? I am trying. Sir.
Yeah, please take your time.
Actually, I've selected some other residue, and the total number of atoms showing is 29.
Not a problem, ma. So lower is good. Okay, that means you will be able to complete. Okay, so again, this is a test case, just a test scenario. In real life scenario, so what you should do in actual case, in your case, in your project, what you should do is, so there are 3 scenarios. First, if the metal atom is present close to your binding pocket, in that case, you have to select your ligand and at least the residues which are connected to the metal atom, and the metal atom supposed to be in QM region. Okay, you can go through the paper which I showed you. Okay, you can go through Schrödinger website in the Q site page. You should be able to find that paper. So that paper explains why we should treat that area into QM region. Okay, so that should be treated for the metal when you have metalloproteins, or when you have a binding pocket of all the amino acid residues, only the ligand is there, and I want to calculate, do the QM amount to check that my binding affinity is very good or very close to what I want, what I'm expecting. Then in those cases, you can treat only ligand alone. You don't need to take any residues. So ligand alone is going to be treated in QM region, and then let's see how it changes. Then the third thing, you can take your ligand alone and the side chains which are coming from the nearby residues. That's the third case scenario. Okay, if you take the entire residues which are surrounding the ligand, it would be more than 100 atoms, but that would take really a lot of time, days of simulation. But what kind of gain we are going to get from it? Sometimes it would be useful, but more often it's not going to be really that useful. Again, those things are being clearly explained in the paper. So when you take only the metal atom groups, when you take the entire residue, how it is changing. Okay, how the orientations are changing, and those things are explained. So you can check that paper, read that paper. That would give you a clear-cut idea what you should or what you should not use for your QM region. Okay. In case if you face any trouble in selecting this kind of regions, you can always again come back to us. We can discuss it also. That's the only paper which I showed you, but there are plenty of other papers which show how to select the QM region.
Sir, what criteria should we follow while selecting the residues or selecting the side chains? For say, like I need to perform transition state search, and I know that what are the residues will be involving in the reaction.
So okay, so that's a good question. That's a very good question. In case if you are running a transition state calculation, okay, so then you definitely must include the residue which is going to react with your ligand, right? So you have to take the residue and the ligand into the QM region for sure. Okay, so that first you optimize it. That is going to be your reactant structure. Then you have to optimize your product structure also. So when you see there is a chemical reaction, you can make a bond between your ligand and the residue. You can optimize that also with a bond. So that is going to be your product structure. So once you have the reactant structure optimized and the product structure optimized, you can use the transition state optimization method. You can see, you can go back to the transition state optimization method. You can select LST or QST. In case if you have transition state case, you can provide both entries, reactant entry as well as the product entry.
Sir, optimize, I did not understand. Optimize.
So optimize in the sense minimization. Okay. So
means first I need to minimize each complex initially using Q side. Then I will go for the second step will be my transition state, sir.
Yes. Okay. So you have to minimize both reactant structure as well as the product structure. That means these two are already done. So here are the entries for the reactant and the product. So now tell me how the reactant is transforming from reactant to the product. So that's what the transition state search would do.
Yeah, so this is the scenario when I know which are the residues which are involving in the reaction. Now in second scenario, I don't know anything about what are the residues involving.
But you are sure that there is going to be covalent docking, there is covalent interaction happening?
Yeah. Means I'm sure that there is some kind of reaction and there is some covalent bond formation. In that scenario,
that case it's a guesswork, right? Because we have to see what are the residues which are there in the vicinity of your binding pocket. Okay, so vicinity of your ligand, and in ligand, which is the most active group that is potentially going to involve in a reaction. Okay, so that can be done. So again, that can be done using a QM calculation. So when you do a QM calculation, we can find out what is the most active group in my ligand molecule. So then we can find out which is the nearest neighbor residue for the most active group in my ligand. It can't react to residue which is far away, right?
I need to make a possible reaction guesses. What kind of reaction can be happen inside that binding side with that ligand. And then multiple permutation and combination I need to try with the LST or
it won't be multiple permutation and combinations. Okay, if you look at amino acids, okay, not all amino acids are to be involved in covalent bonding. Okay.
Yeah.
So out of 20, there are 3 or 4 hardly they are going to be involving in covalent bonding. Okay, so you have to see whether any of 3 or 4 amino acids are in my vicinity of the binding pocket. First thing. Second thing, what is the most active site in my ligand? Whether this ligand is closer to any of that amino acid, that's what second you have to find out. Third, if they are finding, even if you are finding it in your all different binding modes, XP, SP, you can do a detailed XP docking, and then you can see what I mean by XP docking in case if you are not using glide. So that means it's an extra precision mode docking. That means you can have multiple different orientations within the binding pocket. So within that orientations, any of the such orientation is facing in the right orientation for a chemical reaction to occur or not. That can be also calculated. In those scenarios, you can pick up that particular conformation and go for a chemical reaction. So you can optimize that as a reactant, and you can make a chemical reaction. Then you consider that as a product. Then you have the reactant structure and product structure optimized. From both these two structures, you can try to get the transition state structure.
Okay.
Okay.
Thank you.
Yeah, that's how it has been done. Okay. So now we are going to go back to single point energy. So now we have set up the QM region. Okay. So QM region has been selected. So then there are QM parameters that has to be set up. Okay. So when we say OPLS force field, that's going to be taking care of our MM part. When it comes to QM region, there are 2 things that we have to select. One, quantum basis set, and then quantum mechanical basis functional. So here you can see the quantum mechanical method, which is DFT B3LYP, right? So B3LYP tend to produce good calculations for the structural information. Okay, so B3LYP tend to produce good calculations for the structural information. So you can see, so there are different methods which are available. Hartree-Fock, MP2, DFT, MO6. MO6 tend to produce good energetical calculation, energetical values. Okay, so there are semi-empirical methods also which are available. So semi-empirical methods are less accurate compared to the And DFT methods, you can see Hartree-Fock is also available, but the DFT methods are most generally used QM methods. Okay, when we say quantum mechanics has multiple methods for solving molecular systems. One of them is Hartree-Fock, second one is DFT, third one is MP2. Okay, so in the order of accuracy, MP2 is going to be more accurate for the linear systems. The moment it comes to a ring systems, it's going to be as equal to the DFT. Then DFT is going to be the second accurate method, and third it's going to be Hartree-Fock. But if you look at computational wise, MP2 is very expensive, DFT is less expensive compared to MP2, Hartree-Fock is as expensive as DFT, but it gives better results. That's why about 99.9% of the time people use DFT. Okay, so quantum mechanical calculations by default becomes synonym to DFT, but DFT is not only the quantum mechanical method, DFT is one of the quantum mechanical method. Okay, so that's why people use DFT by default in most of the cases. So we are going to do the same. So DFT, B3LYP, which is kind of very good basis set for doing calculations. Okay, and then you can see QM basis. So when you click on QM basis, you can see by default it selects LACVP star. Why it selects LACVP? As I told you, in most cases we come to QMMM when we have metal atoms in the vicinity. So when you have metal atoms in Schrodinger, we use LACVP as our basis set. When you have metal atoms, in this case, particular example case, we don't have a metal atom, but we have phosphorus in there, which is not very well treated in 631G. So that's why we are going to use this LACVP basis set. So if you want to change this basis set, you can see once you select it, this particular basis set option is highlighted. Okay, you can see here. So you can drop down, you can change it to anything you want, basis set. So what is basis set? If you want to understand that one, please do join us for our other workshops where we would be teaching you what is basis set, what is basis functional, and how to do a quantum mechanical calculation. Okay, so that's again a detailed lecture for 1 hour, which I can't complete in here. But to understand this one, simply in one statement, this basis set is going to represent our molecular orbitals in the right way, so that we can calculate the energy of the system in the right way. So that's why we need to select this basis set. Okay, so here we have LACVP as our basis set, which is okay. We don't need to change anything. Okay, so now we have set up our QM region. The rest of the things are going to be treated in MM region. We are going to run a single point energy calculation. If you go back to properties now, you can see some of the properties are being opened up now. If you go to surfaces, molecular orbitals, you can calculate the homolume orbitals. Okay, you can calculate the homolume orbitals. So if you want to calculate the homolume orbitals, you can calculate it. There is this atomic electrostatic potential charges, which would calculate partial charges of each and every atom in the QM region. You know, recalculate the ESP charges using the quantum mechanical approach. So which would also be another quantity which would be of interest when you have metal atoms in the vicinity. So once you calculate that one, that would be a good comparison between your docking and QMMM method. You can even visualize them in a contour way. So that would look really a beautiful picture to explain what is happening in the binding packet for the ligand. So that would be a good thing to calculate. But now we are going to calculate these properties again, considering the time factor. Okay. Once you have selected these two options, go to this job settings. So which is next to this run button. There is this tooth wheel, mechanic wheel. Click on that one. There is a job settings button. So in this job settings button, don't submit it on localhost. So if you drop down, there should be a CPU core which should be available to you. CPU core which should be available to you. So put it in the CPU core, give it 4 CPUs and give run button, please. Okay.
Hello,
so
sir.
Don't run it on the local host. If you drop down, there should be a CPU which is available to you. Okay, so I have a big list here, but you should be having only one other entry. Okay.
So how did you reach the job settings?
Yeah, so there is a mechanic wheel next to the run button. Do you see that? In the bottom right hand corner, there's a run button. Next to the run button, there's a drop down below next to that mechanic wheel. If you click on that mechanic wheel, there's the job settings option.
Yeah, got it, sir.
Yeah, so next drop down below the local host, you will be having a CPU, queue system. Just check the CPU queue system, give 4 processors, and then click the run button. That's how you launch your first QMMM job.
Hello, sir.
Yes, ma'am.
Sir, in QM settings, you have the charge 1, where it is showing in my computer that the charge is 0.
Yeah, because of the residues which I selected. Okay, so the residues which I selected, so one of the residue has some one charge, plus one charge. Okay, so in your case, you might have selected a different residue. So that's why those residues not having any charge, that should be 0. Okay, ma, it's depending on, you have expected same residues as me?
No, sir.
Yeah, that's why, ma. So one of the residues here has a plus one charge.
Okay. Can you show which of the properties we need to calculate?
Well, we are not calculating any properties now, but if you want to calculate, you can submit it, but it's going to take at least a couple of hours to complete it. As I said, your machine is going to be shut down at 9 o'clock. So you have to sit in front of the computer after maybe 1 hour or so to visualize your results. Okay, so you can calculate these two properties. Surfaces, homolumo surfaces. Yes, ma'am.
Sir, I pressed the run button and then it loaded for a while and then it stopped. So how do I know it's still running?
Okay,
or?
I'll come back to that one. I'll come back to you in a minute. Okay, so let me complete this one. So the surfaces, once you click on the surfaces in the top, you have to select the molecular orbitals in the second half. You can see the molecular orbitals. And the second property would be of interest is atomic electrostatic potential charges. These two you can calculate it. If you have patience to wait for 2 hours for your results to come out, so then you can select those two and run it. So once you run it, then it's going to, as I said, it's going to rotate here for some time, and then it's going to launch it. So once it launches it, you can come back into the interface. You can click on the jobs menu, which is on the right hand top corner next to the tasks. Okay, click on the monitor, then you should be able to see the job which is running. Okay, so in this case I have to launch it. So if I launch it,
sir, in CPU section it is showing 1. So should I?
You can change it to 4, you can change it to 4.
Okay.
You can change it to 4 so that it can go faster. We have enough CPUs available. You can see once you come here, then it should be showing running. So that means QSET calculation is running now. So it would take, as I said, if you have selected properties, it would take for hours, couple of hours. If you haven't selected any properties, it would complete it in half an hour. Okay, but we don't have half an hour to complete. Wait for the job to complete. Instead, what we can do is we can visualize the output right now itself. All of you are able to submit the job already?
Sir, it is saying waiting.
Waiting in. So did you put it in the local host? You should not put it in the local host.
Yes.
No, sir. We put it in CPU and it's showing that waiting.
Well, probably it would go in a minute, because it has to check for the CPUs and then it should go in a minute.
Yeah, it's running now.
It's running now, yeah, because it has to find a server, right? This is a cloud-based computer. So the cloud-based computer are scattered across the continents. So it has to find a CPU where it can run the calculation. So maybe you are launching the job in New York. The calculation is running somewhere in Japan, in the cloud. Okay, so that's why it's taking time.
So, sir, I have one more question. Hello.
Yes, please go ahead. Yeah.
So like in case of covalently bound inhibitor, whenever I am selecting the ligand actually, so it is showing me that means it is a complete, it is selecting the complete protein. So in that case, what we need to select means what option I need to select there.
Coming in, what is the exact question here?
Means whenever the ligand is bound to protein covalently and I am selecting that ligand, no, so it is considering complete protein and ligand complex as a one system.
So your ligand is a amino acid?
No, it's a protein only. No, sorry, it's a small molecule only, but it is covalently bound with the protein.
Okay, so in that case, if you want that to be treated as a separate ligand, so what you can do is the bond which is there between your ligand and the protein, you can convert that into a zero order bond.
Oh, okay.
Okay, so instead of a covalent bond, consider that into a zero order bond, convert that into a zero order bond, then your molecule would look like a separate molecule.
Okay, then I can use the option this free ligand or /ion.
Yes, you can pick it up. Yes.
Or in the another case, if I don't want to convert that into the zero bond order, and I just want to consider
what happens in a quantum. So that's why we need an understanding of what happens in a quantum mechanical calculation.
Yeah.
So when you do a quantum mechanical calculation, whatever the bond order which you are giving, it doesn't matter. Okay. So what it's going to do is it's going to calculate the minimal distance, or what would be the best distance between these two atoms, which are having this kind of electrostatic charges, which are having this kind of electron cloud around it, each atom. Okay, so then it connects these two atoms, makes the bond order by itself.
Okay.
Though you call it a zero order bond, it would automatically put it back as a covalent bond once you've done the quantum mechanical calculation. Or if you don't want to change it, okay, so let's go to that approach. So I don't want to change it at all. So in that case, if your ligand is bound to that particular residue, anyway you are going to consider that residue for your quantum mechanical calculation, right?
Yeah.
So once you have selected that residue, the entire residue for your quantum mechanical calculation, it will automatically take your ligand and the residue for the QM region.
Okay. So in that case, what option I need to select? Entire residue.
So you have to select the residue, entire residue. You click on that residue, it will select both the ligand and the residue.
Okay.
Okay. Yeah. So that's the second option. So in case we have such kind of molecules, you can try to set up the calculation now here itself. You have 3 more hours to try out those options. Okay.
Okay.
Yeah. So that's the thing. So what the output would look like. Okay. So here's the output would look like. How the output would look like. Okay. So here we have the HOMO-LUMO calculation which has been done. Okay. For the given molecular structure, we can go and apply labels for scale charges. Yes, ma'am.
So it is showing that one job has been incorporated, and it's showing 2 options: view in project and queue site. So
yeah, view in project. So we are going to view it in project here. So that's the output. So now, so this is the simplest of calculation which we ran here. So that's why it's not really much of a change which you see here. Okay, you can see. So this is how it looks like. So I just activated the partial charges. Okay, so you can just switch off that option also by just clicking this button. Okay.
Sir, how did you open the output? One job has been completed, it is showing. After that?
So just wait for it, it would incorporate automatically into the interface.
Okay, in progress it is just showing completed now.
Yeah, just double click on the completed job, it would automatically incorporate into the interface.
Okay.
Just double click on the completed one, it would automatically incorporate. This will take a minute to incorporate it, it would incorporate it in a minute.
So sir, it will be incorporated automatically?
Yes. You give it a minute, yes.
Yes, it has been incorporated, and now view in project.
Yeah, so it's you can select the incorporated entry. So you can select the incorporated entry. So if we have selected the properties, it would show the properties. Otherwise, I can look at the properties. So, but this is how it is now orienting. Okay. So after quantum mechanical calculation, so this is how. Did it change anything? What we can do is just we can visualize it together. So if you include the input and the output together, okay. So you should be able to see some small difference. Let me change it to this way. Yeah, so that's the entry in this. This is the output. Let's change the color to red. So you can see that there is not much of a change between the input and output here, because this is a crystallized ligand. So it basically gives the same kind of interactions between the reactant and the product. In this case, it's the simplest of example, but in actual cases, you would be seeing small differences in the distances and the angles, and you would be able to see what kind of interaction changes which are coming into play because of the QM optimization. Okay, in this case we picked up a smallest of example that is possible, so there is not much of a change between reactant and the product. Okay,
Hello,
so yes, ma'am.
So it's showing that your job QSite 1 has been incorporated. In next line, it's saying that view in project and QSite. Yes. So what should I select?
View in project. So when you view in project, you should get a new entry in your project here, right then, here. Okay. Click on that entry, select that entry. You should be able to visualize it, the output.
So, like how to select the entry?
So just click this blue color button, ma. This blue button on that entry.
So I'm clicking, but it's not doing anything.
Once you click it, it should show the different structure in the interface. In this case, we did a single point energy, right? We didn't do the optimization. We did a single point energy. So basically, we didn't change the structure. So if you did the optimization, what would happen is it would change its structure a bit. Slowly, it will change its structure a bit. So we will have different interactions. In this case, we have 1, 2, 3, 4, 5 hydrogen bonding interactions. Actually, if we minimize it, energy minimize it, we would have one more interaction actually. Okay, we didn't do the minimization, we did a single point energy calculation. What you can try is in the next 3 hours, you can run the same job with a minimization option. Okay, so in here in the Q side, we have the same settings. Don't change anything else. Just go to the QM optimization, change it from single point to minimization, run it. It would run for 2 hours. By 8:30 it would complete it. So you can visualize your results. What you would be seeing is the number of hydrogen bonding interaction changing between your reactant and the optimized structure. Between your initial structure and the optimized structure. Okay, so here we simply did the single point energy calculation, but if you do a minimization calculation, you should be able to see a change in your ligand orientation a little bit, and which would consequently change the number of interactions with the neighboring residues. So how do I visualize the neighboring residues interaction? Just click on this bottom right hand corner. There is this match stick kind of button. If you click that button, it would show you the interactions.
So can you once again tell how to incorporate?
Yeah, so if you have the monitor button open, so if it says completed, finished, just double click on that link. It would say incorporated finished. That would create a new entry into your project.
Okay, sir. Okay.
Okay, so once you have that entry, we have done a single point energy calculation. That's why there is no difference between our reactant and our input structure and the output structure. Okay, if we have a right kind of output structure, which we have actually, which we can import also, file, import structures. So if you remember the ones which we copied, there are 2 structures, right? One TPB and then Q site 1 TPB. So you can import it. So this is the Q site 1 TPB structure. So that is the output structure. So here is the, just for the sake of clarity, they have shown only this entry. We can create this entry also by ourselves by rerunning the calculation without single point. Just change it to minimization and rerun it. It would run for 2 hours. So by the time 8:30, this should be able to complete the minimization job. So once it's done the minimization job, you can visualize it. So this difference would appear between the input structure and the output structure. So the molecule would be more relaxed in the binding pocket. You would be having more hydrogen bonding interactions. How would we visualize the hydrogen bonding interactions? Right hand bottom corner, click on this matchstick kind of button, which is the second button from the right hand corner. It would be able to show you the hydrogen bonding interactions. Okay, so this is how. Sorry, come again.
Yeah, how many iterations should we consider while we are going for the minimization?
Well, by default, it is set to 100 iterations, but normally this structure would optimize it in maybe 8 to 12 iterations. But if you have no metal atoms and all, keep it to default to 100 iterations. Sometimes it can go up to 70, 80 iterations also, because when you have this metal atoms and this time sometimes it takes really fine tune to get to the right kind of optimized structure. By keeping it to 100, you are not doing anything. Basically, if it converges in 10, it's going to save it in 10. Stop it in 10. The maximum iterations which you can take is 100, but it's not necessary. It's going to take all the 100. As soon as it's optimized, or as soon as it's minimized, it's going to stop the calculation. So setting it to default 100 is not going to impact your calculation time.
Okay. And one more thing, like after doing the single point energy calculation, can we interpret the single, like I have 4 different ligands bound with this one single protein?
Yeah.
Okay. But I was able to rank all those compound 1, 2, 3, 4. Can I use the single point energy calculation to do that particular ranking?
Yes, what we can do is, okay, so there should be a strategy here. So the strategy is, when I have these 4 ligands in the solution phase, okay, when I put these 4 molecules in water, how their charges are distributed, okay, how their charges are distributed. And now I have these 4 molecules in the binding pocket, okay, so I docked them using Schrödinger's docking tool or glide. I docked them. So now I took the dock pose, I'm doing the single point energy calculation, and I calculate the partial charges on them again. Okay, so when I have the partial charges calculated, so then now how the partial charges distribution is changing. So that would tell you a story of whether the molecules are getting more comfortable in the binding pocket or the molecules are not getting comfortable in the binding pocket. For example, if there is a more negative atom in your molecule, so that is getting less negative now. That means it's donating that excess electron to the protein structure. That means what it does is it's offloading its stress. It's offloading its excess energy, right? Excess potential. So if it is offloading, I'm going to be very happy because I was so heavy. Now I'm losing my weight. So that means I'm going to be comfortable. Instead, okay, if there is a positive part which is taking up the electron from the protein and it's becoming less positive, that's also welcome. But if the negative is getting more negative or positive is getting more positive in the binding pocket, that means that molecule is not going to be liked in that binding pocket. Because I am already having a burden, now my burden is further increasing because of being here in the binding pocket. Maybe some other part of the molecule is getting benefited, but that particular part of the molecule is not getting benefited. So based on these values, you can really tell the story and you can really rank order them. So in which order they are going to be preferred in the binding pocket.
Okay, so means I can correlate this loading and offloading of this partial charges with my activity data. I have.
You can really extrapolate it to that level. Yes.
Okay. So from how we will calculate this partial charges?
Well, that's there in the properties. As I told you, if you go to the properties, okay, so atomic electrostatic potential charges, this ESP charges, are the partial charges. Yeah, these are the partial charges.
And okay, then I will get these charges in my project table.
Yeah. So here, see, I selected this molecule. Go to style, apply labels. Oops. So not on this one. This one on this output. You can see. So these are the partial charges. Okay, split by a ligand and water. So here's a ligand. Okay, so here are the partial charges. We can go at a surface and then manage. So again, I'm doing it quickly. Don't worry about it. Okay, so you can show it like this. Okay, so how it was in the gas phase, how it had changed. This charge distribution changed in the binding pocket. So you can compare them. You can see.
Okay, means in presence of binding site and in presence
when it is the binding pocket, here is the charge distribution. When it's on its own, here is the charge distribution. So the charge distribution is getting more distributed or more favorable in the binding pocket or less favorable in the binding pocket. Because of that, I'm telling this molecule is going to be liked in the binding pocket or this molecule is not going to be liked in the binding pocket or less liked in the binding pocket.
Okay. Okay, means we can explain the thing like if I have a congenering series, why one particular molecule is more active and why one particular molecule is more.
Actually, we usually tell them from the interactions. It's not only the interactions, from the partial charges also we can tell the story. Yeah. Because the interactions are going to change the partial charges. Yeah.
Okay. Thank you, sir.
Yeah. So with that one, we are going to stop the session. I believe it was useful for you.
Sir?
Yeah.
So in the entire protein, how did you make the ligand part so prominent? Like previously you had it in a sky blue color.
Yeah. Okay.
How would we know that the output structure has relaxed?
Yeah, just I selected the molecule. I selected that molecule. I clicked on the molecule. I selected the molecule. Went to the style and changed its color. You can see I change it to back to blue color. I can change it to any color I want.
So we can select any like fragment. I cannot select the ligand as a whole. It is only selecting like a bond or something.
Yeah, if you want to select the bond, so you have to press B in your keyboard. Or you can go here. You can change it to atoms. So then, if you select B, I think, well, if you click on the bond, it would select the bond. You can see I selected the bond. So that bond, I can go and change it. See these two bonds. I can go back and change it and change it to red color. So the bond is red in color now.
No, but sir, I cannot select the entire ligand at once.
Yeah, if you want to select the entire ligand at once, go to this option, atoms, change it to molecule. So once it shows M here,
yeah.
Then once on this atom, it will select the entire molecule.
Okay. Yes, sir. Now it has selected.
Yeah. Okay.
Okay.
Okay, so with that one, I'm going to take a leave. If you have more questions, I'll be glad to answer the more questions. But you can use the instances till 9 o'clock. Okay, if you want to practice something, please go ahead and make use of it.
Sir, if I do the minimization, I should be able to select vibration frequencies also, right?
Well, actually, in the binding packet, the vibrational frequencies are not yet activated because it's going to be expensive.
Okay.
Okay, so you can do it on alone, but when you are in the binding packet, well, I wouldn't recommend that one because it's expensive. And I think that option is not even activated.
Yes, that's why I asked. Okay.
Yeah,
okay,
it's computationally expensive. It's very intensive, yeah.
Okay, sir, thank you.
Any other questions?
No, sir.
Thank you. Okay, so with that one, we'll close this particular session. Okay, I'm going to give you my email address in case if you have any further queries, you can send me an email. Okay, so my email address is sudarshan@studinger.com. I'm going to put it right here. So in case if you have any follow-up questions, you can write to me anytime.
Is it possible for you to share this presentation?
This particular presentation, well, I had to get a permission. Let me see.
Okay, if you get the permission, please send us.
Yeah.
Then
so that's my email address. So this is a separate talk on how quantum mechanics is used in drug discovery. Okay, so that probably in another occasion I will give you a complete overview of how it is done. Quantum mechanics in computational drug discovery. So probably in some other occasion I'll speak about this one. Okay.
If you can just scroll this because we have some time. So without explaining, if you can just show for.
Yeah, I can go through quickly. So basically, Schrödinger has many tools, and material science is one of them. So the word material, basically, you can put it behind any word, right? Anything that has mass, anything that occupies space is a material. So in Schrödinger's material science suite, they have different model builders, quantum mechanical engine, machine learning engine, a molecular dynamics engine. Also, we have property-based workflows and a periodic DFT engine. Okay, so people really started to understand that the electronic level matters, even in the drug discovery. So anything that's happening at the macroscopic level can be traced back to the molecular level. So that's the whole fun of doing it. So all these molecular level concepts were started, with this guy, Sir Isaac Newton in the 17th century. So he changed the world at the age of 45, and he discovered these equations, Newton's equations, during a situation like this, like COVID, which we have. There was a second wave of plague at the time during 1680s. So at that time he was quarantined. He was self-quarantined for 3 years, complete isolation. That time he had access only to limited number of books. Using those limited number of books and limited number of scriptures, he was able to derive this beautiful equations which we are using today for doing molecular dynamic simulations. Okay, so with a limited amount of sources, there was no internet, there was nothing, just only he had about 20 odd books and some manuscripts, and on his own, all alone. So all the things which happen in his head, with that one he came up with this kind of theory. That's why he's special. So with him we started with the molecular physics. Then 200 years nobody questions his legacy. Then a band of bandits came along. They explained things happen at the macroscopic levels. Those things can be explained using Newtonian dynamics. But when things are happening at the subatomic level, those things are not able to be explained using Newtonian dynamics. So then came the quantum mechanics. Up to 1926, people proved that they have proven that Newton mechanics is not working, but there is no way to make it work using quantum mechanics. But then this guy came along, 1926, Schrodinger's wave equation was published, and with that one we are able to solve Schrodinger's we are able to solve the molecules using quantum mechanics. So this particular conference is the start of new quantum mechanical theory. So in 1927, it was a private conference organized by a private company called Solvay. So you see 30 people participated in it, out of which 17 are Nobel laureates. It's as crazy as it can get. Okay, so then new quantum theory came into picture. Within next 15 years, people developed multiple methods to solve molecules using quantum mechanical approaches. As I told you, DFT is one of the approach. DFT is not the only approach. There is Hartree-Fock approach, there is Thomas Fermi approach, which actually is the base for a DFT approach. So there are multiple approaches which came into picture, but all these were done using paper and pen. Okay, paper and pen people are using because there were no computers. In 1945, we had first computer, but still no simulation was not possible. In 1964, these two gentlemen, this was a postdoc of this guy. Okay, so these two gentlemen, they came up with a theorem which allowed us to solve molecular levels of density functional theory equations in a very simplified approach with using the one electron approach, the electron gas approach. So because of that, they published this paper in 1965. So the software started to develop. In 1970, the first ever DFT software came into picture. 1990, Schrodinger started our engine, Jaguar engine, which was published, and this guy got a Nobel Prize for developing basis sets in 1998. Now, if you look at it, there are 100 plus quantum mechanical software which are available. 100 plus, okay, yeah, which are available. So using different levels of computational approach, you can really explain different levels of properties. In the molecular level, you do the docking, you do the understanding of the docking properties and stuff like that. But if you want to really explain things at the subatomic level, at the partial charges level, or at the level where you want to understand the metal organic interactions, so you have to come to the quantum mechanical level. Okay, so that's the difference between the simulations and the experiments. In the experiments, people use spectroscopy to explain things that are happening, or microscope to understand things. But in the simulations, we use the electronic structure theory and electronic structure calculations. Using that, we can explain it. Yeah. So molecular mechanics uses Desmond or macro model in Schrodinger. So in quantum mechanics, we use Jaguar. So Jaguar is probably the fastest DFT engine. It has all the possible, things that you want to do with quantum mechanical calculations. Again, this is the fastest engine. Probably you can run very complex molecules in Jaguar compared to any other code which is available out there. Optimizing this kind of molecules in Gaussian, you can't even imagine that one. But here you can really do that one. So what happens in a Jaguar calculation is you draw the structure. Basically, you have to select the basis set and basis function. Then you select the calculation type, you get the properties out of it. You can get the properties out of it. So there is basis set and which has to be selected during the Jaguar calculation. There's a theory which we can use to select this kind of basis set. So simple things you can do it with a smaller basis set. Simple things you can do it with the larger basis set. For the geometry optimization, you can do the larger basis set. Again, functionals, there are different levels of functionals which are possible. So people don't use the LD, local density approximation anymore. This is not used in practical approaches. Nobody publishes this data anymore. But if you look at other functionals, people are still using it. Then you have these gradient corrected ones, which are used in the case of material science when you have metal surfaces or metal systems. This is hybrid, which combination of all the different methods. posteriorly corrected, where when you have non-bonded interactions are involved. In this case, in the case of QMMM, you should use the posteriorly corrected ones, and in the case of charge transfer ones, we have to use this long-range corrected ones. Okay, so when you do molecular docking, you do this one, ligand-protein interactions you are calculating. So, but when you do the ligand-protein interactions, it's only the non-bonded interactions which dominate, right? So you have this pi-pi interaction, pi-cation interaction, or hydrogen bonding interactions, ion dipole interaction, and stuff like that. So these are the ones which are predicted using this force field parameters. But in the force field, these two things, Qi, Qj, when two non-bonded atoms are coming closer to one another, the partial charges that play a crucial role, and the van der Waals interaction which plays a crucial role. So that gives you effectively your binding energy. So OP+3 force field is very good force field. That gives you very good predictions in the binding affinities. But when you look at the partial charges, if you look at compare the different force fields, OP+3 is very good, but DFT is kind of best which you can get. That's what I explained to Dr. Srivastava. So basically, when you do the docking, when you do the QMM calculation, you can calculate the charge distribution. From the charge distribution, you can explain the story. Okay, so that's where it's going to be very important. When you have metalloproteins, it's better to run the QPLD minimum or QMM calculation to get the right kind of docking orientation as well as the delta G calculations. When you have covalent docking, you want to understand how the covalent docking is happening. You can really understand the reaction mechanism, how the reaction mechanism is happening. What is the delta G? So from the delta G, we can really explain what kind of covalent docking is happening and what kind of warheads we can take for nice kind of covalent docking. So when I want to take a fragment, what kind of fragment would give me better reaction at the covalent docking? Okay, when I have a structure-based approach, these things are valid. When I don't have a protein structure, then we use QSAR approach. When you use QSAR approach, we try to calculate the intrinsic properties or the chemical properties based on which we try to find out the biological properties when we don't have the protein structure, right? So, but whereas we can use QM-based descriptors also. People use co-molar energies, which effectively means electron affinity and ionization potential, and also multiple moments, which would tell us which way the molecule is going to be specifically orient, and also electrostatic potential, basically which is the most charged atom in the molecule, which is the less charged atom in the molecule. So using this descriptors, you can also fit a QSAR approach that gives you good results. Okay, with the QM, you can also understand how the molecule is going to behave under different conditions also. Like when I put it in different pH conditions, what is the most reactive side? That is going to be key when we don't know what kind of covalent docking is going to happen. So we want to find out what's the most active reactive side and find the weakest bond. So how this molecule is going to behave or break into pieces. So those things can be understood. And we discussed about this QMMM, Q-site approach. So how the Q-site can be used to understand the interactions between ligand and the protein. So basically, these are the things which you can use, in which you can use quantum mechanics to understand computer-aided drug discovery in a completely new perspective. And this is being used already in a very limited amount of labs, and people really produce some meaningful data. And with this data, they were able to explain things that could not be explained based on normal understandings. So some things which are not able to be explained using simple docking approach, when you have this conjuring series, why it is working, why it is not working. So that kind of scenarios, you can use this kind of approach to provide a completely new insight from a subatomic level context. So that would provide good understanding of materials at the molecular level. So that's the presentation in a quick dirty way. Usually it takes about 45 minutes to complete this presentation, but well, we have to complete it, so that's why I finished it in probably 10 minutes. Okay, but if you need more details, you can write to me in this email address. We can discuss on your specific problem. Okay, if you are interested in some specific problem of yours, you want to have a discussion with me, you can write to me, then we can discuss about it also. Okay, probably not this week, but by next week we can have the discussion. Any other questions?
Thank you so much, Dr. Pandian. The session is open for questions, if any. I hope it was understandable to all of you. And as Dr. Pandian said, that this facility is available till 9 p.m. today, Indian time. So whoever wants, they can submit their job and download the calculation and can interpret the results. And whatever questions you have, you can send it to him. Whenever he will get time, he will reply to you. If you want to interact now, you want to ask something, you are welcome. Anyone have any question?
Hello, sir.
Hello, ma'am.
Sir, actually, I have mailed it to you.
Video in the interaction session.
Yeah, sorry.
Okay.
Yeah, I just stopped sharing, so I can switch on my video. Yeah. Yeah.
No, all the person who are asking the question also, they should turn on the video and ask. Yes.
Oh, okay.
Hello sir, I'm Sanjana Raichaudhary from Naipar, Guwahati. Actually, I have already mailed it. I faced a problem and there I got stuck. So I have mailed it already. Like.
Let me check it. Let me read the mail. Sorry, I have to check my mail. So once I check it, I'll reply to you.
Okay, sir. So since it's available for like 9 p.m. only, so it will be really great if you
quickly reply to you. Yeah, I'll check it out. I'll check it out. If I can reply you quickly, I'll do it. Okay. If not a problem. You can write to us. We will give you an evaluation license also.
Okay. Sir. Thank you, sir.
I think Dr. Hemant, you are in mute, or I don't know, something is happening there.
Any other person is there who want to ask question? Okay, so there are some people who are asking question in the chat box. So one.
Okay, let me check the chat box. Okay.
If I had, yeah, you can also check that question.
Yeah. Will I be still able to practice?
I can read that. So in traditional MD packages, we generally use all bonds constraint or hydrogen bond constraints using Link's algorithm. But it is actually important to run completely unconstrained simulation, or we can use constraint simulation. This is what.
Yeah, actually, okay. Links is not completely constraining everything, because links constrains only the bonds which are in the high frequency. So when you look at the IR intensities or IR frequencies, like what happens, the bond vibrations. The fastest bond vibration is the hydrogen vibration. Okay, so that happens in the time steps of 1 to 2 femtoseconds. That's why in MD simulation we have to keep a time step of 1 or 2 femtoseconds. So when you use 1 or 2 femtoseconds, when you want energy to be conserved or energy to be maintained within the limitations of the temperature setup, the temperature which you're going to set for the simulation, we have to constrain that kind of fast mode vibrations. So that's why we use Link's algorithm to constrain that kind of fast modes. But if you look at carbon-nitrogen, the kind of heavy vibrations, which usually happens at the high frequencies, that means low in energy, so they are going to be not constrained. If you constrain all the bonds, you can really go for a very high simulation times. In those cases, I would suggest when you have polymeric chains and stuff like that, there we can do that kind of things. When you are doing ligand protein interactions, I would suggest all the heavy bonds to be not to be constrained. In Schrodinger, what do we do is we constrain only the bonds which are not high frequency bonds. So we constrain only the bonds which are high frequency. So the low frequency bonds are actually freely allowed to vibrate between the distances between the bonds. Look at the force fields. The force fields were initially developed only for the amino acids in the picture. Okay, so force fields were developed only for the amino acids. But then in the 1970s or 1980s, you can say the small molecules came into picture. Before the 1980s, all the pharma companies were concentrating on peptidomimics. That means all the drug molecules they were trying to develop based on amino acid residues only. So when the small molecules came into picture, people started to realize all the small molecules are not having parameters there because the force fields were developed only for the amino acids. Then they slowly started to diverge from the protein. They started to develop force field parameters for non-amino acids. But still, these are only organic molecules, not organometallic molecules. But when the metal atom is introduced, the polarization introduced by the metal atoms are really huge, which are very, very difficult to parameterize. That's why many force fields which you take, okay, CHARM, AMBER, Schrodinger's, OPLS, they have only limited access to the metal atoms. Why? Because parameterizing them is very, very difficult. Okay, so that's why the moment metal atoms comes into picture, people try to take up some other approach also on top of force field based approach. When you do the docking, so it works, but if you can do on top of that one QPLD or QMMM, that's even better.
Okay, and someone is asking, can we do QMMM for a DNA ligand system? Yeah, of course it can be done. So
of course it can be done. Yes.
Yeah. So, but probably he can do full QM also. So for DNA ligand, based on the facility. One person is asking what is the relation of electrostatic potential in interaction?
Well, okay, so what happens is like it's like when you consider the molecule as a neutral molecule, but still each and every atom in the molecule has its own partial charges, right? Because of the electronegativity differences between the local connections. So when a carbon is attached to chlorine, so chlorine is going to pull the electron from the carbon. So chlorine is going to be negative, carbon is going to be positive. So this slight positive, slight negative. But these small slight differences, these are the ones which lead to the interactions with the neighboring residues. Okay, so when this carbon sees a negatively charged neighbor, it tries to make a bond with them. So they want to share that electron, excess electron in that neighboring atom. Or the chlorine, which is having excess charge now, so that wants to have a bond with a hydrogen atom, which is having a positive charge. So because of that, there is an interaction that is happening between the ligand and the protein. So when the ligand molecule is in the binding pocket, it is going to have different kind of charge distribution compared to the ligand molecule in the solution. Okay, so whether this charge distribution is going to be more favorable inside the binding pocket, or it's going to be more favorable in the solution. That's what we have to find out. If it is more favorable in the solution, that means the molecule is not going to stick to the binding pocket, it's going to come out. If it is more comfortable there, that means it's going to stick to the binding pocket. Okay, so basically you call them interactions, I call them partial charges, that's it. So it's one step lower. So these interactions are named easy to count or easy to look at them. But if you look deeper, basically there is a charge interaction that is happening. A hydrogen bonding interaction is nothing but so hydrogen bonding donors, hydrogen bond acceptors. That means hydrogen bond donors are donating the electron, hydrogen bond acceptors are accepting the electrons. So if you can look at the electronic level directly, so you will be able to explain all the other interactions also in a much more detailed way.
It would have been nice if you could explain NBO analysis, but I think no time now.
Yep.
So I think we will do it in the other time.
Do a separate workshop for that one. We can do a complete quantum mechanics for a drug discovery workshop. We can do that one separately. We can plan it sometime.
Fine, that will be excellent actually. And this time we will plan physically so that we can invite people here in our campus, including you. So that will be very good. Actually, Nidhi is saying that she faced some technical issue and then she want to practice Schrodinger online on workstation. Of course, Nidhi, all this is available on YouTube and the system is available till 9 p.m. Indian time. So you can see whatever you missed, you can see on YouTube and practice. No problem. Till 9 p.m. you can
practice it. In case if you feel that you want to try out more, you can write to us for the evaluation license. So the evaluation license can be provided to the students who are coming with the consent of their supervisors.
Yes.
Okay, so if you come with your supervisor's consent, then we can give you, because we faced some issues where we issued the license to the students, but supervisor didn't like it. So that kind of situation should be avoided. For that matter, you have to come with your supervisor's consent, then we can provide you the evaluation license.
Yes, very good. So if there are no more questions, so thank you so much for your wonderful session. It was very informative, and I am sure that our students and all the participants, they have really enjoyed and learned a lot in QMMM calculations and basically in Schrodinger. And I hope that many of you will be requesting their supervisor to order Schrodinger for their lab work. So thank you so much, and let's meet in the next workshop where we will be doing a detailed 1 or maybe 3 days workshop. We will plan it accordingly. Thank you so much. Thank you so much, Dr. Vania.
Thank you, Dr. Hemant, and thank you for this opportunity. First thing. Second thing, sorry, it was only short workshop. That's why I couldn't able to provide much of a detail. But when we have this next workshop, we will be providing all the complete details, how to set it up, what are the logics behind it, and what would be the analysis part. We can discuss everything in detail. Okay. So in case if you face any research-based problems, do write to us. We can provide you some kind of support also in that front. Thank you. Thank you guys. Thank you.
Thank you so much. Thank you so much. Okay, so this is the end of day 2. Now we will share the link for day 3 to all hands-on participant and invited speaker, and also to all the participants who are going to attend the day 3. Day 3, we are going to have first talk from Professor P.B. Bharatam, who is a well-known medicinal chemist. He is going to talk about CADD in metabolism and drug toxicity. And then it will be a very interesting talk by Dr. Sandeep Kumar Dhanda from USA, who is not going to talk, but he is going to take hands-on also. So be joined by 11:15 for his talk and hands-on. This will be followed by a very good talk. I mean, he is Professor Sam DeBezer from University of Manchester, UK, who is going to explain a bit more research problem of QMMM. Today you have seen how to do QMMM using Schrödinger, and Professor Sam DeBezer will tell that what he has done. He has done so many QMMM calculations, and not only QMMM, he has worked on computational chemistry problem in last 2 decades and have very good profile. So don't miss that. And then we are going to have Jastra hands-on in the afternoon session after Sam's talk. I shared the license to each hands-on participants. They are requested to install this so that you check your email, you go to the details, you are having user ID and password there. You installed it in your computer, and then you do it like you are doing today. After that, it will be a demo for Autodoc and GrowMax. So fully packed day tomorrow. Whoever want to learn Autodoc, whoever want to learn GrowMax, this will be done by our group. And Jastra tutorial will be done by Jastra company. Dr. G. Pillai will be the coordinator there, and he has already provided the license to all of you. So thank you so much for joining today. It was a very good learning day, and now this is time for the break. See you all at 9:30 a.m. tomorrow with Professor P.V. Bharatam's talk. Thank you so much and good evening.